protection technologies. In this year program, I welcome each and every participants. First, I welcome our Honorable Director, Dr. Dipankar Maiti sir. We welcome Dr. Proveen Barma, though he, in spite of his busy schedule, he has given us time to deliver his plenary lecture. I must welcome Dr. Robin Gogoi. I also requested our President, Dr. Panjib Chakraborty, but due to his busy schedule, he is not able to attend this. And I welcome all the respected members as well as our chief editor. Other members are there, like I see, and all the participants. I see Professor B.N. Chakraborty sir is also there. I welcome our guest of honor, Dr. Prakash Raut also is present here. He's the head of the Department of Crop Protection. I welcome Dr. Bach, who is the counselor for Eastern Zone. I welcome Dr. Linka, Dr. Raghu, and all other committee members. And the program has been designed in such a way that we'll first, after inauguration session, we'll listen to Dr. Proveen Barma, who is a professor at Jawaharlal Nehru University. And after that, we'll have 4 sessions: plant host interaction, then microbes in plant health management, and then integrated pest management, and then biotechnological interventions in plant protections. So, next program, please. So now I request our Honorable Secretary of Indian Phytopathology Society, Robin Gogoi, to address the our audiences. And sir, please.
Yeah. Thank you, Dr. Mukherjee. So good morning, everybody. Now this entire northern part of our country is very much cooling and dense fog. This we are covered with the dense fog, and despite of that, we are able to assemble here for this nice cause and greater cause for our academicians, students, and farmers. So at the outset, so I would like to welcome all of you. And before that, the respected Dr. Dipankar Maiti, Director, this National Rice Research Institute, and also renowned plant pathologist, and Dr. Arup Mukherjee, Zonal President, Eastern Chapter of IPS, Dr. Manas Kumar Bag, Zonal Councillor, IPS Eastern Zone. Then from our society publication wing, Dr. B.N. Chakraborty, and also so many renowned plant pathologists, and chief guest of this today's function is Dr. Praveen Verma from JNU. Of course, he was working in NIPGR, but recently he had joined in JNU. So my dear colleagues, plant pathologists, students, friends, and all the viewers from YouTube. So I welcome all of you to this today's journal chapter of Indian Phytopathological Society, Eastern Zone. And about the society, these members are aware about the all activities and how it was established. So it is known for all the members, but for the students who have joined, suppose in MSc program, especially in MSc program and also PhD program, they may not be knowing about the detail about our Indian Phytopathological Society. So this society popularly we spell as IPS. It is not Indian Police Service. It is Phytopathological Society. Of course, very. I used to mention that the way IPS in this Indian police service is a custodian of our country, our civil people. Similarly, so we also would like to claim, Indian Phytopathological Society, IPS, would like to claim that we are also custodian of our farmers. True, we can tackle the problems by managing or addressing the plant pathological problems in the farmers level. So here about the IPS, Indian Phytopathological Society, So the society was established in the year 1947 by this Bala Chandra Bhavani Shankar Munkur. He was the founder, a most respectable person, and that was established in the year 1947 and registered in 1949, means after 2 years. And aims and objectives of the society is there, based on which we are always trying, we are putting our effort to address some of our objectives, like so advances in the cause of mycology and plant pathology. But why this mycology and plant pathology? Initially, the society was established. This mycology was the wider field, and later on other fields were added, like bacteriology, fungal pathology, virology, likewise. So now, even though we are projecting here our main motto to address the issues under mycology and plant pathology, but at present we are addressing entire plant pathology discipline. And also, here we are very proud that this is the third largest society among the plant pathologists in the world, because we are having more than 2,500 members. So most of the more than 2,300 members are life members. Next. And the society is having in a country total 8 zones. Total 8 zones we are having, and in zone wise, this number of these members also distributed. So, and activities, some of the activities of our society, and for any organization, main mirror is the publication. So here, Indian Phytopathology, this is the renowned, fully established journal. Regularly we are publishing, you know the frequency, this is quarterly journal, and it was directly published from IPS, Indian Phytopathology Society, this journal. But for the last 3 years, so we started publishing under the banner of Springer Nature. So that's why now it's this distribution and also publicity is more widening. We hope so it will be improved further. And again, another small publication we have added to it. So this is newsletter of the society in the name of Indian Phytopath News. So this is also quarterly. This is quarterly publication. And the motto of this Indian Phytopath News, since it is a newsletter, so first-hand information we can publicize. And suppose our members are working sincerely and they are able to come out with good findings, so they can flash through this Indian Phytopath News, through this newsletter. And time to time, so regularly again in that newsletter, so some of the achievement and also landmarks, some of the meetings, awards, promotion, obituary we include. But since to begin this Indian Phytopath News, so we initially received and we even published all types of this news information without much editing. But again, we are now planning to go for peer review it, so that all the types of these matters will not find place. And we hope by peer reviewing, the standard of this Indian Phytopath News will also be improved. And also the society uses to publish different types of books. We have long list of publication in the form of books, reports, annual reports, some of the proceedings. That way, you can also propose for publication of any book, but before that, you have to send the request to our executive committee, and after decision only, so your publication will be allowed or not allowed. The decision will be taken by the executive committee. So next, and since we are under society, so regularly we used to hold some of the meetings, symposia, conferences. And so far, Indian Phytopathological Society has organized 7 international conferences and 72 national conferences. And first international conference was held in January 2nd, 1948 at Patna. And last year in 2020, January, this January 16 to 22nd, 2020, we hold the 7th international conference. I think most of you participated in that 7th international conference. And the society is always coming forward to honor, to recognize the good workers who are very prominent or always pioneering work they are doing. So the society is having an initiated total 17 awards. And first award was started in the name of Munkur Memorial Award that was instituted in the year 1962. Next. And I think you are experienced by handling the IPS website. And we are trying to develop this IPS website. Already website was there, it is functioning, but we have upgraded. And many activities you can operate through this website, like online payment for taking membership, and also you can create your subdomain, also registration fee payment, again for some symposia or conferences, online submission of your abstract, and then online election and online access of journal, newsletter, ebooks also can be taken by the members. We have got archives where you can go back to the our collections right from our this IPS history. And this is also another added advantage we are putting for our members, that is online this payment gateway through Paytm. So it is more easier than transferring the money or this fund through internet banking. So it is very easy. Then Indian Phytopathological Society election from this year, election 2020, which will be which is going on in 2021. So already we started through this online mode, of course. So this is the last year of holding this offline mode also parallelly. And for offline, so those members whose proper correct contact ID like email ID and mobile numbers are not available with our society's collections and directory. So for them only we are keeping offline voting. So far till date we have completed one step of election, that is this nomination, and all of you have taken part in nominating your this preferred candidates for final voting. And for now again we have already started main voting. Of course, this ballot paper, final ballot paper for the offline voting we have posted on last 15th of January. And for online voting, so that portal will be functional, will be live from the 1st February, and will be keeping for 28 days, means entire February months will be open for online voting. And by the time we hope the members of this offline voting also will be taking part, their online this ballot will be received by our society office. But interestingly, for your information, since I already mentioned that one step of election we completed, that is nomination. But in nomination, so surprising, we posted 664 ballots. Out of 664 ballots, none of the ballots, these preliminary ballots, came back to society or society office. Very surprising. This may be the reason that the members may not be that much of interested to take part in the preliminary election, that is nomination. But I hope they will be very, very active in taking part in the main election, which for them already we have posted the final ballot paper. So this way we are trying to upgrade our website as well as some of our activities through online mode. And how to take part, how we can successfully vote, all the steps also mentioned last time, and this time also we are this highlighting. So it will be, this time it will be very easy in final voting. So for your information, so there will be only election for the post of president-elect, and also election for zonal president and zonal councillor of northern zone. Out of total 8 zones, rest other 7 zones are this elected unopposed. And award application also we have started from last 15th of January only. No, sorry, 15th of January we started. And those who are interested intend to this contest for this award, so you can just open, and that will also be closed in February 15th. But at the time of applying, when you will be feeding your data, you will be supplementing with your supporting documents, you may face some of the problem. So if you face some of the errors at the time of uploading your documents, so immediately I request all of you who are actually going to apply for awards, immediately you should inform us so that we can rectify the problem, so that you won't face any trouble. It should be hassle-free application of award for the award. And this is the actually first page. First phase of the application, the which award you want to apply, so you can select from the first page itself. And for this purpose also, your email ID and mobile number should be registered in our society. Then only you can proceed further. Next,
Login is mandatory.
login for login purpose.
Login is mandatory.
Login is mandatory for application. And regarding our zonal symposia, so we know that we are having 8 zones. So far, 5 zones completed their symposia, like Southern Zone, then Northeastern Zone, Delhi Zone, and Northern Zone, and Central Zone. And today we are very happy. We are assembling for the symposium, this national symposium, for the Eastern Zone. And this is also mandatory for for any society. So that annual conference we are planning to hold during March 11 to 13, 2021. Means after 1.5 months time there. So this information, this brochure we have flashed in the website. I think you have received this one. And also for offline this voting purpose, and also those members whose proper this electronic contact ID is not available, we have sent them this printed copy of this brochure of national conference. So I hope all of you will be taking part in this national e-conference. And some of the important dates you have to note down. You will get every detail in that brochure. So with this, I'd like to conclude about this brief detail of our society. I once again welcome all the participants and who are present now in this symposia. And I wish all the best to Dr. Arup Mukherjee and Dr. Manas Bhatt for making this function a great success. And this will be very much fruitful from the discourse of 2 days program, today and tomorrow. Thank you.
Thank you very much, sir. Thank you for your briefing about the society and its activities. It's actually this program we would have not dared to conduct without your inspiration, and also from the very beginning, one person is behind everything. He is none but our honorable director, Dr. Dipankar Maiti, who is very active member of Indian Phytopathology Society. And now I request our director, sir, to just address the audiences virtually. Sir. Sir, unmute, sir.
Okay, I'm visible now. I'm audible now.
Yes.
Okay, thank you, Dr. Arup.
Thank you, sir.
A very good morning to one and all. Honorable President of Indian Phytopathological Society, Dr. Pranjit Kumar Chakravarti, and member of ASRB, as you know, who could not attend today due to his preoccupations in ASRB. Today's guest of honor, Professor Praveen Verma, Professor, School of Life Science, Jawaharlal Nehru University. Guest of honor, Dr. P.C. Rath, Head Division of Plant Protection, National Rice Research Institute. Secretary, Indian Phytopathological Society, Dr. Ravin Gogoi. I see also the Professor B.N. Chakravarti on board. President, Eastern Zone Chapter, IPS, Dr. Arun Mukherjee. Office bearers of the IPS. And August delegates of renowned plant pathologists and plant protection experts of the country on board today. It really gives me immense pleasure to learn that the Indian zonal chapter, Eastern Zonal Chapter of the Indian Phytopathological Society, along with National Rice Research Institute, is organizing a national symposium on robust plant protection strategies for sustainable agriculture. As you know, plant protection is a very important strategic component in agriculture to ensure food security. It is estimated that the amount of crop loss due to biotic stresses annually is sufficient to feed about 30 million people globally. So you can understand the importance of this subject. And the area of cultivable land in our country, not only in our country, worldwide, is decreasing every year, and with the increasing population. So we need to increase the productivity sustainably to feed this growing population. Hence, not a single strategy should be unturned to protect our crop from different biotic stresses. In the era of biotechnology and information technology, we have several tools in our hand to combat the crop loss due to biotic stresses. To make the crop protection strategy sustainable, we need to design the crop protection strategy in such a way that it not only ensures proper crop health, but also conserve the healthy environment. At our institution, National Rice Research Institute, we are combining information technology and biotechnology and other conventional technologies to ensure the sustainability in developing crop management strategy known as integrated disease management to form a major component of integrated pest management. Now, in this regard, I must mention that the theme of the today's symposium is rightly fit to the current demand of research. From the list of speakers and their topics of deliberations, I could see that all the modern aspects of plant pathological research have been covered extensively. It is also interesting to see that all the abstracts are selected for the oral presentation. I hope that all the participants will enjoy this 2 days national symposium. I wish that it could have been done in physical mode, but due to COVID-19, as you know, it was not possible. So I wish all the best to the contestants of the Professor M.G. Narasimha Academic Merit Award. Despite having this on a virtual platform, with this new normal mode, I wish all the success of the symposium. So thank you very much. Thank you.
Thank you, sir. Thank you very much for your encouraging word. It would have not been possible without your encouragement and your involvement throughout the planning and programming. And now I request our head of the division, Dr. P.C. Rath, to say a few words to the audiences. Dr. Rath.
Good morning, everybody. Respected President, Indian Phytopathological Society, Division of Plant Pathology, IRA, New Delhi, and member, Agriculture Scientist Recruitment Board, New Delhi, Dr. P.K. Chakravarti, as a President of the inaugural session. Honorable Director, NRRI Kotak, Dr. Dipankar Maiti, as a Chief Guest of the inaugural session, and Dr. Robin Gogoi, Secretary, Indian Phytopathological Society, and President, Indian Phytological Society, Eastern Zone, Dr. Mukherjee, Councillor, Indian Phytopathological Society, Eastern Zone, Dr. Bag, and plant protection scientist participating in the IPS Eastern Zone, Dr. Burma, Professor Praveen Burma, guest of honor in the inaugural session, and all the participants of this symposium. I feel extremely happy that this is the first time Crop Protection Division of ICAR NRI Kotak, led by Dr. Mukherjee and Dr. Bag, is organizing the symposium of this prestigious and high-valued society on the important topic, robust plant protection strategy for sustainable agriculture, in which eminent plant protection scientists and scientists of other related disciplines are participating. I think it is a better platform in which all the participants, researchers, faculty members, students can, having their rich experience, will share their knowledge on plant protection, especially the plant pathological research developments. In this regard, I will tell something about our division, what we are contributing to the plant protection. During last year, during the pandemic year, we are able to produce 6 products from crop protection division. We have 20 scientists, out of which 10 are pathologists, 8 are entomologists, 2 are nematologists, and 1 is agrochemist. So during this period, our scientist, Dr. Basana Gowda, and his team, they have developed the trichocard using the species Japonicum and Chilonis, Trichogramma japonicum and Trichogramma chilonis, and Bracken card using the species Bracken hebator. We have already tested in farmers field, and we have fixed the price, and it is in a commercial mode. So, our pathology friend, Dr. Mukherjee, and his team members, they have developed 3 trichoderma products using species Trichoderma hergenium, Trichoderma erinaceum, and Trichoderma atroviridae. These are used, commercialized as NRI biostimulant. And these are also, price has been fixed and commercialized. During last year, we have generated about 1.8 lakh by selling of these product, trichocards and biostimulants. Besides that, our scientist, Dr. Prabhu Karthikeyan, he has identified the new species causing the sea thrush disease in rice in eastern zone. Our scientist, Dr. Prasanthi, she has identified 6 new parasitoids, out of which two are pupal parasitoid and 4 are larval parasitoid against the leaf holder of the rice. These are some of the achievements in our division have achieved during last year, and this is the contribution of all the scientists led by Dr. Mukherjee, Dr. Prasanthi, Dr. Basanagowda, Dr. Prabhu Karthikeyan. They have done a very good job, and I feel honored to be part of this important symposium, and also offer united thanks to the organizers particularly Dr. Mukherjee and Dr. Bag for giving me opportunity to speak in the inaugural session. Thank you.
Thank you, Rotha sir. So nicely highlighting the achievement of the division as well as for being behind the guiding force always, and all the meetings, whenever we call a meeting, you are the first person to attend that. Thank you very much. Thank you. Thank you for a nice talk.
Arup, please. I beg your pardon because I have to leave the meeting. I have to attend another meeting at 10:30.
Okay, sir. Thank you, sir.
So, Professor Verma and everybody, please. Okay.
Thank you.
Okay.
So, in this continuation of Dr. Rotha's lecture, I want to tell you also that we are involved in biofortified rice breeding, and we have released 2 rice varieties. One is high in protein content, another is high in protein and zinc content. Besides, we have developed many varieties which are climate smart. They are drought tolerant as well as they are resistant to bacterial blight. They are also tolerant to submergence. So these are the different activities. We are also we have a very good team of scientists in crop protection, crop physiology, biochemistry, as well as crop improvement, social science, as well as and then these are the different divisions. We have excellent scientists working. So at present, I really feel honored to invite our guest of honor, Dr. Praveen Barma, to say a few words before his plenary lecture. So Praveen Barma, sir, mic is yours.
Thank you, Dr. Mukherjee. This is indeed my pleasure to be associated with this IPS symposium of this June. And I'm a member of, life member of this IPS for, I think I have started in 2005. So near about 15 years, more than 15 years, I have already spent, and I have delivered many lectures in the different symposium, but this is my first visit to the virtual visit to the IPS East Zone Symposium. So, Honorable the Director, Dr. Dipankar Mehti, and Dr. Rath, and also Dr. B.N. Chakravarti, I can see, and all other members and of this participants and the faculty members as well as the students and researchers in this particular meeting. So I'll be briefly discussing in my lecture today, but right now for the sake of doing something which we can give insights to the IPS and the research, current research whatever is going on in the Indian context, that we have to understand in many regards. That that since I am a person who has completed many things like many genome sequencing of fungal pathogens. And then everybody used to ask that what you will do after this genome sequencing. So now this is a time to come to explore this opportunity that we do have in our background, and with the genomics is really contributing. It is really enormous in this particular field of plant pathology. And the availability of the genomes of many pathogens has given answer to many unanswered questions, and also opened many avenues in this of the research. Particularly, what I can tell you that is for the molecular mechanism of the resistance, and also contributed the novel like markers for rapid detection. That is also one of the important thing that we also have to look forward. And also the breakdown of the resistance nowadays. You must be experiencing all these things that earlier the one pathogen was not, the one resistant variety was, resistant for a particular pathogen, but now because of the new virulent strain, now it has become, susceptible. So this type of, challenges we all are facing right now. Also, we are also facing some of the problem of host jump, that earlier the one pathogen was for, some genus and species. Now it has, jumped to other genus and species. So such type of things are happening. So this is maybe the modern technology can give a good answer in this regard. And my opinion that many people are working in India in this regard, and this is, we all have to get together and work together in this particular field of research, as well as IPS. I can find that IPS may have a bigger role in in this endeavor. Each pathogen has evolved in a different, niche or a particular microenvironment. So it has, all the pathosystem has to be addressed individually. So that's why, that there is no general rule of, the pathogen and their resistance mechanism in different. What we have now come to know after this many years of the modern research. So each pathogen has, suppose there is a X pathogen, we have discovered that, okay, this molecule is, there. But probably, the other pathogen may have a different, even though they are taxonomically close, but they do have a different type of mechanism. So we all have to consider this fact that there is no clear-cut and general rule for pathogens. So that's why, now it is important that how we can encourage our research community that traditionally, doing all these things for many years, and in this case, IPS can take advantage for, using the platform like different, zonal chapters and even the national symposium and international symposium to ignite such type of, the activities in the mind of young researchers in this particular field. And what is the best way, maybe to the diverse research community working with the different or many pathogens, is to develop potential that as fast as possible, because this field is really, fast evolving. So beyond generation of just resources, what I can say that many people, take that, that we have developed some primers, or we have, done the genome sequencing, or, many other things. We have to go beyond that. And the time has come. If you see international publications and international researchers, what people are doing right now is they have gone one step, ahead of all these things. We sooner or later, we all have to adopt the same thing in Indian context. We have to, adopt all these advanced technology for the development of the strategies for successful disease management. And also this will help in to minimize the risk of future disease outbreaks, and also assist the preparation for emerging pathogen outbreaks. Most importantly, the new policies, the new education policies and other things has come, where people have to go for certain, that how we can support our farmers from this knowledge, the modern science knowledge, for this in the terms of the pathogen resistance and the pathogen susceptibility. So all these things, that we all have to consider in near future. So this is, that what I can see from the schedule which has been circulated, that I can see that many, young researchers and many other researchers have, very interesting work that they are actually doing in this particular field. So, there are certain things that we all have to consider, that we all have to, exchange knowledge from each other. So in that case, the IPS can, it can serve as a good platform to where people can come together and certain type of things can be done. So I can suggest the General Secretary, IPS, Dr. Gobay, who is a very good friend of mine, and I can suggest something which we can have, that if you see that American Society of Plant Pathology and other, even the British Society, they do have certain platform in their website. So now, this is new normal. We all are coming together and, discussing our research online. And so such type of platform can be, developed in the on the website, where we all can, discuss. And, sometimes people do hesitate to, to come forward for all these things. Many the young researchers, they come to me and then they ask, they said, like, initially, it has what I could see, in last 15, 20 years, that, initially the classical plant pathologies and the modern, giving the modern technology, there was a huge gap in between. So now the time has come where we have to, come together. We cannot, survive without, the modern science cannot be survived without, the getting the help of the classical plant pathologists for the, the development of or isolation of different strains, different, outbreaks, the pathology and the disease cycle determination, many other things that is also, is going to help us in this regard. So my suggestion is that we should have a sort of discussion forum. Many discussion forums are there. So we can, have a discussion forum where even a young researcher will not hesitate to, come to for this platform, and then we can exchange knowledge. We can, nourish each other in this particular regard. We can gain from the, the other researchers what are the things are going on. Even the each part of our country is having different type of problems in terms of the plant pathogens. So we and even the crops which are growing in the different areas. So we have to, think in locally as well, where, the how we can, develop all these things in to, get exchange of all these things together. So many such type of things that I can share with all of you. And another thing that I do have this thing in mind, that many kits or the modern technology based detection method has been developed by some researcher, which I could see, like some of the publications which are coming every time in the Indian Phytopathology Journal. So we all have to, make a platform. Whoever is publishing such type of things, they should also share in the website along with the sequence and everything, so that, we should have a, such type of exchanges in the future. And we can, any researcher can start this type of research right from the beginning. So these are some of my suggestions, which we all, I am also part and of this society, and I am ready to help every scientist in this particular field. I've associated with many other institutions in past, like IRA. I'm still working, and even the Sugarcane Research Institute of Lucknow. I'm right now doing the sequencing analysis of the Polytotrichum, as well as, the many IIHR also I have involved in those people, and Indian Institute of Plantation Crop Research. There also I am involved in all those things. But many people, that since I am from a university, I extend my, help to all of you, because we do have a certain, capability. So we can, that help the different researcher in the field, not only for the benefit of the the basic research, but also which can be applied to the farmers. And ultimately, we are going to foresee all these things to prevent the outbreak of the different diseases in the futures. So thank you very much for inviting me. Thank you.
Sir, thank you very much. Thank you for your really enlightened speech. And you rightly said that we really have a brainstorming session. We have interaction among all the researchers who are working on plant pathology and the common interest of the farmers. And sir, what I want to say you that actually we plant pathologists who read in plant pathology, classical plant pathology, we are a little bit afraid of to touch your molecular parts. So if a person like you guide us, then yes, we can go miles ahead. Absolutely no problem. And the earlier problem was that we used to tell that it's a lab to land. But now I feel it's time that we should come land or field to lab. That means we should know what the farmers need, and accordingly we have to design our crop and our research. Because it ultimately the stakeholders are none but our poor farmers. So we have to think in that way. And sir, one new area of research has emerged recently, that is pathogenomics.
Yes.
And I know that you are working on that in detail. And sir, I request you that if you kindly guide us to work on that, and if you guide a few of our young scientists as well as older like us also, then we will be benefited from your research, and we are looking forward for active collaboration with you also. And so thank you very much. We all are eagerly waiting for your plenary lecture. But before that, I think now it is the time we should go for Thanksgiving. Dr. Bagh will give the Thanksgiving lecture. After that, we will have tea break, and then we will start 11 o'clock your lecture, sir. We are eagerly waiting for your lecture. Dr. Bagh, please.
So, thank you, Dr. Mukherjee. This is my privilege to give formal vote of thanks. First of all, heartfelt thanks from the Eastern Zone, President Dr. Mukherjee and myself, Councillor Dr. Manas Kumar Bagh, for giving us the good opportunity to conduct this 2 days East Zone Indian Phytopathological Society virtual symposiums. So first of all, another thanks should be given to President Dr. Panjib Chakraborty, Indian Phytopathological Society, Secretary Dr. Robin Gogoi, and other member of the Executive Council of Indian Phytopathological Society for helping us for conducting this program. We are also thankful to our Honorable Director, Dr. Dipankar Maiti, for helping in different way to conduct this program and also supporting for different things to conduct these programs. We are also thankful our HOD, Dr. P.C. Roth, for supporting in each and every cases so that we can conduct this program smoothly. Also, thanks and heartfelt thanks to Dr. Professor Praveen Verma for accepting our invitations for giving the plenary lectures. And also we are thankful to each and every colleague of our crop protection divisions to help us before starting these programs, and we also expect their help and support to conduct to complete this program successful. Thank you all. So please we take 15 minutes breaks for tea. After that, we will present sharply at 11 a.m. to listen our plenary lecture from Professor Praveen Verma.
So, Dr. Mukherjee, will be having some break for 15 minutes.
15 minutes tea break will be there, then we'll assemble.
Virtual tea break.
Yeah.
Okay.
But sir, we want that everybody should be there. I don't know why so very less attendance. I don't know.
Participants lesser.
Yeah. Even those who have registered, they are also not there.
Uh-huh. No, yeah, so in that case, you can make a call if you know somebody who they may face some problem in joining.
I don't.
What is the reason? Those who have registered but not yet joined, this is surprising.
But
Can you find out what is the reason?
in advance and telling everything, and also ask them if you feel any problem joining, please call back or mail us. But not yet, we have not given any mail or any phone call.
Repeated reminder we have sent.
And not only us, both Mukherjee and both number is also there, mail ID is there. We tell any problem of joining, please call us.
Yeah.
So that is the problem with virtual mode.
Mostly in your institute.
They join as per their wish.
Sir, hello. Sir.
Sir, the entire presentation is being recorded from your end.
Yes, yes, sir. It is available in YouTube, sir.
Okay, thank you very much. Thank you.
YouTube.
So, Dr. Mukherjee,
Yes, sir.
I have sent one message. You please see and respond. You try to, if you can do that one, it will be better. Otherwise,
So,
our
sir,
tradition.
I have replied that also. Please see that.
No, it will be better because otherwise the tradition will be this. Yeah. Translate is not done. At least.
No.
Yes.
Yeah.
ब्रेक कर देते हैं सर
Video.
Hello.
Next, we wanted the
Join link में देने से कुछ नहीं होगा.
नहीं हो रहा बोलते.
ये भी मेरा भी नहीं आ रहा मोबाइल में.
It's not coming.
अच्छा, मेरा भी नहीं आ रहा.
So it is not.
Okay. I am sending one more link. Okay.
অনেকে বলতে পারছে না.
मिल जाएगी. यहाँ पे कहीं मोबाइल.
ঠিক আছে, পুরো সবাই যারা ছিল.
Dr. Mukherjee,
Yes, sir.
Keep your video off.
ये प्रॉब्लम है ये करोगे.
हाँ, ठीक.
मैंने म्यूट करूँ. रोहित को फोन कर दो ना.
Is on, no? Okay. This one, see how what should be the best person. Like this. Like we're going to waiting room. Who are waiting? Admit all. So welcome back. Welcome back. I think it is now that that we will listen to our plenary speaker, that Professor Praveen Kumar Verma. I don't want to introduce him in details in this session because you know that he has already given his brief work, and also particularly in the Phytopathology Society, most of them they know very well who is he. But even then, it is my duty that to give a formal introduction to Professor Verma. Professor Verma did his MSc and PhD from Jawaharlal Nehru University. He is one of the most famous universities in the, not in only in India, it is known worldwide. And he joined as a scientist in NASA Institute of Plant Genetic Research, and his work area is plant pathogen or fungal pathogen and host interaction. And I have seen his list of publications. He has excellent publications. The publication range starts from scientific research, scientific report, then BBRC, and even I have seen he is having a paper in plant physiology. It's a very prestigious journal, plant physiology. Because I know that when I was in Israel, my guide who was there in Israel, he was editor of plant physiology. It's very tough to publish there. Though if you see the impact factor, it is very good, it is more than 7. But if you compare with the online journals, it may not be that much. But 7 impact factor journal in plant physiology to publish is very, very tough. Okay, so he is having paper there, and he is having another many other publications in many very famous international journals, and it is continuing also. And as far as I know, most probably 2 of his papers are accepted on, and they are going to be published in high impact journal. He has guided many MSc and PhD students, and recently he has joined the famous Jawaharlal Nehru University, Central University, as a professor. And now, sir, Verma sir, the stage is yours. We are eagerly waiting to listen to you, sir.
Thank you, Dr. Mukherjee. It's indeed my pleasure to talk to all of you in this particular platform. So now I'm going to share my screen right now. Is my screen is visible?
Yes, sir. Yes.
Okay. So I have for the benefit of the young researchers, I have divided my talk since this is a plenary talk. So I have given divided my talk in 2 parts. First part is the general, the how this genomics can help in the plant fungus interaction understanding, as well as developing the strategies to control the fungal diseases. So that we will be discussing that, many people are doing the genome sequencing, then what beyond that? So everybody is having this thing in their mind that after the genomics, what you can do in this particular field. So that that I have given. And the second part that I will be discussing little bit of some of my unpublished data, which we have recently got in our field. That will be giving you some glimpse of the current genomics research, which is going in the plant fungus interaction or plant pathogen interaction as per se. So I will be discussing, my major research area is working on, in my laboratory there we are working on two different aspects. Both are interrelated. One is the plant defense mechanism and against invading fungal pathogens, and second is that the pathogen side we are working on pathogen strategies to counter disease progression in a hostile host environment. And third part, we are doing some translation research where we are doing the genomics assisted breeding for disease resistance with the help of Indian Agriculture Research Institute. And also I do have one dream, and we are trying to work with one startup company, the RNA based biopesticides in this particular field. So altogether, many people ask me whether you are a plant scientist or you are a pathogenologist. Actually, nowadays, whenever you talk about plant and pathogen interaction, you cannot ignore, one side of the coin. So the both things that you have to understand, whether it's a plant or pathogen. So understanding plant pathogen interaction is that you have to understand the plant immune system and how they are countering the intruding pathogens. And second thing is that the pathogen use weapons to suppress such responses. I have deliberately given a term, this weapons. So this entire in my lecture you will understand that how these weapons are important for a particular pathogen to suppress the pathogen, the host immune response. So there is a war between the intruders and the host. So the winner is going to decide whether will be a disease or no disease. So this is one most important thing that we all have to understand. And in the fungal virulence or even the bacterial pathogen or even in viral pathogens, many such type of things are coming, like in the virulence strategy. When we used to study during our MSc, that time we used to talk about that, what are the different components of virulence. We know about the virulence, but how it is being generated, and how the new type of race and new type of disease is coming. So that we will be understanding at molecular level in this particular lecture. So there you all know that the frequent breakdown of resistance or emergence of new virulent strains. That is, every times it's coming. So the breeder who are actually breeding for certain resistance crop, but it is actually the new emergence of new races has come. You all know about the UG99, which has been emerged from the somewhere in the African country. So this is all such type of things are the major threat of for the crop plants. So we all have to understand that how this frequent viral breakdown and resistance or the emergence of new virulent strains are coming up. Also, we have to understand the host jump or host shift, what we call. So this is very, very, very important in that nowadays we all are facing in this case. And also host specificity. You must have seen that many pathogens are, they have multiple hosts. I think Sclerotinia has near about 400 hosts. But some of the pathogens, they are only have one or two host plants. So what are the actual mechanism in those things? That we can, if we can understand that why a pathogen is becoming, is infecting many plants, but some pathogen is infecting only a few set of the crop plants. So we all know about this typical lifestyle of fungal pathogens. This is a necrotrophic pathogen where the pathogen after the infection, it actually kills the host cells. So mind you that whenever there is a necrotrophic pathogen, we all think that it is just going to kill the host cells. But you must have seen that early phases is also its sort of biotrophic phase. So the necrotrophic pathogens also do have a early biotrophic phase, as well as very short that we that and then there will be the cell death type of phenomena, where the biotrophic pathogens, they do actually grow with the living cell. And the symbionts, you all know that symbionts are basically they are getting symbiosis with the plant, and this symbiotic association is there where both are getting mutually benefited. And the hemobiotropic pathogens are some of the pathogens which are having a clear-cut biotrophic phase, where in necrotroph there are no clear-cut biotrophic phase, and but the necrotrophic phase comes after 24 hours or something, sometimes 48 hours of the infection. So I think the next lecture is on the necrotrophic pathogens, so you can understand in a better manner in all these things. So what is I will be discussing mainly about the pathogen weapons. So in the end of the lecture, probably I expect many questions. I really welcome those questions that also help me in the understanding of this particular subject. Or in the myotroph, we all talk about the pretending harmony, the fewer cell wall degrading enzymes than a non-myotroph, and then they evade detection and avoid elicitation of the defense responses. Like necrotroph has smash and grab technique, and they produce toxins and cell wall degrading enzymes. But as I have told you that the producing toxin is the latter part of the necrotrophic lifestyle. Initially, they all try to establish themselves in the host system, so that they can grow properly, or they can suppress the host generated immune system. That we will be discussing later part of my lecture. So many people think about that the toxins are only important thing for necrotroph, but it is not. Basically, there are few reports are there when they have mutated the toxin genes, and after that also you are getting the fungal virulence, not very much compromised. So that means the initial pathogenesis is important for the necrotroph as well. So the pathogen effectors is one from my topic. You must have realized that there is a term we call as a pathogen effectors. Nowadays, we all call this thing after the genomics research, we call as a effectoromics. So this is a fascinating word that we all use, but actually, first we have to understand what are these effectors. So the effectors are basically the secreted effectors are the pathogen weapons. So what are these weapons that we will understand that how they are important in the particular pathosystem. So the reason behind the enhanced virulence and host jump will be also answered by these pathogen effectors. So these are very, very important point that this all have to understand. So there are two different types of pathogen effectors. So suppose this is a fungal effector or bacterial effector. Both do produce effector molecules. Like bacterial effector molecules, they actually inject in the host cells by using the type 3 secretion system. And the fungal pathogen do have a different type of system when the biotrophic pathogen do have a particular very prominent the aprisoria, and the hostoria region is also there, and then the invasive hypha and many other places the different effectors are getting released. So by up to this, you must have realized that pathogen effectors are produced in pathogen, but they will come out from the pathogen. And then there are two ways that they can act. One is that they will be, in the intercellular space or hypoplastic space, they will be confined to themselves, or some of them may enter into the host cell and may be sometimes to the different cellular organelle of the host cells. So these are the new concept which are coming up nowadays. So apoplastic effectors are they secreted and function in plant extracellular space. Cytoplasmic effectors are they are secreted and translocated across the plant membrane and into the host cytoplasm, where they can be targeted to different subcellular compartments, maybe nucleus, maybe mitochondria, maybe chloroplast, or maybe remain in the cytoplasm. So altogether they called as a cytoplasmic effectors. So now up to this, you must have realized that the pathogen effectors are the pathogen molecules which actually comes from the host, the fungus, and entered into the host, or they remain in the apoplastic space. So if you see the different pathosystems, like these are bacterial
Avoid
pathogens.
those pathogens to get entry, to prevent those pathogens to get entry. Now see, there are various techniques. Now, first one is the proper selection of geographical area. That means proper selection of geographical area.
Mr. Rakesh, please unmute yourself.
Many of the
Mr. Rakesh, please unmute yourself.
In many places where the environmental conditions are favorable,
Yeah, Manas,
particular
Manas, you unmute him.
For an example, for
Manas.
Bajra. Okay,
Manas,
mostly prevalent in wet areas. Okay, in wet.
Mr. Rakesh, yeah, okay, thank you. Thank you. So maybe he's teaching, the similar thing. So, the most important thing here is that if you see this particular slide, there the bacterial pathogen, and these are the oomycetes, and this is biotrophic, and this is nephrotropic pathogen. All of them are these. The red dots are basically effectors. So red dots are entering into the apoplastic space, and the yellow dots are entering into the cytoplasm. So that means, the effector proteins, some of the effector proteins, they remain in the apoplastic space. Some of the effector proteins, they enters into the host cell. So this is a very, very important thing that we can understand. So what are these, and how they manipulate this host gene transcription? So many pathogens, they actually target, some of the key components of the host. Whether I'm talking about cytoplasmic effectors, apoplastic effectors, they do have cell wall degrading enzymes. Sometimes they do have certain the components which actually helps in the suppression of host system by flooding themselves into the host receptors for the fungal pathogens. The chitin receptors is one of the example in that regard. But I'll be mainly concentrating today about the cytoplasmic effectors, where they enter into the host cell. So they enter into host cells. So there are two different types of effectors. Now we call, one is the proteinous effector. That means is getting synthesized as a protein in the fungal system, and then it enters into the host system. And another thing is that they are coming as a microRNA or system. So these are the some of the microRNA is also coming to there's a new area of the research, and last 4 or 5 years many such type of things are coming. So the trans kingdom, the transfer of the microRNA from host system to the fungal system and vice versa has already come in the picture. So they may either going for the either RNA silencing machinery, they may be interacting to the transcription factors, or they may be interacting to the some of the genes which is important for the resistance. If they will suppress, then there will be disease, and if they will not be suppressed, then there will be resistance. And mind you that these effector proteins, they not only confined to one particular cell, they can move through plasmodesmata. They can go to adjacent cells, nearby, and they can work in this particular context. So there are small RNA. I have already talked about the dependent, the plant immunity, and in this things, you must have realized that that they may be either interacting with a signaling component, or which leads to the defense mechanism of the host, or they may be interacting with the certain transcription factors. So those are the some of the important factors that we all have to understand. Similarly, the small RNA dependent regulatory regulation is that suppose this is a fungal cell. So this will be the small RNA will be generated from the fungal cell. It will enter to the host cell, and then it will be making the double-stranded RNA and suppressing the host, important host, the messenger RNA by using the PTGS or post-transcriptional gene silencing methods. So similarly, that I have talked about effector proteins. So these are some of the examples which we discuss about. So how this the genome sequencing helps. So this is genome sequencing helps with the effector discovery. So genome, a typical fungal genome, I mean the filamentous fungal pathogens, is having near about 13,000 genes to 20,000 genes, not more than that. So many some times people annotate the genes after the genome sequencing near about 13 to 14 or 15,000 genes. So after these genes, since this has to enter into the host cell, so there should be and they have to secrete out from the fungus first. So they should have a secretory signal peptide. Signal peptides are basically the small peptide which will get cleaved during the processing, and from the exocytic vesicles they will be secreted out after the cleavage of those things. So most of when you look at the genomic sequence, so in any effector protein or any secretory protein which is having a secretory signal, that we can consider as one of the probable candidate of the effector. And then you go for the target discovery. Target discovery, that means where it is, when it is entering to the host cell, where it is actually targeting. So the second thing is target discovery, and then you discuss about the target function. What are the function? What is the mechanism of this target function? This is basically both host and pathogen. So this we have to see everything together, and if we will understand all these things, that will help the crop improvement scenario. So there are this recent review article has been published in molecular plant pathology, where people talking about this proteinous effectors for the discovery and characterization of the filamentous phytopathogen. So how you can discover? I have told you one thing is that they can search for the motifs. There are a few motifs are present in the scenario, and I will be giving you some of the ideas of the motifs when I will discussing about my own work presentation. And then the bioinformatics pipelines are also very, very important. The homology searches, although it doesn't help much in fungal pathogen, as I have told that all fungal pathogens, they have evolved in a different environment, different niche. So the homology you can get, but not that much prominent in many of the effector molecules. Or sometimes the transcriptome wide association studies or genome wide association studies also help in the isolation of these things. So the new era which has started from the isolation of the first AVR gene or AVR9 in 1988 to the current era, where people have talked about with all these motifs and the effector molecules. So the earlier era was the proteomics-based approach, then map-based cloning, then genome landscapes comes, then comparative genomics comes, and then lineage specificity, which is coming. So altogether, is called as a effectoromics. So now we call, and this is one of the fascinating area is what if you are working in plant pathology. So don't try to think about that. These are all the rocket science. We all can do. I think even the classical pathologist can do with the little bit help from others. So these all things that like many the genome and transcriptome data is already present in the database. So there are you have to select the effector candidate, which I have already told about that you can select by secreted proteins, and then upregulated during the infection you can see, and then candidate validation that you can do the implanter functional expression that you can look for, or the gene knockouts. Knockouts is basically that you are suppose there is you want to know the role of a particular gene, you just remove that gene from the genome, and from there you can know the function of it. Then the fungus should become from virulence to avirulent. So that way you can validate these all these things, and this you can do by RNAi, and all these key effectors or the common host target that you have to look for, and then you can look for either doing we call as a transgenic or genome engineering or genome editing platform that you can go for host-induced gene silencing. And host, I will be discussing a little bit in the latter part of my talk. And this all leads to the crop improvement. These are some of the things that we have given a brief idea. So everything about effector has started in 2010 when 4 papers has been published in the same journal, Science, which is a very prestigious journal, and in the same issue, and where you can find many. If you read the titles, you can see the what people are talking about. Like here, the host jump is there. Then there is the signature of adaptation, how this pathogen has got adapted. Then the pathogen determinants of the smut fungi and the genome expansion and all these things. The most important thing is that when people started, the sequencing of various pathotypes of various genomes, they have found that if you see that the rapidly evolving genes are the genes which are coding for the effector proteins. So that means the pathogen always try to change themselves. It's just like that if a terrorist is coming to our country, and he will try to hide themselves in such a way that it should not be recognized by our security forces. So similarly, the pathogen also try to develop certain mechanisms by which they can get unrecognized by the host machinery. So this is one of the most important factor that we will talk about. So my second part of my talk is about my own work, which will correlate of all these things that what I have discussed today. I will be since we have already communicated this paper in a very big journal, but I will be not discussing entire data. Only the few data which will help you in the understanding this particular field of research, as well as the utility of this type of research. So my lab is working on 3 different pathogens. One is the Ascocata blight of chickpea, and another is the spot blot disease of wheat, and blister blight of the tea. So this is one, it's a necrotroph, this is a hemibiotroph, and this is obligate biotroph pathogen. So I do work on all 3 types of lifestyles, and mostly I have most of the data I'm presenting today is Ascocata blight of the chickpea. The both genome has been sequenced of the Indian strain that we have sequenced in our laboratory, and we have done this genome sequencing by Illumina platform. I think long back in 2016 we have published this paper, and after this this we called, nothing to, get worried about all this data that we write 100 GB or, the 178x, doesn't matter, to anybody, but you have to understand what we have got out of these things. So actually, after the isolation of the gene, we have, isolated near about 13,864 genes, and we have predicted by using computational programs. And out of this computational program, we have got near about 858 secretory proteins. Out of these secretory proteins, we have got many, genes which has been some of the motifs which is already known in the different, fungal effectors of or oomycetes effectors. So that we have, looked forward. So one of the effector which is important that I am going to discuss today is the pexel motif effector that we have got near about 122. And we try to see the expression of all these genes, and we found that, this PEC25 that we named after, that is a pexel-like effector candidate 25. 25 is the number of clone. So these all are, isolated in my laboratory. So then, it is having one motif. So that's why, we have done with the different strategy. So most important thing is that just after the infection, the fold change, I mean the expression of the gene goes up to near about more than 500 times. So that means this that's why we have selected this particular gene for our future research. So this pexel motif that I have just named is basically a plasmodium export element. You all will be surprised. I am not putting some of the data of the plasmodium today due to lack of time, but I do have even the data of the plasmodium, where we, the similar type of, mechanism is going on in the plant and pathogen. So if you think about this sequence containing this motifs, so this basically we named as a PEC25, which is a novel Escocita rebae effector, and this is we called as a pexel-like effector candidate. So this candidate is having, as I have already told, that all the effectors should have a signal peptides. So it is having also a signal peptide, and it is having it's a very small one, near about 134 amino acid long, and it is having, this one pexel motif and one arginine rich motif. So this some of the function we have identified, where, there's this pexel motif which has been first identified in plasmodium, where people have found that first the arginine and the lysine and other D, art L and D motifs, which has been given in red, they are important for the function of this particular gene. So this is important now that we have to understand that whether this protein is a secretory protein or not. So for this, we have to, what we have done is that we have not told about all these things, that you must have heard about many fungal many transformation of plants by using agrobacterium mediated transformation. But here what we have done is that, that agrobacterium mediated transformation of the fungal pathogens that we have established. And this we have established, many other pathogens. I think 8 we have established right now, and where we transform the fungal pathogen by using certain tags we call as a fluorescent tags, and then we go for the Western analysis in the culture filtrate. And here we can see that, the culture filtrate is there. So that means, the culture filtrate is there. So that means it is it's important for the all this in that this protein is actually secretory protein. So the most important thing that we have to, what we have seen is that it is having high implantar expression study, regulation, upregulation during oxidative stress, and also we have done the knockout. Knockout we do by using the agrobacterium mediated transformation and the knocking out by of a particular the gene in the genome. So that we use the method we called as a homologous recombination method, and by which you can see that this is the wild type symptoms. These are some of the symptoms of the knockout, where the symptom has gone, the number of lesions per plant goes down, and this is in the case of the complementation. So complementation is basically after the knockout you again transform the fungus with the same gene, and it should restore the fungal virulence. So that means this particular protein is very, very important for the Escoqueta virulence. So now the case is that we have proven by this experiment that it comes out from the fungus. So if it comes out of the fungus and if it interacts to the host cell, then where it is getting localized. So what we have done with the using the GFP as a tag, we have found that this particular protein goes to the nucleus of the host plant. So that means that it is coming from the fungus and entering into the host nucleus. So this is very, very important, and what we have got. So this this particular gene is having the what I have discussed about, which we have done many experiments in the Plasmodium, which I am not showing the data, where even in the Plasmodium case we have shown that this particular motifs is important for the Plasmodium. So now up to this, this work is done mainly by the Srinivas and Sandhya. And this now, up to now, you all know that it's localized into chickpea nucleus, and it must be interacting and manipulating the transcription factors. If it is nucleus has transcription factors or a transcription regulator, or it may act directly to the promoter sequence of a particular gene. So that helps in the idea that what is the target of the PEC25 in the host. And mind you that still people have isolated some of the effector proteins of the necrotrophic fungal pathogens, but this is going to be the first report from any fungal pathogen where it is actually we have identified a target. Not only target, we have underlined the mechanism by which it is actually suppressing the host defense system. So in the next few slides, you mute Divya Tiwari, please. Yeah. So this first time we have done in our country. And not in our country, internationally also we will be the first person to do that thing. So how to isolate these host targets? So for the isolation host target, again we have to transform the fungal hyphae. Then we will use that fungal hyphae where the transformed our system is there, the PEC25 is there with a tag. Tag we call as a basically say a known tag which is having a either antibody or anything which is already known in for all these things. So then we infected the plant system and then isolated the plant the infected leaf, and then we have gone for the isolation of protein, identification of the protein by tandem affinity purification, and finally confirming whether this particular target is is correct or not by using Y2H, that is called the E2 hybrid system, or by molecular fluorescence complementation. So ultimately, these are the pathway that we follow. So in this case, I'm not going to the technical detail of all these things, but just I want to give a glimpse that how people do about all these things. Like after the isolation of the target sequence by tandem affinity purification, we go for LC-MSMS, that is liquid chromatography, MSMS mass spectrophotometric analysis, and by which we have found that one of the interacting protein is a CA beta limb 1A protein, which is a transcription factor. So now we can understand that the transcription factor, this PEC25 is coming out from the fungus, is entering into the host cell, and it is interacting with a transcription factor that we have isolated. And this we have proven by pull down and interaction, as well as many other techniques that you will see in my next slides. So the structure that of this transcription factor is basically is having two limb domain. Basically is identified long back in the case of C. elegans, not in the plant system. But plants also, they do have a dual role. They do have a role in they bind to the PAL box. PAL is basically the first enzyme of the phenylpropanate biosynthetic pathway. That is the phenyl ammonia lyase gene. So it bounds to the PAL boxes of the promoter of the host cell. So this we have proven by using the E2 hybrid analysis, as well as in-plant apolocalization assay, where PEC25 and beta limb 1A is both are interacting in the nucleus of the host plant. So this is very, very important now. And then we have gone for the gel retardation assay by using the in vitro binding of with the PAL boxes, which is present in the promoter sequence of the PAL gene, CA PAL gene. So, and then we have also done by using the East-1 hybrid analysis that this CA beta limb 1A is actually interacting and with the PAL box of the host plant. So this is a way. Then now question is that this is actually in the normal condition. It is actually this type of function is happening where the CA beta limb is the PAL box is so then what is the implication of the binding of this this PEC25 to CA beta limb 1A? So that means that means it may be either hampering the binding of this particular transcription factor to the promoter, because this is the most important pathway, phenylpropanate biosynthetic pathway, which leads to the synthesis of lignin. So it leads to the synthesis of flavonoids, phenolics, isoflavonoids, many other, even salicylic acid also gets synthesized by one offshoot of this pathway. So that means that the first enzyme, and which is a rate limiting enzyme. So if it is interacting with this particular transcription factor, so it must be down regulating the transcription. So this we have proven by first by using the gel retardation, where here we have used the different concentration of the PEC25, and here you can see that this binding is actually getting depleted as we increase the concentration of PEC25. Similarly, for in vivo data, we have used a luciferase-based system, a reporter system, and then we have identified that the PEC25, the presence of PEC25, the suppression of this gene is in the expression system is there. So the similar things that we have gone beyond, because since this pathway is having the is a phenylpropanate biosynthetic pathway, leads to the synthesis of lignin, different type of lignin, S-lignin, G-lignin, and S-lignin. So we have looked for the different components of these monolignin biosynthetic pathways, that whether it is getting infected after the infection or not. So we have done with the treatment and other things, that you can see that cinnamic acid, caffeic acid, and even this syringic acid, they are, and chlorogenic acid, all these are getting, these are the mock, I mean the just for the and the treatment, it has these phenolics, or monolignar pathway phenolics, they actually goes down for the synthesis of the lignin. So ultimately how this is going is basically that it is coming from the host cell and then entering from the fungal cell, entering into the host cell, and then is interacting with the PAL promoter by inhibiting one of the regulator of this PAL promoter, that is the CA beta limb 1A, and then it actually regulating the lignin biosynthesis. So you all know that lignin biosynthesis is one of the most important the after effect of any fungal pathogenesis, where the plant do strengthen themselves for the lignin deposition. So this type of things is happening. So in the finally, this is my working model. So you just try to understand that this is the normal case where these are the limb transcription factor, which is having 2 limb domain, and it is actually binding to the 2 boxes present into the CA-PAL1 gene. It is having the limb1 and limb2 is binding to the B1 and B2 PAL boxes, and it actually helping in the synthesis of the secondary metabolites and mainly the lignin biosynthesis. So there will be less infection or no infection in this case. But in the case of the virulent, the strain, this ARPEC25, which is getting infected, and these are PEC25 effector protein which are getting synthesized. As I have told that the expression is very much high in the case of infection, and then you just see these dots as an animation, and then it actually enters into the host cell. These are the host cell, and then it enters in the host nucleus. So it is entering into the host nucleus, and then it actually interact with the limb domain transcription factor and inhibiting the expression of the gene. This will result in the disease. So this is a one type of the understanding the virulence mechanism in the plants. So this is the one I think I'll be able to I would have been able to discuss about all my work along with the background knowledge to the sum of part of my work, so that you can understand that how this thing is giving answer to some of the unanswered question in plant and microbe interaction or plant pathogen interaction. This is my complete group. Since I was working in mainly in the last 1 month only, I have shifted to JNU. So my institute was NIPGI. So mostly this work has been done in NIPGI, and these are some of my workforce which work in my, and this is a site of NIPGI. And thank you very much to all of you. The questions are open. Thank you very much.
Thank you, sir. Thank you very much for your really enlightening and enlightening lecture. We have learned many things about ascocita and chickpea interaction. And actually, in plenary speaker said whether he'll take the question or not, that depends upon the speaker himself.
I am ready.
So if any question,
no problem.
has kindly agreed. Sir is agreed to take the question and answer the questions if any question is there. Then
see, one thing is that I have given an example of how this type of mechanism is happening. This type of mechanism happening. A person can ask this question that how many such type of effector molecules are there in the genome. So sometimes it is 300 to 600. Such type of things are there. Sometimes people have annotated 150, 180 like that. So thing is that sometimes the pathogen, the host system, they try to develop some sort of resistance against these. These are effectors. Basically, earlier we used to call as a virulent genes of the pathogens. So these are virulent genes. They used to recognize and develop some certain type of resistant mechanism. So then the new type of effector molecules, they come in the picture. They change themselves and then they try to infect the plant. So that's why the frequent breakdown of the resistant mechanism is there. Frequent even the new virulent strains are now emerging because new effector molecules are getting in picture. So this type of mechanism is not confined to one pathosystem. This is in all pathosystem. Right now we are working on the similar strategy and we have isolated 2 effector molecules from the bipolar pseudoquinina which infect wheat. So the similar type of mechanism is happening everywhere. So any question?
So, if there is no question, then sir, thank you very much. Thank you for your lecture. We will be having interaction in future also, and as you suggested, we will try to conduct a brainstorming session if possible through the Indian Phytopathology Society in future. And then sir, we'll invite you there to be as either a main resource person. And thank you very much, sir. We are looking forward for future collaboration with you. Thank you very much for accepting our invitation.
Thank you very much.
Thank you. Now we will start our first technical session, and Dr. Pavu Karthikeyan will conduct the session. Dr. Pavu Karthikeyan, please. In the these are the persons who are requested to be ready. And the first lecture will be by.
So, good morning, sir. A warm welcome to all of you for the first our technical session. We are starting our first technical session post first interaction. Chairman sir is connected.
Yeah.
Dr. Srikant Das sir.
Yes.
Uh,
Am I audible?
yes,
Yes,
sir. Yes,
yes.
sir.
Okay, thank you very much. Good morning, everybody. I am first.
Sir, just a
Minutes.
Okay.
minute. We
Sir,
introduce yourselves and Madam Surekha Kundu. Then we'll hand over the mic to you, sir.
Okay.
Yes, sir. Yes. So first of all, I would like to welcome our chairperson of this session, Dr. Srikant Das, Professor Plant Pathology from BCKV and ex-Dean Faculty of BCKV. His specialization is in post-pathogen interaction. He has guided many MSc and PhD students, and he has published in peer-reviewed national and international journals. I welcome you, sir. Also, I would like to welcome our co-chairperson, Dr. Surekha Kundu, Associate Professor, Calcutta University. Sir, we have 2 invited lectures followed by 6 oral presentations. The time limit for our invited lectures are 20 minutes, and the time limit for the oral presentation is 8 minutes. And additionally, we have 2 minutes for discussion. So the 2 best oral presentations will be selected by our chairperson and co-chairperson, and the result may be sent into our zonal president, sir, to email. Sir, let me start, sir. Our first.
Okay, thank you. Thank you all. Particularly, I thank you to organizing committee and secretary, Dr. Aru Mukherjee, Eastern Zone,
Thank you, sir.
and also our Eastern Zone Councillor, Dr. Manas Kumar Bagh,
Thank you, sir.
and also thankful to our secretary, IPS, Robin Gogoi, and our president, Honorable Dr. Pranjit Chakraborty, and also I am thankful to Dr. Dipankar Maiti, Director, Honorable Director, NRI Kotak. And I have had a very important lecture, particularly host pathogen interaction in biochemical reactions by Professor Praveen Verma, very thoughtful lectures, and also very interactive and important lectures. Now I start particularly the technical session host pathogen interaction. 2 invited lectures are there. By 1 invited lecture by Dr. Surekha Kundu, my co-chairperson. Are you present? Dr. Surekha Kundu is present?
Yes, I'm present.
Okay. Next, Dr. Subroto Dotto, associate professor from my university. Whether Dr. Subroto Dotto is present? Hello?
Hello.
Dr. Dotto is present?
Yes.
And another 6 presentations, oral presentations are there. Girish Chand, Poulomi Shil, Ranjit Kundu. I think this will present by Dr. Mr. Another person will present. Ashish Kumar Anand, Boapati Rohit Tej Naidu, and Somosetti Ravin. I first call Dr. Surekha Kundu, my chairperson, kindly present 1 invited lecture you are giving, plant necrobe interaction in the early stage of infection, a game changer for the outcome.
Sir, Sir, let me introduce Madam Surekha Kundu.
Okay, okay.
Sir, Dr. Sureka Kundu did her PhD from Bose Institute, Calcutta, and did her postdoctoral research for 6 years in the University of California, Los Angeles, USA. She has published more than 45 papers in reputed journals and 10 patents in her name. And her main area of interest is to study the post-pathogen interaction, especially in the early phases of infection, cloning of defense genes, regulation of defense genes, and transgenic research to develop multi-stress resistant crop lines. Madam, we are eagerly waiting your invited lectures, please.
Okay. Thank you very much. And I'm really grateful to the organizers for giving me this opportunity to speak. A very good morning to my esteemed colleagues and to students. I will now share my screen. I'll stop this video. So is this visible?
Yes,
Yeah,
yes,
yes,
yes.
Okay, so the topic of my talk today is plant necrotroph interaction in the early stages of infection, which is a game changer for the outcome of the disease. Now today I'll be not going into any technical details of the experiment, but rather I'll be showing some pictures, photographs, where you will be able to see exactly what is going on between the fungus and the host, because there is a real struggle going on for existence. And every time I observe it, I feel this sense of respect, reverence, and awe. And I wish to share this feeling with you. So I've divided my talk into different sections. The interaction before any contact is made between the fungal hyphae and the host. So even before any contact has been made, there is a lot of interaction going on. And then interaction on the surface of the host. Then behavior of the hyphae within the host tissue when it's infecting the host. And what is the host doing? So the host defense response, as well as a little bit about the suppression of hypersensitive response, which is often induced by a necrotrophic fungus through RNAi approach. And as Dr. Verma was saying, it is very difficult to choose a side when we are working with host pathogen interaction. One time you're supporting the fungus, another time you feel like you're on the host side, because both of them are equally good fighters and competitors. So I have chosen 3 pathosystems. We're working with all of them. So I'll be sharing some published data as well as some unpublished data, even recent data that I have from my lab in the end also. I've chosen Rhizoctonia solani and rice, where we have 2 varieties. One is tolerant, Shornodhan. One is susceptible, which is Shorno. Macrophomina fasciolina and sesame, resistant susceptible variety. Alternaria solani and tomato, again, resistant and susceptible variety. Here, I must stress that no plant is completely resistant to a fungus. So when I say tolerant, I just mean it as a comparative term, comparative to the other variety that I'm choosing. You can call it more susceptible and less susceptible if that pleases you. So tolerant means less susceptible, susceptible means more susceptible. So before I go into this, the first thing that we did is we transformed each of the pathogen with green fluorescent protein gene. So we were the first laboratory to transform some of these fungi. We have used a construct and we've used different methods to transform these fungi. We've even gone into nanotechnology mediated transformation. I won't go into the detail. This is Rhizoctonia solani. This is Alternaria solani with its typical conidia with vertical and horizontal septa expressing GFP. This is Macrophomina fasciolina expressing GFP. So I'm coming to the interaction before any contact is made. So what is going on the phyllosphere and rhizosphere? These are only a few pictures which I can show you within the limited time. This is susceptible rice, showing that if you place a fungal inoculum away from the root, within the same time period, it has already reached the susceptible root surface. Whereas in the tolerant variety, no hyphae has reached yet. So clearly the fungus shows a preference to move fast and forward towards the susceptible variety, while it delays and is reluctant to approach a tolerant variety. And in fact, given a choice, it would go towards the susceptible variety and not towards the tolerant variety. This in case of Macrophomina and sesame also. So these data have been already published. You can see a lot of hyphae near the rhizosphere of the susceptible root, whereas near the tolerant root, the hyphae is tending to grow parallel to it and is reluctant to interact with the root. And when you draw the graph, it shows the same thing. So what is the interaction taking place on the surface of the host? So once it reaches the surface of the host, this is a very interesting picture which we got repeatedly, scanning electron micrograph of interaction of Rhizoctonia solani hyphae on rice leaf. So in the tolerant variety, the hyphae tend to grow in bunches. Okay, and they don't want to come in direct contact with the host surface. So bunching of the hyphae probably gives them less contact area with the host surface. Whereas in the susceptible variety, the hyphae is freely growing singly and is in intimate contact, is tightly attached to the host surface. So on the sheath also the same thing, there is bunching of hyphae and reluctance to come in contact with the host surface. Whereas in the susceptible variety, it is freely coming in contact and growing singly. So the reason for this might be two. One is very simply the tolerant host is secreting something which is antagonistic to the fungus, antifungal in nature or whatever, and the fungus clearly does not want to interact so much, while the susceptible one is not able to secrete anything. Or it might be that the tolerant variety is avoiding detection by the fungus. The fungus is not able to detect so much the presence of the host, whereas in the susceptible one, it is able to detect those chemicals or whatever it is exuding. Or it might be a combination of both of the factors. Okay, this is the interaction of fungi on the root surface. So this is rice root, which is completely surrounded by the fungus in the tolerant variety, but the growth is much less and no microsclerotia or sclerotial development. At the same time point in the susceptible one, there is encirclement of the engulfment of the root with the hyphae, but also formation of many microsclerotia. This is sesame. This is the tolerant root, and this is a susceptible root, the same thing. When you look at the confocal microscopy with the GFP transformed pathogen in the sesame root, you can clearly see a big mantle glowing, which is the susceptible root with lots of microsclerotia, whereas in the tolerant variety, there is much less growth, much less interaction with the fungus. Okay, now this is Alternaria solani hyphae interacting with tomato leaf stomata. These are two pictures of the same dark field and the merged field, where in the merged field you can see the conidia germinating, Alternaria solani conidia. Here the stoma has been directly intercepted by a hyphae. And this is a simple microscopy showing a germinating conidia sending a hyphae directly intercepting the stoma. These are two electron microscopy showing the stomata are being intercepted by the hyphae. So this clear interception of this stoma by the hyphae kind of makes you wonder if the fungus can really sense the presence or the existence of a stoma nearby in the locality where it is growing. So this is my most favorite picture, I would say, of perhaps my career. Okay, this is a simple microscopic picture, but this is a rice leaf where the stomata are arranged in a row, along a row. And this is a Rhizoctonia solani hyphae, which is growing parallel to the row of the stomata. And at each point, it is giving out a side branch to the hyphae, to the stomata, which is lying adjacent to the main hyphae. And it is intercepting each of the stoma through the shortest distance. So it is a very surprise for me that the fungal hyphae can actually detect the presence of a stoma and then branch at that particular point, and through the shortest distance, it can intercept that particular stoma. It is very precise and very targeted. Okay, so this is what I wanted to share. We've published this data. This is another scanning electron microscopy of the same thing, a stoma being intercepted by Rhizoctonia solani hyphae. Here, the protrusions that you see are papillae. So this hyphae is taking the help of the papillae. It is tightly wrapping itself around it and approaching the open stoma. Here, the stoma has already been infected and it has formed a plug in the stomatal opening, and it is reaching the underlying tissue. So this plant is already in trouble. So it has already infected the stoma and is reaching the underlying tissue. This first picture was so dynamic, and it shows a lot of action. So this was chosen to be on the cover of the Plant Pathology that year, that issue. Now I'm coming to the interaction of hyphae with trichomes or surface structures. Trichomes are hairs, as you know. And while trichomes have been evolved, has been produced by the host as a defense organ or a defense mechanism to prevent herbivory, to prevent falling of spores coming in contact with the surface. Yet fungi over the years have actually evolved to attack these very trichomes and utilize these very trichomes for its own advantage. So trichomes are utilized for anchorage of the fungal hyphae, and also as penetration points, surprisingly, and also as zones for hyphal colonization. So we see a lot of colonization of fungi around these particular some trichomes, depending on the types of trichomes. So I will not go into details of the different types of trichomes that we've studied, but I've briefly showing you the data. The blue square is for rice, where you can see a bulky prickly trichome being intercepted by hyphae. Here the long hairy trichomes are being circled by the hyphae here at the top and at the base. We've seen repeatedly that the fungus tries to enter through the base of the trichome. This is not just a broken trichome by mistake. Here the GFP shows a colonization at the base of this spike trichome. Here a delicate prickly trichome being encircled by the hyphae, where it is interacting with the trichome. In case of Macrophomina facilina, this has been published. This rice part has not been published yet. The Macrophomina facilina, you can see the different trichomes, two trichomes being intercepted by the hyphae. Here it has been basically strangulated by the hyphae. Here this has been circled by the hyphae. In case of Alternaria solani tomato also, this is a simple microscopy showing infection, actually penetration already of the trichome. This is a better picture where you can see a pore or a hole being intercepted by the trichome, and it is entering the trichome on the surface of the leaf. So these are the different interactions that take place with the trichome and the fungus. So what is the host doing? The host itself is also trying to protect its trichome, and there is actually, we see callose deposition more in the trichomes compared to the surrounding tissue. These are the filamentous long hairy trichomes of rice. These two pictures, these are the bulky prickly trichomes, which shows callose deposition. Compared to the surrounding tissue, there is more callose deposition in the tolerant variety. These are trichomes on tomato leaves, which are clearly glowing, showing more callose deposition in the trichomes. And when we draw a curve, you can see that the susceptible variety has less callose deposition in the trichome, and therefore it is susceptible, whereas the tolerant variety is able to accumulate more callose in their trichomes, therefore they are tolerant. So this is a game-changing event too, which is happening in the first few days of infection. Now I'm coming to the host pathogen interaction during infection and establishment phase. These data have been published. So these are 3 stages of formation of infection cushion of Rhizoctonia solani on rice leaf. First, the hyphae is loosely branching and forming kind of like a cluster, like a nest. Then it becomes more aggregated and it solidifies and it becomes an infection cushion. And with a simple microscope and just cotton blue staining, you can see these clusters, which are called infection cushions, which are forming on the susceptible leaf compared to the resistant leaf. And we were interested to see what is going on underneath these little matrices or cushions of the hyphae. So we made a cross section through the cushion, and this is the top where the solid arrow shows the cushion, the infection cushion, and the dotted arrow shows direct forceful penetration of the host tissue with infective hyphae, and it directly vertically sends down these infection hyphae and it invades the tissue underneath the infection cushion. Okay, this is another good picture in my set of experiments where we were able to capture the cells, intact cells, surface cells of the rice root, which appears like a big pillow or a side pillow. And these are the hyphae, which is growing in between the cells. So this is intercellular growth of the hyphae. So these are puncturing the cell wall, kind of like a sewing thing, which is anchoring itself and growing in between the two cells. It tends to grow intercellularly during the initial phases. So it becomes rectangular. It encircles the cells without entering the cells. But later it infects the cells. So this is tomato root where the hyphae has entered the cell. Here, this part has been published in the sesame root where you can see the entire cell being colonized by the initial biotrophic phase of the necrotrophic phase of the pathogen, where you can see the hyphae filling up the entire space of the cell. Here the SEM microscopy showing presence of hyphae within the cell. In this picture, you can see intercellular as well as intracellular growth of the hyphae, where you see the rectangular green parts, GFP parts, those are encircling the cells, intercellular growth, and directly growing through the cells as well as intracellular growth. So once the fungus reaches from the outer layers of the plant tissue to the inner layer, once it comes in contact with the vascular bundle, it is a good news for them. So they enter the xylem vessel and it becomes like a big highway, a freeway to go forward and backward wherever they want, undeterred. So they do tend to grow and plug the xylem vessels. This is the leaf seen from the top. You can see the Rhizoctonia solani right angle branching. It is moving through the xylem vessels and also sending out side branches to the surrounding tissue. This is a xylem vessel showing, I think, scalariform thickening, where the hyphae is growing through the sesame root xylem by Macrophomina fasciolina. Here, the entire bunch of xylem vessels has been attacked by the pathogen Macrophomina fasciolina. Here, some plugged vascular bundles are being seen in the cross section. Now, very briefly, what is the host doing? The host defense response. This is a very busy picture. I'm not going into the detail. We've already published this. The fact is that Macrophomina fasciolina has been known as a strictly necrotrophic pathogen, but we have discovered that there is a short biotrophic phase, which is occurring in the first 2 or 3 days or the first days, depending on which host it is infecting. So there is a shorter biotrophic phase for susceptible variety, and there is a longer biotrophic phase for the resistant variety. So when we analyze all these genes working in the secondary metabolite pathway, the SA signaling pathway, J signaling pathway, ROS, calcium ion signaling pathway, etc., we see that the host has to adjust the change in the pathogen. So when the pathogen is changing from the biotrophic mode to the necrotrophic mode suddenly, so it is a BNS, biotrophy necrotrophy switch, the host has to immediately change its strategy of defense. From SA pathway, it has to change over to the JA signaling pathway. And the faster it can change its defense strategy, the tolerant it will be, the better will be the outcome for the host. And if it's slow to intercept or anticipate the change in the pathogen, then it will become a susceptible variety. And in each of these graphs, we can see there is a change in the trend of the different in the susceptible and resistant variety, and that happens to coincide exactly with the biotrophy necrotrophy switch that is taking place in the tolerant and in the susceptible variety. So it is that this interaction in the initial phases does decide what is the outcome. So these are some of the publications that I've talked about and also added some new data to it. Okay, now I'm coming to the next part. This is directly from my laboratory right now. So the raw data kind of out of the oven. So whatever we've done is we've already completed this part of the work. We are trying to suppress the hypersensitive response, which is often triggered by necrotrophs. So necrotrophs sometimes trigger hypersensitive response, pretending to be a biotroph. And once the plant goes into hypersensitive response, it can actually utilize that response in the necrotrophic lesion and it can attack the plant more easily. So here we've used a virus-induced gene silencing method. We've tested it with the GFP reporter, the leaf infiltration work. The gene that we've chosen is AVR9CF9 induced FPOX1, ACIF1, which works a little bit downstream in the hypersensitive response pathway. And we decided to suppress it through RNAi technology and see if it gives resistance against necrotrophs. So we have cloned these two genes under the two constructs, TRV2 GFP, TRV2 ACIF1 construct, and these are some of the experiments. But very briefly, if you look at the last two columns, when we use the blank like TRV2 GFP only and the RNAi line where the ACI1 is suppressed. So in this line, there will be no hypersensitive response. And in this line, there will be hypersensitive response. When we infect these areas with Alternaria solani or a necrotrophic pathogen, we see a development of symptom, whereas there is no suppression of hypersensitive response. And if we can suppress the hypersensitive response, there is less infection or less symptom. So this has been already done. So in summary, I can say during the early phases of infection, I believe that is the most interesting part, where a small advantage taken either by the pathogen or the host will amplify into a big difference in the end. So it is a game-changing part of the interaction process, the first few hours or the first few days of the infection. And the pathogen and host both are competing with each other, and they have evolved numerous ways to avoid each other. I mean, one is trying to attack the plant and the other is trying to avoid the pathogen. Similarly, the host have evolved many ways, and they were able to alter its defense response as required, as we've seen, depending on the situation the pathogen is in. So if the host is able to anticipate what the pathogen is going to do, it is a tolerant variety. If it is not able to respond quickly, it becomes a susceptible variety. And we have also seen that hypersensitive response is often induced by a necrotrophic pathogen. And by suppressing this hypersensitive response, we are able to increase tolerance towards that particular necrotrophic pathogen through RNAi. So thank you. And this slide again is showing the Rhizoctonia solani hyphae growing in between these two papilla and is encircling this papilla as if it can literally see or feel the surface of the host leaf. Okay, thank you.
Thank you very much, Dr. Surekha Kundu. Your nice presentation regarding through scanning microscope or electron microscope, we have seen the how hypersensitive reaction or how the pathogen interact before or after infection or within infection. Very thankful to you, because this is very impressive to the particularly students. Seeing is believing. Seeing is believing, how they see and they can believe how interaction takes place within the host plant. Thank you very much, Dr. Surekha Kundu. Okay. Any questions? Hello? Okay. Thank you. You asked.
Sir, one question. Sir, one question from me.
Are you okay?
Sir.
Yes, yes.
Madam.
Yes.
I am familiar with your work by your publication and other way. As outstanding presentation, I should say.
Thank you.
But one thing I want to know, madam, that you showed that Rhizoctonia solani is able to recognize or sense the stomatal aperture in the plant's tissue, leaf tissue. Okay. So, is there any signaling molecule identified from the plant?
No, I haven't done that. I haven't found any research on that. It can infect through the stomata and also through direct brute force, through the epidermis also. So I'm not working in that area, but it would be interesting, yes, to find out what it can sense in the open stoma. It is the thing that I would like to work on in the future.
The stomatal aperture, that's really very
yeah, yes, it can actually detect open or a closed stoma also.
Yeah, it's right.
It is so amazing. It is really amazing.
Madam, thank you very much again for accepting our invitation. And
thank you for giving me the opportunity.
Please,
just one question, just
yes,
question.
Please.
Like the infection cousin that you are talking about, that maybe because this particular pathogen do have a large scale anastomosis, so that may be happening because of that. That is.
Yes, absolutely.
Yes. Okay, thank you.
Thank you.
Next speaker, Dr. Subroto Datto, are you ready?
Yes.
Are you okay? You can start.
So, sir, let us introduce Dr. Subroto Datto, please.
Before, hello. Sir, Dr. Subroto Datta completed his PhD from the most prestigious institute, Indian Agricultural Research Institute, and then joined as assistant professor in Vidhan Chandra Krishi Vidyalaya, BCKV, His research area includes epidemiology for plant pathogen, genetic diversity of plant pathogens. He has guided many MSc and PhD students and published several articles in peer-reviewed journals. Sir, please, you can.
Okay. Dr. Datta,
Yes.
kindly present.
Screen is not visible. Screen present now.
Okay, ask him. Okay.
Is visible?
Okay. Okay.
Respected chairman, co-chairman of this session, distinguished scientists, the office bearer of the Indian Phytopathological Society, research scholar, and dear students. Today I'm going to present the topics of changes in disease and pest scenario under Gangetic alluvial region of West Bengal. Tomato is an important vegetable crop consumed worldwide. India occupies second position in vegetable production. And among the vegetables, tomato ranked third in priority in India after potato and onion, but second in the world after potato. West Bengal is the third highest producer of tomato. Among the several diseases, foliar blight, that is target leaf spot, early and late blight, tomato leaf curl, bacterial and septorial leaf spot are a very serious destroyer and for the disease development. And it can kill the whole crop in a few days under favorable weather conditions. The control of foliar blights of tomatoes relies mainly on the frequent application of fungicides, even at 7 days interval. In today's agricultural motto, there is a need for reduction in the number of fungicide application, while maintaining the disease control could achieved only by fungicides at times when conditions are favorable for disease development. For that reason, the present study focused on influence of various weather parameters on initiation and spread of the disease, and the development of weather-based prediction model for foliage diseases of tomato and insect pests for tomato under the Gangetic alluvial region of West Bengal. For any disease, so far we have studied that host pathogenic interaction. The three factors are important, that is the disease triangle, that is for susceptible host, virulent pathogen, and favorable weather condition. If the pathogens are present, but weather conditions are not favorable, in that situation, there will be no disease. And there are different pathogens which are very much weather sensitive, and each and every stages of their pathogen biology is dependent upon their weather conditions. Now, just give some highlights. During 2011-12 to 2019-20, what are the weather abnormalities present? If you see, in the month of February, the Tmax was highest during 2014-15, 15-16, and 16-17, and 17-18. Whereas the average Tmax of November, December, January, February were comparatively higher, 2 to 3 degrees comparatively higher in case of your 2014-15, 15-16, and 16-17. If you go for the T-mean, we find that among the 10 years, only the 2 years, that is 2012-13 and 17-18, the T-mean was less than 10 degrees centigrade during the month of January, which is very important, especially for late blight disease. In case of month of February, we find the highest T-mean was in the month of 2015-16. In that way, if we go for the rainfall conditions, we find throughout the October from 2011-12 to 2019-20, only the 2 years where the rainfall was more than 200 millimeter, that is 240 millimeter in 2013-14, and 2017-18 it is 237.7. If you consider the rainfall in the month of January, in the last 10 years, we only get more than 20 millimeter of rainfall, that is 57 millimeter of rainfall in 2011-12, and 20 millimeter of rainfall in more than 20 in 2018-19 and 19-20. If we consider the month of April, we find that the more than 100 millimeters of rainfall we get in 2012-13 and 2018-19. This data indicated that there is really a variation in weather situation occurred during the last 10 decadal period. If we consider the rainfall in Gangetic West Bengal region, we find that there is a major shift in the rainfall from 2001 to 2020. That is, we find that the July-August rainfall was increased. That is, there is a more skewed distribution of rainfall we observed during that period of 2011-12 to 2020. And we simply just plotted a disease which is more newly in West Bengal situations, especially in tomato, that is target leaf spot of tomato. And we find that in 2014-15, 15-16 and 16-17, when the average rain temperature of November, December, January, February were high, in this condition, there was a less disease infestation as compared to others. So that's now, for conducting this experiment, we have done this experiment in 3 different districts and under our experimental condition in Kalyani, that is in Nadia, in Hooghly, and in North Chobis Pargona, but which is 30 kilometers of aerial radius distance from our experimental station. In case the tomato was cultivated as a Rabi season from September, October to January, February, and also for summer season from January, February to April, May. And these are the locations where consecutively 10 years studies were conducted. This is a few glimpses of few important tomato diseases: early blight, leaf curl, late blight, sclerotium, also target leaf spot of tomato, bacterial wilt. And these are the experimental plots where one is maintained under protected condition and one is maintained under non-protected conditions without the application of any insecticides and fungicides. And insect pest and disease data were taken for last 10 years. And this is the map of geopathological map was developed, prepared by ArcMap 10.3.1 software. The disease severity near the surveyed regions are based on the probability of the software. And this is the target leaf spot disease, how this spread in these 3 regions. And this is the early blight, and this is the leaf curl disease situation in that particular places. And as per the disease data, we find that especially for Rabi tomato, target leaf spot and leaf curl are the major problem in the Gangetic alluvial region of West Bengal. This is the target leaf spot, but the symptom is very similar to the symptoms of early blight. Coronispora cassicola causes a major economic losses in more than 70 countries throughout the world. And if you see the publication, we found that there have been 451 research documents, reports of Coronispora cassicola from 1983 to 2014. And 82 reports have been published only during 2015 to 2019. We also find our Coronispora importance problem in 2010 and onwards. This is how the disease progressed in the last 10 years in different standard meteorological weeks. And this is the maximum disease severity in last 10 years of different, last 10 years of data of target leaf spot disease of tomato. And whenever we divided the disease spread from 2011-12 to 2015-16 and 2016-17 to 2019-20, we find that the disease was comparatively higher, average disease in case of 2011-12 to 15-16, and is comparatively lower than during 2016-17 to 2019-20. And we find that due to the low rainfall during this period of time and average days of relative humidity above 975% is less in 2016-17 to 19-20 may be related to the incidence. These are the target leaf spot disease of tomato, different types of symptoms. And whenever we studied the pathogen and pathogen symptom, we find in field there is very distinct 5 different types of symptoms that has been expressed. In one, very small dots with or without yellow hallowing. Number 2 type of symptoms we find spatially same of your target board-like appearance with yellowing. One is irregular necrosis, and other is tip necrosis, and last one is the irregular necrosis, which ultimately causing branching and withering of leaves. And we also find there is a difference in also the morphology of the pathogens, colony characteristics of the pathogen. These are in details how the different symptoms appeared in the field condition. We go for the ITA sequencing and we find that this is Coronispora cassicola. Then we go for the UPPCS studies of 5 different types of symptoms along with the pathogen from brinjal and pathogen from tulsi, and we find that symptom 2, 3, 4 remains in single cluster, and symptom 2 is another cluster, and symptom 5, which is very devastating, which is also in another cluster, and on the basis of host, brinjal and tulsi isolates were also in different clusters. Then we find what are the frequency of symptom types in different months. In October, we find there is no symptom type 5 is present in the field, but majority of the symptoms are symptom 1 type in case of your November, October and November. Symptom 2, 3, 4, these 3 type of symptoms we find throughout October to January, but symptom 5, which is very detrimental, we find this type of symptom during December and January. Then we grow this pathogen at 2 different temperatures, that is at 20 and 27 degrees centigrade. We find that at 20 degrees centigrade, the growth rate of symptom 5 is very faster, and simultaneously our field data also validate this thing, because this disease we find more, this type of symptom we find more prominent during December, January, whereas at 27 degrees centigrade, the growth rate of symptom 5 is less. The same was also their in vitro pathogenicity test. Now, we follow the disease grading scale of Horsfall and Barrett for taking the disease data in case of Coronispora cassicola. And for development of the modeling, we go for multiple regression analysis based on Gompertz model, algorithm-based disease prediction model, multinomial logistic regression, support vector system, and 1D convolution neural network prediction model. So whenever we study the biology of this pathogen, we find that the temperature at 20 to 25 degrees centigrade is very optimum for the conidial germination, and especially on the light and dark condition also. These are the temperature condition. And whenever we go for the relative humidity studies, we find the highest relative humidity, mid relative humidity we observed at 95% condition. Based on the conidial germination, conidial growth, and also the effect of temperature and humidity, we try to develop an algorithm. Such type of algorithm we also find in case of potato lead blight developed by Wale. Here, we considered the average temperature 10 to 15, 15.1 to 21, 21.1 to 27, and 27 and above, and average relative humidity 50 to 60, 60 to 70, 70 to 80, 80 to 90, and above 90. And the base of these, we give some scoring
Dr. Datta, your sound is not coming. Something is problem. Some network failure or something. Please check it.
transplanting, we take the data, and when the value approaching 18, we find that there will be disease within 7 to 14 days period of time. In that way, we try to forecast the initiation of the disease, and we find that in the last 10 years, last 6 years, we get the 70% of successful prediction of disease initiation through this index model. We go for weather-based regression, multiple linear regression, algorithmic-based model, and multinomial, and we find that temperature average, RH mean, and RH average, these three are the major important factors associated with the disease development. We first go for simple regression analysis, and we find that 66% of the cases can be successfully explained by this regression equation, where the RMSE value of 0.89, and in the for development of the model, we take the data point from 2011-12 to 2017-18. This is 125 data points we have taken. At 2018-19 to 2019-20, we go for testing of data point, and we find that our testing value is 62.8% prediction accuracy was given with RMSE value of 1.18. Then we go for multiple linear regression of Gompertz for prediction. We convert the data as per the Gompi, and we find that model 3 was effective because its adjusted R-square value is high, whereas the CP and PIC value is comparatively lower, and this model we have selected. Again, we go for multinomial logistic regression analysis. For that, we first go for principal component analysis, and based on this eigenvalue, we go for the classification system based on the multinomial logistic, and we find the different equation, and on the base of this model, we find that there is prediction accuracy in training data is 60.8%, but for prediction data, it is 68.57%. So for whatever the model we are using, that is regression, multiple regression, multinomial regression, that differentiate the data point on the basis of a single line of differentiation. But the support vector machine can differentiate the data point in a hyperplane basis. Therefore, we use this support vector machine for classification of this data on the basis of different grades of the disease through this hyperplane support vector machine system. And we find that here the training accuracy is excellent, 93.6%, whereas prediction accuracy is very lower, that is 54.28%. Then we go for 1D convolution neural network prediction model. Here we find this model, however, training accuracy is 96.72% and test accuracy is excellent, that is 90.90%. And this is the data point, and this is the actual and predicted data point. And we find that the nearly 90% of the data are giving correct prediction. These are the model summary of the different models we have tried to forecast the target leaves for disease. We find that 1D-CNN is the best model for predicting the target leaves for disease of tomato. Then we go for, on the basis of precise regional climatic model output, we get the data from 1991 to 2050 precise climatic weather model. From this, we use the data from 1991 to 2020, that is 30 years climatic average data. And from this average data, we try to understand that be less, we find that up to 2010, the disease will be less, whereas 2011 to 20, the disease will be more. Again, there will be less of the disease, but from 2031 to 40, there will be the increasing trend of target leaf spot disease of tomato will be occurred in that Gangetic alluvial region of West Bengal, based on the precise regional climatic model out. We also have the problem of tomato leaf curl. There are different types of symptoms we observed in the field, and we find that from 2012 to 15 and 2016 to 20, there will be reduction in the disease. And whenever we go for the white fly population, the relationship, we find there is a positive relationship with the temperature and negative relationship with the RH max and RH mean and RH average. Simply, we have also find that the higher average temperature, the higher T mean, and also the lower T mean and T average and higher relative humidity was associated with, and high rainfall is associated with lower 2LCB infestation during this period of 2016 to 20 as compared to 2012 to 15 period. Lead blight is one of the most devastating disease, but in all the year we have not observed the lead blight disease. Lead blight disease is frequently observed in different years, and whatever the important criterion for lead blight, we find that RH max, RH mean in the period of especially of January and January, RH average, rainfall, and also foggy days is important. Number of days T mean less than 10 is also the very important. In the year, the number of days less than 10 is more, there will be high chance of the disease development. And disease trigger day is considered as the night at which the temperature is above 10 and the day where there is foggy days is there, or there is a drizzling days is there. These are considered as disease triggered day. And we find that majority of the year, the disease triggered day is very less as compared to 2011-12. There was disease triggered day was more, and also 2015-16, the disease triggered day was more. If you consider 2001 to 10 and 2011-12 to 2020, we find that number of days T mean less than 10, it is high in case of 2011 to 20. Foggy days is very low, RH average and especially the RH max is low. Therefore, in 2011-12 to 2019-20 period, we observed the less attack of lead blight disease as compared to the earlier period. And 2012 to 15 and 2016 to 20, whatever the lead blight situation is occurred, we find there is more in 2016 to 20, and this is associated with the more number of foggy days, the disease triggered days, and RH average. Thus, for lead blight, as in West Bengal situation, we have already find that last 10 years, only 2 years we find the rainfall, that is 2011 and 2020. And rest of the years, we won't get the rainfall in case of month of January, more than 20 millimeter of rainfall. Therefore, fog is one of the most important criterion for lead blight development under Gangetic alluvial region of West Bengal. Whenever we go for the classification of lead blight, we find there is the data are very imbalanced. The class 0 having 76, class 1, 22 data point, class 2, 22 data point, and class 3, 6 data point. Whenever we maintaining the ratios of this data, we find class 0 to 3, 13:1, class 0 to 1, 3:1, class 1 to 3, 4:1. Therefore, to smoothening the data classification of data classification, we go for synthetic minority oversampling technique. That is mode technique we are using. As mentioned in the first example, that there is a huge imbalance in the data representing each class. Thus, to ensure the testing mainly done on real data, we are using the testing in real data by biasness way. We are doing 80 to 20% of the split data for training and testing. And for 20% of test data, more the real peak data we take for the validation. How we develop? We get the data set, generate the synthetic data based on spot technique, normalize the data, then feature elimination we done, and ultimately required number of iteration we give, and ultimately the model we develop. For algorithm we use, that is a smart techniques we use, recursive feature elimination we use, and we also use random forest estimator. And by 10 iteration, these are the 10 iterated data, and by applying the above algorithm, we were able to achieve around 75 to 80% of accuracy. Till that, for lead blight model, we are not gaining more success for forecasting development. This is the early blight situation, and also the early blight disease of
hello, Doctor. You are already crossed the limit time. Kindly we short.
Okay.
Kindly short it immediately.
Okay. And the early blight disease is more in case of 2016 and 20. High relative humidity and rainfall is associated with this, and especially we are just in giving the examples of two important pests, that is Spodoptera, and Spodoptera is really problem, and we predict that there will be an erratic distribution of Spodoptera through up to 2050 period. But the major problem now everyone is facing, especially in West Bengal, that is leaf miner problem, and leaf miner is very important problem in present day tomato cultivation. And we also developed the linear regression model and also the multinomial logistic regression. And based on this approach, we go for basis on the precise model, we find that there will be continuous increase, and leaf curl will be the future problem of tomato. These are the conditions. From 2001 and onwards, there will be a huge increasing of leaf curl problem we will be facing in tomato. And simply one experimental study, we indicate that at 15-degree average temperature, the male and female take 60 to 63 days of time to complete their life cycle. Wherever we increase the period of 30-degree, it requires 16.4 to 17.9, 16.4 days to 17.9 days. Really, we find that nearly 3 to 3 times reduction in their life cycle occurs in the increase of temperature. Thus, this will be a really challenge in the future for tomato cultivation. I acknowledge NICRA, NCRPM, ICR for providing fund for conducting this research, AICRP and Meteorology for getting the agromet data, Department of Information Technology and Computer Science, Kolani, KGEC Kalani, and Department of Computer Science and Engineering, Jalpaiguri Government Engineering College. Thank you, and thank you, the office bearer of IPS, for giving the chance for presenting. Thank you.
Thank you, Dr. Doctor. Now, no chance to answer any questions. I totally gone to for oral presentation. Already time is up. Okay. First, we call for oral 5-6 oral presentations are there. I call Girish Chand for his first presentation.
Tell sir. No and tell sir. Please tell them to
Sir.
Hello.
Sir, please tell oral presenters to stick on the time, sir.
Only 10 minutes. Only 10 minutes for every presenter. 10 minutes and 2 minutes for discussion. Any questions?
8 minutes for the presentation and 2 minutes for discussion.
10 minutes for presentation and 2 minutes for discussion.
Yes, sir. Sir, another thing, sir, totally 5 oral presentations. So the best two best oral presentations may be selected, sir. And please send the details to our zonal president.
Okay. No problem. Okay, Girish Chand. Hello. Next, polymyxil, screening of chili genotypes grown in North Bengal for resistance against dengue viruses.
Sir, she is not presenting.
She is not presenting.
Sir, yes, sir. She is not present.
The third, Girish Chand is absent.
Sir, he is there.
Paulomi is absent. Next,
Type type.
Ranjit Kundu. I think Ranjit Kundu not present. Next. Next paper, development of a rapid leaf detached assay for the phenotyping of spot blotch of wheat. Start. Who is presenting?
Sir, 1 minute.
Hello? Hello, sir. Am I audible?
Yes, you are presenting.
Yes, sir. Sir, I'm Sunanda Chakravarti and I'll be presenting my seminar.
So, yeah, please share your video. Sir.
Yes, sir.
PowerPoint.
Slides.
Slide.
Tell them also to ready with their
Sir, is my screen visible?
Yeah, you are visible. Yeah, your slides.
Please present.
Please
Start
present.
Yes, sir. So, good afternoon, sir. I am Sunanda Chakravarti and I am going to be presenting my seminar on development of detached leaf assay for the phenotyping of spot blotch of wheat. So, the question arises is why spot blotch? Wheat is grown around 730 million tons globally, out of which 170 million poor family members are supported by the cultivation of wheat. But spot blotch causes around 15.6 to 19.6% of global yield loss throughout the globe. It not only affects the non-economic parts, that is leaf and sheep, but it is also causing significant losses to the economic portions of the plant, such as the nodes, the blooms, as well as the grains that causes black spot. The pathogen exhibits dark brown lesions which enlarge and coalesce, and that leads to yellowish discoloration and finally the death and the drying of the entire leaf. That causes losses of the uneconomical part but indirectly affects the yield as well. It leads to grain shrinkage, the discoloration of the grains, that leads to around 60% yield loss in the country itself. The causal organism is Bipolaris sorokiniana that prefers warm and humid climate. Around a temperature of 18 to 32 degrees Celsius is favored by the pathogen. The control is basically achieved by managing different kind of strategies, out of which resistance breeding forms the most important of them. However, resistance breeding in the field condition is both time and nutrient intensive. So the need of the hour becomes to introduce a rapid and reliable assay that can detect the resistance as well as the susceptibility reactions of the pathogen against various genotypes. So keeping that in mind, four objectives were set. The first one was to identify a highly virulent strain of the pathogen Bipolaris sorokiniana. Second was to identify resistant and susceptible sources among the promising genotypes that are grown in West Bengal condition. The third was to compare different screening methods, that is field, greenhouse, and detached leaf assay, using partial disease resistant components in case of detached leaf assay as well. And the fourth, to draw a comparison with the help of PDR components and correlation study in the detached leaf assay. So for meeting up the objectives, firstly, 10 isolates were collected from different places in West Bengal condition. The accession numbers are given in the table along with the latitude and longitudinal information. So taking all these 10 isolates, the screening was done under greenhouse condition. The mean disease scores were recorded And out of all the 10 isolates, the isolate 7, that is BS7, exhibited the highest mean disease score, indicating that it is the most aggressive among the collected isolates. Now, with the help of the isolate collected, we assess the different screnotypes. Field screening was done using Sari-Prescott double-digit score. The greenhouse and detached leaf assay was done using the Liatkos and Rusga scale of 1 to 9. For using this assessment assay, the isolate BS7 was utilized as it was observed to be the most aggressive. And in case of detached leaf assay, the PDR components IP and LP, that is incubation period and latent period, were considered. Now, with the help of the isolate BS7, we screened the genotypes, 15 genotypes, under field conditions. It was observed that the genotype DBW39 exhibited resistant reaction among the 15 genotypes observed. It showed the least disease severity in all the 3 grade stages observed, as well as the AUDPC value was lowest in this genotype. Many other genotypes was exhibited moderately resistant and moderately susceptible reaction, while 2 genotypes, PBW703 and K1317, exhibited susceptible reaction. Here is a graph depicting the overall assessments that we observed. The bars indicate the disease severity observed, and the lines indicate the AUDPC value that was observed under the field conditions. Followed by that, assessment was done under all the 3 conditions, that is field, pot, and detached leaf assay. For field conditions, the 15 genotypes that were grown were assessed using the Sari-Prescott double-digit scale as mentioned earlier. But for the plant assay and the detached leaf assay, we used the Liatakos and Rusga scale, and as a result, the mean scores observed were plotted on the basis of a tabular form. Here, it was observed that the DBW39 exhibited the least mean score under both conditions, whereas EBW703 exhibited the highest mean score under both the conditions, exhibiting a resistant and susceptible reaction respectively. 5 genotypes exhibited moderately resistant reactions, while 8 genotypes exhibited moderately susceptible reaction. The partial disease resistance components, that is IP and LP, incubation period and latent period, were also exhibited by the different genotypes, and they were plotted in a tabular form. It was observed that the IP values were lowest when the mean disease score was highest, and the same trend was observed in LP values as well. Now, to draw a comparative analysis between all the genotypes, we plotted the AUDPC values exhibited by the 15 genotypes that were taken. It was observed that the AUDPC value under field condition, pot condition, and detached leaf assay was found to be the lowest in DBW39, and it was found to be the highest in PBW703. We had earlier observed that DBW39 exhibited a resistant reaction and the PBW703 exhibited a susceptible reaction. This indicated that the AUDPC values could be used as a measure for indicating the resistance and susceptibility reaction of the pathogen against different wheat genotypes. This is a graphical representation of all the AUDPC values observed under the different screening techniques. Here we observe that the field data exhibits a significantly higher reaction as compared to the other 2 screening techniques. However, under field, greenhouse, and detached leaf assay, the trend that we see among the genotypes is similar. That is, the genotypes that exhibit the highest AUDPC values under field is also the highest under greenhouse and detached leaf assay, while the lowest that is exhibited is also the same in all the 3 screening techniques, as observed from the graph. Finally, a correlation study was drawn between the field screening, the pot, and the detached leaf assay. It was observed that the field, pot, and detached leaf assay all exhibited significantly positive relation to each other. This indicates that the screening of Bipolaris sorokiniana for the 15 wheat genotypes can be conducted successfully using any of the 3 methods, that is field, pot, and detached leaf assay. However, DLA method takes considerable less time, effort, and resources as compared to pot as well as field screening. Also, Field screening takes a lot of time, and it is also exhibited the reaction on the basis of the environmental condition. That is, it is prone to give false positive or false negative reactions, which is eliminated in DLA process. Furthermore, the WPAF technique, that is the field assay, also utilizes a number of other nutrients such as the fungicides and insecticides, which are also eliminated in DLA, making it more economically feasible method of screening the genotypes. The correlation study was drawn between the partial disease resistance components, that is the incubation period and latent period, under detached leaf assay. Here, we observed that a positive correlation was observed between the partial disease resistance components among each other. However, the negative correlation was observed in case of PDR with the detached leaf assay disease score. That is, PDR components indicate that if it is more, then the mean disease score exhibited will be less. That is, the more resistant the genotype, the higher will be the PDR components, and the least resistant, the PDR components will be lower. Finally, the results that we observed on the 4 objectives mentioned earlier was that the isolate BS7 was found to be the most aggressive out of all the isolates collected from different places. The DBW39 genotype and PBW703 genotype exhibited resistant and susceptible reactions respectively. 5 other genotypes exhibited moderately resistant, and 8 other genotypes exhibited moderately susceptible reaction as well. The correlation between the AUDPC values observed under field, greenhouse, and detached leaf assay were found to be positively significant. And finally, the incubation period and the latent period as PDR components express significant negative correlation with the disease severity that was recorded under the detached leaf assay. So with this, we can say that among the isolates that we observed, isolate BS7 can be further utilized for different susceptible or resistance reaction studies in the future under West Bengal conditions. Now, for our future outlook, we can say that genetic variability of the different genotypes that we observe plays a huge role in deciding the resistance or susceptible reaction. So for future works, we can analyze the molecular basis of the resistance, that is whether it presents the resistant gene within its genetic material, so that we can use these genotypes for future breeding program. The use of detached leaf assay can be further expanded to other foliar pathogens of wheat. The morphological characters of the wheat genotype, such as the arrangement of the leaves, can be used as resistance response, so that we can screen the wheat varieties easily and before time. And the fourth one is the PDR components, such as lesion size and conidial count, can also be used for assessing the rapid detached leaf assay, and works need to be conducted on using them as a reliable tool in the rapid detached leaf assay. Finally, I'd like to conclude by saying that DBW39 that we observed throughout our study exhibited a resistant reaction under various conditions, field, greenhouse, as well as detached leaf assay. So it can be utilized in breeding programs as a resistant genotype against Bipolaris sorokiniana. Secondly, the PDR components that we studied, that is latent period and incubation period, can be used for identifying resistant and susceptible reactions within the genotypes rapidly without using land, fertilizers, fungicides, etc. Third is the detached leaf assay forms a reliable tool, as we have observed in our study, against Bipolaris sonokiniana, and it can be used in the future for fighting different fungal or foliar pathogens of wheat crop. Thank you.
Thank you, Shomanda. Any questions from any? Hello. Any questions? Am I audible?
Yes, sir. Yes, sir. You are audible.
Okay. 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 Next. No questions. Next. Ashish Kumar Anand.
Yes, sir.
Kindly you immediately present your 10 minutes for presentation and 2 minutes discussion.
Sir, 8 minutes is the presentation.
Yes.
8 minutes, sir.
Okay, sir. 8 minutes. Okay. Start. Ashish. Your paper is evolution of brown plant hopper, Nila Parvata lugans, resistance in rice varieties.
Assis, you can share your slides.
Yes, sir. Yes, sir.
Assis?
Yes, sir. I'm sharing my slides, sir.
Oh, Sunanda Chakrabarti,
Sir.
you ask him to unshare his Sunanda Chakrabarti. Sunanda Chakrabarti. Sunanda Chakrabarti, please unshare your screen.
Sunanda Chatterjee.
Sunanda is already present. Already have completed.
Sir, ask her to on screen.
Ask the screen.
On screen.
Yes.
Okay. Okay.
Just to close.
Sunanda, you also you are viewing Sunanda Chakrabarti screen. Why?
Okay.
Now.
Okay.
Sir, how I share my slide?
Ananta, Mr. Anant, you will share your screen.
Now you share your screen.
Anand, there will be one icon share screen, green color share screen. Just a green color arrow.
Downside is bottom of the
In
slide.
the bottom, you can see one green color arrow. Just share screen.
Yeah.
Okay.
Good afternoon, all of you. My name is Ashish Kumar, PhD research scholar agriculture entomology. My presentation topic on evolution of brown plant hopper Nila Parvata lichens resistance in NRI released rice variety. So let's start with introduction. Rice is the essential cedar crop and regarded as the principal food crop in the world, and especially in Asia where rice is the prominent growing in Asia. Rice productivity is adversely affected by numerous biotic and abiotic factors, and approximately 52% of the global production of rice is lost owing to the damage caused by biotic factors, of which 21% attributed to the attack of insect pests. Among those insect pests, brown plant hopper is one of the major destructive pests of rice and causes significantly huge yield losses in every year in tropical, subtropical, and temperate region of the rice. There are two types of damage can be done by brown plant hopper. First is direct sucking the sap, mainly on leaf sheath, which turn into browning and drying up to the plant, which is known as hopper burn. Apart from direct losses, it is also act as vector that can be transmit viral infection, resistant virus and drug resistant virus. Among those management strategy, host plant resistance is the regarding best one for control BPH. Management of this resistance variety will contribute to developing strategy for BPH control because it is environmentally friendly and non-toxic to natural enemy and other organisms, and reduce the use of chemical pesticide. Screening of rice germplasm at global level and breeding of rice variety were initiated during 1970, and several resistance variety have been released for cultivation. However, the mechanism of plant resistance to pest is poorly understood. In this analysis, resistance to BPH has not yet been explored in NRI released rice variety, and their mechanism of BPH is not well documented. Hence, our present study mainly focused on investigation was aimed to assess the BPH plant hopper resistance status in NRI released rice variety. The experimental material consists of 99 NRI released rice variety were collected from rice gene bank NRI Cuttack, Odisha. Likewise, 10 resistant gene differential seed were collected from All India Rice Coordinated Rice Improvement Project program. The 99 NRI and 10 resistant gene differential seed were screened against brown plant hopper using standard seed bus screening technique. Observation was recorded on the basis of visual plant damage symptoms 0 to 9 scale for each genotype, when all the TN1 registable check, TN1 completely died to BPH fatigue. A total of 99 NRI released rice variety were evaluated against brown plant hopper, of which only 2 variety, namely Lunisri and Luna Samper, had least plant damage score and categorized as moderately resistant, and remaining all other varieties were found as susceptible to highly susceptible. In our present study suggested that frequent outbreak of BPH in Odisha correlated with cultivation of popular variety which are lacking BPH resistance gene. Although these rice variety have been released to attain the higher yield, at the same time it is necessary to incorporate the resistance in this popular variety through advanced breeding technique to reduce BPH related yield loss in farmer variety. Additionally, changing climate scenario also favor the BPH multiplication, and in that condition, it essential to develop climate smart rice variety which could handle both climate and BPH pressure. Further, to ensure the resistance level of brown plant hopper, 10 resistance rice assessment containing different BPH resistance gene were evaluated against brown plant hopper. Although these all variety have been previously reported as resistance in terms of BPH infestation, but in our study revealed that the out of 10 varieties, only 2 variety PTB33 and RP2068 were found highly susceptible, and other variety were found as susceptible to moderately resistant. Although these all varieties possess BPH resistance but could not sustain BPH pressure. In present experiment suggested that Indian brown plant hopper, which term as biotype 4, broke the all gene except BPH3 gene, and combination BPH2 and 3 gene proved resistance against Indian brown plant hopper. Besides several resistance variety which are resistance in the other Asian country are susceptible in Indian because the Asian brown biotype of brown plant hopper are more destructive than the other biotypes. Developing rice variety containing BPH resistance might be efficient strategy to control BPH. This finding will provide into host plant interaction and further molecular mechanism of BPH resistance gene and exploit the resistance gene in rice breeding. Notably, changing climate scenarios also favor the BPH multiplication, and in that condition, it is essential to develop climate smart rice variety which could handle both climate and BPH pressure. His future management BPH program should be focused on incorporating biotype 4 specific BPH resistance into popular variety, either alone or combination by parameting gene, with which minimize the yield losses against BPH population. Thanks.
Thank you, Anant. So one question to Anant. Anant?
Yes, sir.
Whether BPH is severe on stagnant water or dry field?
What did you say, sir?
BPH infestation is more in stagnant water in rice field or dry field?
Stagnant water, sir. More infestation are generally wet and conditions, sir.
Water logging condition, BPH is severe.
Yes, sir. Yes, sir.
Okay. Another questions? No questions. Okay, next man. Bhayapatti Ravi.
Okay, sir. Respected dignitaries, honorable chairman and co-chairman, professors, scientists, students, as well as all participants. A very good afternoon to all of you. Today, my presentation topic is cultural characterization and parasexual recombination in Pyraclaria graecina, which causes blast of rice. Myself, Boypati Raviteja Naidu, I am presenting. At an outset, rice is nearly life for more than 3 billion people. So 90% of production occurs in Asia and 60% consumption also done in Asia itself. So here one bar diagram depicting clearly the productivity of world is 4,630 kg per hectare, but India 2,600 kg per hectare. Now, if we consider world with India, India productivity is less. If we consider India productivity with Jharkhand, it is still less, 1814 kg per hectare. Why this was less? It is because of biotic and abiotic factors. Abiotic factors can be managed, but biotic factors, most severe diseases like rice blast, false mud, sheath blight are most severe in Jharkhand state. Now I am telling about rice blast. It is one of the most destructive fungal diseases in the world. According to Commonwealth Mycological Institute, they told that cultural characterization was different in both quantitative and qualitative characters. So although we know rice blast can break down the resistant rice varieties within soon time, it is nearly 2 or 3 years within soon time after release of resistant variety, the resistance nature can be broken down by Pyraclaria graecina, rice blast, causal organism. So the parasexual nature is most important mechanism in bringing gene recombinations in Pyraclaria graecina. According to Noguchi et al. 2017, he told that parasexuality is the only cause of genetic recombination in Japan. I quote again, parasexual recombination is the only cause of genetic recombination in Japan. It will bring various races in Pyraclaria graecina. But no work was done on parasexuality mechanism in India on rice blast. So I am discussing about parasexual and cultural studies. Now coming to methodology, I collected 50 isolates from the state of Jharkhand in 7 districts, 30 mandals, out of which the disease intensity calculated as per ERI rules. The Ranchi is having highest disease incidence 38.3, followed by Garba, Palamu. Lowest disease incidence was found in West Simbhum, that is 24%. All standard isolation techniques were taken and measured. After calculating cultural quantitative and qualitative attributes, the H-cluster R version dendrogram was made based on the attributes showing. Now coming to parasexuality methodology, here the 50 isolates, out of 50 isolates collected, only one isolate, Potka village, West Simbhum district, got parasexual nature. It is only directly found in nature only, not through artificial cross combination. It is first time. Parasexual nature was found and studied in scanning electron microscopy and compound microscopy through bioassays also. It is nothing but heterokaryokinesis in which haploid is converted into diploid. Now, after confirming the results of parasexuality of rice blast, the pathogenicity test was done. So now coming to result, here cultural variability clearly depicting all the qualitative and quantitative is most severe in Ranchi. So as I quote before, in Ranchi, 38 disease intensity is there. So color, sectoring, zonation, wrinkle, radial growth, spore germination. If we take any attribute of cultural characterization, it was high in Ranchi followed by Garwa. As per the disease intensity, cultural characterization also varying and showing different nature variations. So here I am showing 50 spore picks according to conidial ontogeny, 50 isolates here very clearly changing. So from pyriform to obculvate, the spore conidial ontogeny itself is changing here. So from this, the natures can be identified. So the Petri plates here showing here zonation, sector formation, wrinkle, circular raised growth also mentioned, and these all are of 50 isolates, mostly varying in nature. Based on this, the varying nature can be identified. This is the H cluster dendrogram, which was made on the 50 isolates. Out of this was all made into subgroups A and B in 3 groups. The first group, which was depicting 6 isolates, as I shown here, 6 isolates is most showing high variation, high amount of radial growth in both qualitative and quantitative characters. It is much faster, and these isolate areas also showing great amount of variation and causing destruction also through disease incidents. So group 3 was showing low disease incidence and showing lowness nature also. So from coming to germination of Pyraclaria graecina, middle cell germination is the first time reporting in the world. From middle cell also germination taken place in isolate of West Simbhum, followed by apical and basal cell germination also taken place. Now, if we see 4E, it is of both apical and basal cell germination. This is also first time reporting. The germination is varying in case of Jharkhand state, especially. Now coming to cell number also. Actually, in more books and more literatures, it was written only 2-celled and 3-celled Pyraclaria grigia spores are present. But in my study, single cell also were found out in Pyraclaria grigia along with spore also. This also might be possible to produce single cell and causes variation in causing diseases. So now coming to sympodial nature of rice blast. This sympodial nature also in literature it was already mentioned that 3 to 5 spores are arising from the sympodia, but here up to 8 cells was arising in scanning electron microscopy pictures also. So now coming to cultural characterization analysis. After doing this all cultural characterization analysis, spore germination assay was done with fungicide and fungistatic action. So nirgundiol and citronella oil suppressing the Pyraclaria grigia growth, carbendazim showing the fungicidal growth. So from this easily we can estimate that the cultural variation can be easily stopped with the essential oils without interruption of environment. So now coming to parasexuality, most interesting topic here. From nature clearly depicting symptom I was taken and kept on Petri plate. It was shown 2 different sectors was formed in single Petri plate without any interruption, without any cross combination also. From nature directly I brought a sample of symptom which was clearly depicting rice blast. After that I got parasexuality clear sectoring. Then I went for scanning electron microscopy before I did ITS also for analyzing rice blast with ITS 4 and 5. After that, the Pyraclaria spores I was mentioned here clear scanning electron microscopy pics. This half of sectoring shows little jump tube. This half of sectoring shows more jump tube. And here melanized hyphae is more. Here melanized hyphae is less. And both myceliums also, the spore and mycelium sizes also varying in parasexuality. The most interesting topic here is from the tip cells, the nucleus from one hyphae is exchanging into the another hyphae. So there is a presence of aneuploidy condition. Out of these confirmations, I did pathogenicity test in resistant rice variety, that is IR64DRT1 pathogenicity assay by taking this spore suspension with the help of inoculation I did by rubbing method. So after this, I got the resistant variety breakdown nature within 7 days. After showing the resistant variety is converted into susceptible, within 7 days, I got clear-cut symptoms of rice blast in IR64DRT1. So the conclusion is in Jharkhand, rice blast is endemic in nature and causing more yield losses. If we know all these mechanisms, it is helpful for us to identify the mechanism and can create our genes to control these rice blast yield losses in not only in Jharkhand but also in India. So there should be requirement that parasexuality studies should be taken up for further studies. Out of cultural characterization, 3 groups I was analyzed in Jharkhand, 7 districts, out of which 6 isolates were most severe and causing enormous amount of disease causing. Group 3 was commonly observed. So parasexuality nature was found in natural condition only without interpretation of cross combination that can cause breakdown in resistant rice varieties. Thank you. Any doubts you can ask.
Hello.
Sir.
Bhayapati, you have seen parasexuality in one region, na?
Only one region, sir. I observed for all regions, but only in one region sectoring was clearly depicted, sir. So I have taken for that region only. Even I did artificial combinations also, cross combination, but that is not breaking down any resistant variety. Even I did in from different isolates, I did cross combination mating technique also, but the susceptibility was not gone.
Okay.
Sir, I have one question.
Yes.
Okay, sir.
Really excellent work and good.
Sir.
I have a few queries if you allow, if sir allows me. That is,
yes,
is there a sexual reproduction you have observed?
So, sir, actually, I observed disease cycle also. Only one perithecia I observed, sir. That is another topic in my work, but I didn't mention here. No sexual cycle was found. Only perithecia I was observed, sir.
Then why in Jharkhand the blast disease is so severe?
Yes, sir. In Jharkhand, it was so severe, sir. It is a hotspot, sir, mostly due to weather conditions.
What is the reason?
Sir, reason is nothing but they are improving traditional varieties also. They are not cultivating the resistant varieties, and mostly suitable environment occurs in Jharkhand state, sir. Most suitable environmental condition. If I went to 7 districts in each district, each mandal, disease incidence was severe, sir. I observed.
In one slide, you showed the single cell conidia.
Yes, sir. Here I'm showing again.
Are you
Here,
sure?
sir. Yes, sir. Sure, sir. 100% damn sure. It is single-celled conidia along with Pyraclaria grigia.
Let me tell why I'm telling, because usually the blast force of the your triple cell, okay?
Yes, sir.
But double
Yes,
cell,
sir.
seen even I have seen also very elongated type of conidia. I have some photographs with me also.
Okay, sir.
Your single cell,
Yes, sir.
that if you let it mature, then it will
Yes,
come.
sir. Yes, sir. I did matured also, sir. I did humid conditions only I kept, but single cell remained same, sir. But apart from conidial remaining sites, the triple celled spores was seen, sir. Actually, triple celled spores was seen only in field condition, only in laboratory condition. Two celled I mostly observed in field condition, sir.
No, but triple cell also is present in field condition. I have collected many.
Yes, but mostly in Jharkhand state, sir, I got only two celled in leaves, sir. In field condition, in laboratory condition, 3 cell I got mostly, sir.
Thank you.
I think it needs several experiments and other collections.
Yes, sir. Yes, sir.
Circumstant.
Yes, sir. Yes, sir.
Okay. Thank you, Bhayaputti. Next.
Thank you so much, sir.
Shamsetti Ravali.
Yes, sir.
Okay, you present your lecture.
Okay.
Last phone number nahi hai.
Somsetti.
Yes, sir.
Yeah, your Word file is opening.
Yes, sir. Yes, sir. I am sharing through phone, sir. I have problem with laptop. Sir, is it visible?
Yes.
Visible. Okay, sir. Thank you. Good afternoon to all respected dignitaries, professors, and scientists of the symposium, and myself, Somshetti Ravali, and I am presenting the topic name Effect of Agro-Meteorological Factors on Development of Sheath Blight Disease of Rice. And I'm going to give a small introduction about the rice. Rice is the second most important cereal crop after the wheat, and it is the staple food for 60% of the world population. And in India, it is the primary source of calories for more than the 70% of population. And if we observe according to 2019 data, in India, production was 116 million tons and productivity 3.91. Under Bihar condition, it is 8 million tons and productivity 2.45. And rice is subjected to the various biotic and abiotic stresses, which is the causes up to the 50% of losses. And under the biotic conditions, mainly the biotic stresses, mainly the insect and fungus bacteria causes the most severe losses in the rice. And nowadays sheath blight also is the prominent disease and economically important disease. It is causing various losses in the rice. And sheath blight of rice is caused by the Rhizoctonia solani. Already the madam that Sureka madam has already told how the rice and the pathogen is interacting with each other on the host surface and intercellular surfaces, and it causes this pathogen causes the mainly disease above the waterline at the stage of the maximum tillary stage of the rice growth. And sorry, sir. And why this is the most important, it is the most important. Sheath blight is considered to be an important disease next to the rice blast. Is a increasing concern for the rice production, especially under the intensified rice production system, and it is causing the losses up to the 20% in Japan conditions, and also in China, it causing the losses 1.97 times over the rice blast, and in India, in the West Bengal condition, it may cause up to the 50% losses under the favorable environmental conditions, and in Bihar condition also it may cause the 40% condition. And these two states are mainly hotspot for the sheath blight disease. And for the need to understand how the this these sheath blight is affecting in this condition, a need us well to how the environmental factors affecting on the development of the sheath blight disease under the Bihar weather conditions. For that objective is I have selected was to establish a correlation between changing climatic factors for the development of sheath blight as caused by Rhizoctonia solani. For that, I have selected a susceptible variety MTU 7029, which is the locally called as Swarna variety. And for that better understanding, this was sown for 3 times, 3 times with the and it was transplanted 20 days after sowing. First rice was sown on 4th July, and second one is 19th July, and third is the 3rd August. And this is in these rice varieties, it was correlated with the maximum temperature, minimum temperature, relative humidity, evening relative humidity, rainfall, and evaporation. This is and this is mainly experiment was conducted in the RPCAU during the Kharif 2019, and the weather data is obtained by the meteorology department of the uh, college. And here the results of this experiment was percent disease severity and meteorological data for 10 days was has taken. In this, percent disease severity was taken as the um, cumulative CIDS, cumulative increasing disease severity, and PIDS, which is the percent disease increment in disease severities. And the data was taken observed from the 30 days after transplanting up to the 110 days of the transplanting. And in the first sowing rice, that is was considered as early sowing. In this, first disease appeared first disease was observed after 40 days after transplanting. That is the 2.5%. It is the temperature 34 degrees Celsius, and minimum temperature was 97 degrees Celsius, and the maximum relative humidity is 86%. And we can observe here the rainfall is a 110 mm. And also the periodical increment in disease severity was continuously increases up to the 80 days after transplanting. And it is cumulative disease severity was in the first sowing rice observed is 34.5%. And in case of second sowing, normal sowing rice, the disease severity was observed after 30 days after transplanting, at which the minimum temperature, maximum temperature was 26 and 33.3 degrees Celsius respectively, and the rainfall is the 1 or 2.5. Uh, here, here periodical increment is disease severity is continuously increases up to 80 days after transplanting, and the cumulative increasing in disease severity was after 110 days after transplanting was 43.5%. And here late showing in which disease severity is observed after 30 days of initial disease severity was observed 30 days after transplanting only. Here initial stages rainfall is very high, so the in the late showing the initial stages disease severity is increasing continuously, and it is increases up to the cumulative disease severity increases up to the 52.5%. So this is the graphical representing of how the initiation and the progress of disease was occurred during that crop growing period along with weather parameters. Here the blue color bar indicating the cumulative increasing disease severity, and red color representing the periodical disease increasing severity, and these lines are weather parameters. And we can observe here that the temperature ranging from the temperature ranging from the 30 to 34 degrees and the highest rainfall, which is the uh, prone to the initiation and the progress of the disease in the rice. And how these weather parameters and disease was correlating each other in this maximum temperature is significantly correlated with significantly negative correlated with the disease in all conditions. That is negatively means it is the from decreasing 34 degrees of temperature to 30 degrees, the disease is increasing, and also minimum temperature also negatively correlated with the disease severity, and relative humidity, morning relative humidity positively correlated. That means increasing relative humidity is prone to increase the disease severity in the rice. That is means it is above the 80% of relative humidity is most congenial for the disease development. And also rainfall is negatively correlated with the cumulative increasing disease severity, and positively correlated with the periodical increment in disease severity. And this is the regression analysis for the how environmental variables and the disease severity of the risk is correlating. Uh, here the R values are representing here that 75% to 98% of variation in the disease development is mainly due to the variation in the weather parameters. And conclusion of the studies, so it can be concluded that heavy rainfall was congenial for initiation and the progress of the development in the seedbed rice, and at the maximum temperature of 30 to 34 degrees Celsius and minimum temperature of 17 to 24 degrees, coupled with the 70 to 86% of evening relative humidity, was shown to be the critical parameter for disease development. And also it can be said that late sowing crop was more prone to disease development, followed by the normal sowing and early sowing crop. And here the disease severity is significantly correlated with the maximum temperature, minimum temperature, evening relative humidity, and evaporation. Thank you, sir.
Okay, Samsetti Raveli, I am asking you a question where you have mentioned periodical increment of disease. What does it mean? I don't understand. Periodical increment means you are taking data 10 days interval.
Yes, sir. That is the periodical means the variation in the succeeding cumulative increment in disease severity.
No, cumulative increase means total.
Yes, sir. Cumulative.
That is why not calculated AUDPC?
Sir?
Why not calculated AUDPC? and periodical increments, that is ki rate of increase.
Yes, sir. Yes, sir. Rate of increase only, sir.
The periodical increase means rate of increase.
Yes, sir.
Man per day increase. Rate of
Sir,
increase means per day increase.
Per 10 days interval increase, sir.
No, 10 days interval you are giving the data.
Yes, sir. Yes, sir.
This is the 10 days interval.
Yes, actually, this is the 10 days interval. In this 10 days interval, how the
this is
increasing
the 10 days interval data, right?
Yes, sir.
30, 40, this is the 10 days interval data. Okay?
Yes, sir.
And this is cumulative means this is the total.
Yes, sir. Cumulative is the total.
Last. What is cumulative increase?
Actually, it lost 43.5 at the case of
I think here 0.5 and here 0.5. What does it indicate?
That is the that in that 10 days period, and it means before
This is
that
the primary.
it is a zero. Yes, sir. Primary.
This is primary.
Yes, sir.
Just this is the 10 days interval.
Yes, sir. Actually,
43.
Yes, sir. After 40 days after interval, it is 3.5, sir. But in this period, it was increased as 3.
Means
Yes, sir. It is
3.5
a
minus
periodic.
5.
Yes, sir. Yes, sir.
4.3 minus 8.
Yes, sir.
This
Yes,
is the
sir.
minus.
Yes, sir.
So what does it mean? I don't know. Where put this data?
Actually, how the disease is for every how the it is increasing, how the it is periodically.
Why does not mention the rate? I think it will mention the rate. Then you can calculate the temperature, humidity. Then you can go to data. I think if you calculate the rate, this is the primary, then increasing, and you calculate here rate. Rate means per day increase. You have per day increase in date and this meteorological data. Then you got the particularly regression coefficient, a good result. Okay.
Okay, sir.
I think you must calculate it and you can get a publication if you calculate this procedure.
Okay.
Okay. Any questions? Any questions? Okay. Thank you, the organizer. The total oral presentation is complete. I thank you, the organizer, and also the Eastern Zone IPS for selecting me as a chairman and giving me the opportunity to share the session. In the first half, two invited lecturers were there. Am I audible?
Yes, sir, you are audible.
In the first session, two invited lecturers were there.
Dr. Manas, you mute the Himadri Shekhar. Mute Himadri Shekhar.
Okay. Two invited lectures were there, and these two invited lectures are very informative.
Manos?
Yes.
Himadri Shekhar, you just mute.
Mute Himadri Shekhar.
Mute all sir.
Particularly two invited lectures, two were very informative and innovative also there. And particularly Dr. Subodh Datta presented a very a good lecture, but it is very lengthy. I think if he short it and give a two-part, then it will be very informative. But the first lecture was very, in case of host presentation, very informative and very collaborative, and also very good lectures were there, two lectures. And another 4 lectures were also good, but as they are students, some limitations are there. That's why the presentation is not good. Good. I think. Hello, Secretary. I will send you the 2 best lectures among them. the name of the two persons later on. And whether another, my co-chairman, where is my co-chairman?
Sir, she is having a meeting. She has permission and left.
Okay. I will contact with co-chairman regarding this, whether he has learned or whether he has. Otherwise, thank you very much, Dr. Arum Mukherjee and Dr. Manas Bag for this COVID-19 serious pandemic condition. You have arranged this IPS webinar seminar. Thank you very much. I think this seminar will be a good success.
Thank you, sir. Thank you very much. Thank you for accepting our invitation.
Okay, sir. Thank you.
Now, sir, there will be at 2:30. Now there will be lunch break, and from 2:30, sharp 2:30, it will start again.
Okay.
Sharp 2:30.
Okay.
Okay, thank you.
Good.
টিনাকে ফোন করি, ওরা কি খেয়ে পড়ে আসছে নাকি?
Sir, how is it going, sir? Morning, I was attending till inaugural session. Then, I just got a notification that I was seeing any problem had happened there, so that's why I got. So, at what time it will come, sir, again? No? 2:30. Just now it is completed, huh? Okay, okay, okay. Okay. I will be present from now, sir.
Dr. Sriram sir. Hello. Dr. Somesh sir. Dr. Sir.
Yes, Dr. Wag. Hello.
Dr. Sriram sir.
Good afternoon. Good afternoon.
Good afternoon. Yes, sir. So can we start, sir?
Yes. We can start. I'm ready.
Welcome you all for the post-lunch session, that is technical session 2, entitled with Role of Microbes in Plant Health Management. In this session, the chairperson is Dr. Sriram S., Principal Scientist IIHR, and co-chairperson is Dr. Someshwar Bhagat, Principal Scientist ICR NRRI. They were also the invited speaker of this technical session. So for the note of co-chair and chair, in this session, we are having 2 invited speakers, sir, invited lectures. In addition to that, 7 oral presentation has been submitted. In that, 5 people already given their presence. Note, sir, the time allotted is 20 minutes each for invited lectures, whereas for oral presentations, 8 minutes each for presentation and 2 minutes each for discussion, sir. Let me have a strict on to the time and finish it in a very nice manner, sir. So let me hand over the session to our chairperson, Dr. Sriram sir, and co-chair, Dr. Someshwar Bhagat sir.
Good afternoon to all. Am I audible for all?
Yes.
Sir, first of all, I thank Dr. Arup Mukherjee sir for inviting me to deliver the invited lecture on trichoderma. And I thank NRRI also for accepting his proposal and accepting my lecture. And I thank Indian Phytopathological Society East Zone also for accepting this proposal. To start with the session, we have 2 invited lecture, including mine too. And as told by the organizers, we have 5 people already given the presentation out of 7. I think 2 more people also will give their presentation. Sir, Arup sir, if you permit, we will start, sir.
Sir, first your formal introduction.
Okay, sir.
Sir, let us introduce yourself, sir.
Okay, sir. Sir, my name is Dr. S. Sriram. I am working here at Indian Institute of Horticultural Research as a principal scientist here. Before that,
formal from ourselves, sir, actually.
Sir?
Let me have a formal introduction from ourselves, sir. From our side, we will have, sir.
Okay, sir. I thought you were asking me.
No problem, sir. No problem. So just I'm very glad to introduce our invited speakers at this moment. The first speaker is Dr. Sriram, Principal Scientist. Dr. Sriram is a very known icon with respect to biocontrol works in pathology, particularly plant pathology. So Dr. Sriram sir has started his professional career at CTCRI and then served at NBR, formerly PDBC. Currently, for the last 8 years, he is serving as a principal scientist at IIHR, Bangalore. He has nearly 104 research articles, 5 book chapters, and many other forms of publications. His work citations are 533, which H index of 11 and I-10 index of 13. His work area is mostly concentrated on trichoderma, which are many fields he has covered, in which he filed 3 patents, of which 1 for invert emulsion formulation of fungal antagonist for biological management of plant diseases was granted already. And his other explorations on trichoderma formulations with carbon resistance and salinity tolerance are remarkable. Apart from trichoderma, he also has his contribution to develop many other biocontrol agents. So the topic which was selected, that is formulations of trichoderma, more than Sriram sir, I feel there won't be a right person for this title. So it is very opportunity for us and fortunate for us to get Sriram sir to have his deliberation on this topic. Let me welcome Sriram sir for deliberating on this topic, formulations of trichoderma.
Thank you very much, sir, for the formal introduction. I'm sharing my presentation, sir. Now.
Video and audio ours. Now it is there.
Sir, is it visible, sir?
Yes,
I
sir.
hope it is visible.
Yes, sir, it is visible, sir. We can, sir.
Someshwar Bhagatji, I request you to remain by 18 minutes so that 20th minute I can complete it.
It's okay, sir. No problem.
Yes, sir. Yes, sir.
My topic is on formulations of trichoderma. Here I will be talking about the general how the formulations are made in general, and 3-4 novel formulations developed in India I will be mentioning, and how the main problem with the formulations are the shelf life. So I will be touching upon how to improve the shelf life in the formulation. So this I will be covering in this 20 minutes talk. Formulation is a very important word. That is the question, that is the important question, what can transform a potential biocontrol agent from a laboratory curiosity to a commercially successful product. The answer lies in this between the how to transform. Everybody, many people, we are working in the biological control, and we are all having the different strains or the isolates which are having very good potential in the lab. But it has to find a end of the tunnel, and it should become a product. What makes it is the formulation technology. The formulation is very, very important. For the formulation to become very successful, we need the very good shelf life of a microbial formulations. When we are competing with the formulations of chemical nature, which are having very long shelf life, if you keep it as it is, it will be there. But the microbial formulation, they are having live organism inside. So the shelf life is very, very, it is a live material, like fruit we buy and keep, or the vegetable we buy and keep, where the life is there. These microbial formulations have the life in them, and we have to see that the shelf life of the microbial formulation is retained for a longer period. There are two types of ferment. Actually, before the shelf life or the formulation, the success lies in the method of production. There are two methods of production for the microbial products. One is the liquid fermentation. We are having a huge fermenters, 5,000 liter or 10,000 liter fermenters, and we produce the trichoderma or any form biocontrol agent in the liquid fermentation. The another method is the solid state fermentation. Different substrates are can be used as the solid substrate, and we can grow the not only trichoderma, any bioagent. But the comparison is the shelf life in the liquid fermentation derived formulation, it is quite shorter because they are not having the desiccation tolerance. But the solid state derived formulation, especially the trichoderma, when we have the solid state fermentation derived product, we are using the airborne conidia, which are produced. They are functionally, they are totally different. They are having hydrophobins present on their surface. That gives the desiccation tolerance. But if you take the liquid fermentation derived formulations, they are highly vulnerable for the desiccations. The spore walls are also not stronger. I will show in next slide. There are reports how they differ. So always it is better to go for the to have a longer shelf life. We need a solid state derived formulation. You can see the difference in the lower one. You can see the whenever there is hydrophobins are not present, the submerged conidia will not have the strength. But in the aerial conidia, you can see the hydrophobins present. When the hydrophobins are present, that gives the desiccation tolerance. The role of hydrophobins is very much important to have the shelf life. That is the major difference between the solid fermentation derived conidia and the liquid fermentation conidia. But having said that, the shelf life is very good with the solid state fermentation, but there are problems in the solid state fermentation. you must be hearing about the Biocon in Bangalore and many pharmaceutical industries. They are having 2000 kg capacity solid state fermentation unit, and different organizations, private pharmaceutical industries, they have the solid state fermentation units. But the problem is they are using the microbes growing on that to convert the substrate into some byproduct. But in biological control, in the production microbial formulation technology, we want the microbe to be harvested. So if we see that their function, that all the conditions, the engineering aspects, harvesting of the converted substrate and all totally different. Our requirement is different. We want to harvest the microbes itself. So there is a lot of need for the engineering aspects to harvest the microbes grown on the solid substrate. So our requirements are different. So it is tedious. Otherwise, if you are using the small scale, different small bottles or the polythene bags, lot of contamination problem is there, which we are avoiding in the liquid fermentation. If you see in the liquid fermentation, you can continuously run, and it is a short period only. For a trichoderma, you need only 96 hours only you needed to produce in the liquid fermentation. And in a compact room, less space, you can continuously run, have your drying room separately, and you can do it. But for the solid state fermentation, you need a contamination free room and a lot of chances of contamination. Here you have everything under the control. Only disadvantage is the shelf life. So it depends on the our investment capacity and other economics. Based on that, we are going for the liquid fermentation or the solid state fermentation. But with the CIB registration, if you see, most of the people are going for the liquid fermentation only. They mix with the talc and do it. In literature, there are different types of formulations possible. One is talc-based formulation. Another is vermiculite wheat bran-based formulation. Then pesta granules. Then kaolin-based. Then alginate pills. Like alginate pills are used in the artificial seed. With the embryo covered with alginate pills, they are using for the artificial seed. Same thing, it is possible with the trichoderma also, kaolin-based. But the problem is, with the central insecticide board, what we have the data for the registration and all, mostly it is on the talc based. For other formulations, if I develop one formulation, for that I have to develop the separate toxicological data, separate for not only for the organism, for the formulation also. So to make the ease of business, the people are going for already one technology is available. I will go with the talc-based formulation. That is why the other formulations are not picking up in the country. And the toxicological data is required separately. Even the organism is same. If you are going for a different formulation, you have to develop a different toxicological data for each formulation. Then carrier also. There are different carriers have been reported in the literature: pyrax, cellotum, talc, gypsum, peat, lignite, wheat bran. But most commonly used is the talc, vermiculite. Though many people are using in the gypsum, I don't recommend the use of gypsum because it affects the pH of the formulation when there is a moisture content. So most preferable is the talc, followed by the vermiculite, where the pH change is not happening much. But all others, literature says, again, we have to remember the business point of view. With the business point of view, if we develop a talc any formulation, the toxicological data should be available for that. Then simple talc formulation it is very simple to make. If you are having a 10-liter biomass, we have to mix 20 kg of the talc, and we can mix carboxymethyl cellulose. It helps us it helps in adhering. It helps in the seed treatment purpose. So this can be used. This is 1:2 ratio is normally used. Some private companies are using 1:2.5 also. But if there is a facility, it is better to use the completely sterilized and cooled talc material. But if you do the survey of the products available in the market, you can see a lot of contamination coming from the substrate as well as bacteria present in that. So the proper choice of the inert material is required. Then another one is the vermiculite brand formulation. This is from literature, but it is not practically followed because of the registration problems. Vermiculite and bran in 1:3 ratio. That is 1 part of wheat bran with 3 part of vermiculite. And here 20 grams of the vermiculite fermenter biomass can be mixed with that. And here also this vermiculite and wheat bran should be sterilized previously. And this is, though literature available, practically it is not followed by the producers. Then another is pesta granules, like our puppets or other material, food material. The same, it has been tried in Europe and other countries. The wheat flour has been tried mixing with the biomass, and the kaolin based also mixed with the wheat flour, or the bentonite mixed with the wheat flour. These are all possible for the preserving different formulations for a longer period, and all this can be used. But on commercial scale, the reach to the farmers level or the commercial production has not been up to the expected level. One sheet is ground, and again it has to be passed like a puppet. Then it is crushed and made into 18 mm mesh. Through that 1 mm mesh you have to send it. Then that can be collected and stored for a longer period. But this has not been commercially explored, but it is having a very good shelf life. Very prolonged shelf life is possible with the pesta granules. Another one is, as I told earlier, the alginate prills. Here the sodium alginate and the wheat flour 25 and 50 grams equally, and the fermenter biomass is 200 ml. The sodium alginate and the food base, that is the wheat flour, at the rate of 50 grams per 250 ml, that has to be dissolved in water. Separately we have to do that. Then both are autoclaved. Then they blended with the biomass. This mixture is dropped. It has to be very skillfully done. Drop by drop, if you have to put into the calcium chloride solution, they form a soft beads. Then the soft beads, we have to remove the water. Then it will become the dried gel in the beginning. Then it becomes prills. This can be stored for 1 or 2 years. This formulation can be used for smaller requirements, not in the commercial application. For larger area, it is not possible. This is only for shorter duration. Then I will be mentioning about 4 different novel formulations developed in India. One is developed by Dr. Anandaraj from Indian Institute of Spices Research. The earlier 20-30 years back, people would have seen the bevistine was the carbendazim was used as a capsule, and there is an injection into the banana plant for application of the bevistine capsules. But here the concept is slightly different. This biocapsule can be used for not only for trichoderma and other PGPRs. This can be used. These biocapsules can be used. These 20 capsules are sufficient for a 20 kg of talc formulation, because here the air dried conidia is used as the material, and it is put into the capsules. This can be given through the irrigation water, and it can be used for multiplying the FIM also. The requirement of the number of capsules per acre will become very less. And this enhance, because this PGPRs are the trichoderma given through this, they help in the nutrition mobilization and use their plant growth yield. And it is very ecologically safe technology. There are no byproducts formed in this, and less amount of inorganic or organic or the inert. For example, in talc formulation, 95% weight goes to the inert material only. In this case, you are not even using the inert material, and whatever the capsule used, that cellulose also is degraded. And the transportation is the biggest advantage in this, because to long distance, when I send, no need to send in kgs, it will become 100 grams or 200 grams only. And the manpower requirement for the storing the longevity of the product and all, it is very good technology, and energy saving also. Next one technology is the invert emulsion formulation. For this only just our scientist while introducing me, he told about the patent granted. This is for this. There are two, one is emulsion, another one is invert emulsion. Emulsion is nothing but when we put the oil in the water, the oil globules are moving. So we call it as a emulsion. The invert emulsion is the water is inside the oil. Water molecule, water droplets are inside. It is all the sides covered by the oil. So what happens if the spore is present, the water suspension carrying the spores in the emulsion, it is outside the oil, so it is not protected by the oil. But if we make it as the invert emulsion, then the water droplets carrying the Trichoderma conidia, it is covered all the sides by the oil. This gives the complete protection and enhanced shelf life. So this invert emulsion formulation of Trichoderma, it gives the population of 10 to the power 8 colony forming units for more than 8 months of shelf life. And here the liquid fermentation derived conidia cannot be used. We used the solid state derived conidia we used. This is suitable for the seed treatment, soil enriching and foliar application. And it is not phytotoxic. Whatever the components used, whether the oils or the stabilizers or for the dispensing emulsifying agents, they were not causing any phytotoxic effect. Then the bioefficacy is tested under the field condition. For this, we generated data in the groundnut for the soil application, and we have later it was given in the AICRP system of the biological control and tested for its efficacy against the soil-borne pathogens. So this is the one. Mainly the problem with the oil formulations. Generally, you can see in one of these photos in the left one, the separation happens. Separation happens, the oil will float in, oil will be separate and water will be separate. And the Trichoderma is hydrophobic in nature. So along with the oil, it will form a clot in the top. But this formulation is uniformly distributed and 98 to 99% stability. The oil and water separation will not be there at all. And stability is very good, and it can be stored for 8 months at room temperature itself. There is no change in the viability, no loss of shelf life. That is the biggest advantage. And it can be, because this is in the invert emulsion formulation, this can be used for the foliar application also. And because the emulsifiers are there, it will not allow, it will not precipitate, float in the spray material. So we have tested in the groundnut, and this is one formulation. But the problem is the registration. We need the toxicological data to be generated for these people. If industry is willing to do it for the toxicological data, because it takes 15 lakh rupees to develop a toxicological data for each item. Then another important formulation I was mentioning is the salinity tolerant Trichoderma. We are talking about Trichoderma. This works very well. Though generally Dr. A.N. Mukhopadhyaya and Dr. Anandaraj, they used to tell one particular Trichoderma strain, if it has to be successful, it should colonize very well and it should work in different places. If you take the US case, United States, that the TH22 one strain was ruling most of the place because the rhizosphere colonization of that particular isolate was very good. So, but in India, we have diverse ecosystem, and in many places we have the salinity problem. So the pathogen or the crop, particular groundnut variety in Gujarat is most suited for the salinity condition. Slowly the pathogens infect Macrophomina fasciolina, infecting that groundnut also has developed the tolerance to the salinity, but not the biocontrol agents. So we have to select the biocontrol agents for the salinity affected area separately. So one such isolate we got is the HAR4B. This was isolated from ISAR. And it protects the root system with very good root growth, and it protects like a buffering zone between the saline affected soil and the plant. It absorbs We have studied the different studies. It induces the glycerol, it induces the tractose in the salinity tolerant culture. Besides, it works as the buffer. And one more mode of action is you can see in the electron microscopy when it is subjected to the saline condition, sensitive culture becomes completely shriveled. These cultures do not lose their turgidity. And when we did the molecular protein work also, we could see that it is inducing different genes which are related to the highly osmolarity glycerol, HOG gene related pathway. And we could confirm that with the 2D gel electrophoresis also, what are the proteins induced in this salinity tolerant trichoderma when it is exposed to the salinity and HOG gene amplification. We confirmed that the role of HOG pathway is important in that. Then fourth formulation is the carbon tolerant. See, if you see any of the package of practices in India, most of them they recommend carbon. So even now it may be banned in another 1 year. It is in the list of getting banned. Not only carbon, the thiophenate, methyl, and other group. The seed treatment is there. So we need some chemical that is tolerant to this benzimidazole group. So we screened and identified this GJ16B. This is tolerant to the carbendazim, even up to 500 ppm. It was able to tolerate it. And here the mode of action is there is a mutation in the beta tubulin gene. This carbendazim, what happens, it binds with the tubulin and during the cell division, beta tubulin, and it stops. So the fungus, the pathogen does not multiply and stops it. That is the mode of action. But wherever there is mutation, the fungicide resistance happens in the plant pathogens. That is how the many fungal plant pathogens develop the fungicide resistance. So if we have a trichoderma which can tolerate benzimidazole group of fungicides, so that will be useful. So that is why this we can see in the insects also. Biological control agents use the parasites or the predators which can degrade or which are having the tolerance to the insecticide, they perform better. So in the same way, this was identified, and the role is we have identified the mutations in the beta tubulin. This changes the structure so that the binding carbendazim binding protein present, that is not happening with carbendazim binding is not happening with this particular beta tubulin. So I will talk about a few points about the interventions that we can use to enhance the shelf life. Generally, in our production media, if we are increasing that carbon content and reducing the nitrogen, that CN ratio, that plays important role. Another one is we can add bioagent specific nutrition. For example, trichoderma, it produces chitinase. We know it very well, it produces chitinase. So if we add some chitin, colloidal chitin in the production medium and chitin in the formulation, it will use that as the food substrate. And it will reduce the contaminants in the formulation as well as the growing also is good. Third intervention can be the osmoticants or the humectants. Just now we were seeing in the salinity tolerant trichoderma, there is a tractose production, there is a glycerol. Glycerol helps it. So why don't we add glycerol in the production medium and check it? So we did 3 or 6% glycerol addition that protects the shelf life of the formulation. And tractose is nothing but disaccharide produced in the hyphal cell tip when it is exposed to high temperature or under stress. So artificially we can create this situation when the fermentation is going on after 96 hours when we are about to harvest, just increase the temperature to 35 degrees centigrade for 45 minutes. That heat shock increases the tractose. So the biomass becomes tolerant to the desiccation. So once you make it formulation, that tolerance is carried out. Then another important point is the moisture content and water activity. More than the moisture content, it is the water activity. Water activity is nothing but the in a colloquial term, it is the relative humidity inside the formulation. People are more worried about moisture content. Moisture content is not important. Water activity is important. I will tell about it later. Then we can use some methods to wash the spores with the propagules, but it is a difficult, tedious process in the large scales. Then we can change the drying methods. Some of this we have been successful. I will mention about a few points. See, I told about the chitin. If we are adding colloidal chitin in the production medium, that is 0.2% is sufficient. It will enhance the shelf life. Normally, it is only 4 months. We will get the shelf life. But if you are adding the colloidal chitin, you can have up to the 6 months. 2 months we are extending it, shelf life. This is in the in vitro, in the normal scale production I am mentioning. The next intervention I told is the osmoticants or the humectants. That you can use the polyethylene glycol or you can use the glycerol. So the hypal production is restricted and more spores are formed. CFUs will be increased. If you add that in the production medium, either 3% or 6% is sufficient. Don't add more, because 9% or 12%, though it increases the CFU count, but the viability comes down after the 8 months. You can see in the graph. Once you add the glycerol, automatically the water activity comes down. If you see the viability log, that CFUs, when we have the 3%, it comes up to the control. You can see by 6 months it is less than 2 into 10 to the power 6. That is the minimum required is 2 into 10 to the power 6. It comes down. But if you are using the 9% or the 3%, it comes up by after the 8 months it comes down. 6% you can see it is continuously going. So either 3 to 6%, that is maybe 4 or 5 or 6%. If we are using especially 6%, you will get very good extended shelf life with the trichoderma formulation in the liquid formulation, fermentation condition. Then I mentioned about, you can see the moisture content. The moisture content is not important, but the water activity is very important. When we are adding the glycerol, you can see the moisture content changes happening. So 0.6, if you see the moisture content is maintained at 15% almost equally till when after the 6 months it comes little down. But if we are having the low water activity, the moisture content is less. To maintain uniform moisture content, 6% glycerol addition will help. Then I mentioned about the trichlose induction with the heat shock. We tried with 40 degrees centigrade as well as the 35 degrees centigrade. 40 degrees centigrade, though initial population is high, but the viability comes down. Again, 60 minutes, it is not good for the population was very less. When 35 degree, 45 minutes, shelf life went up to 8 months. 35 degree to 90 months, though population is good, but the viability comes down. So the optimum temperature was 35 degrees centigrade with 45 minutes exposure was sufficient. What will happen if all these interventions are given together? I produce the formulation with colloidal chitin and chitin in the medium formulation, and 6% I am adding the glycerol in the production medium, and all the three inventions, my liquid fermentation derived formulation can extend its shelf life up to 17 months. Compared to all this, you can see that 17 months getting the CIB says minimum 8 months they ask. Ideal is the 18 months. Ideal is 18 months that we can get with the solid state fermentation. We can get 17 months easily when we do these interventions during the production conditions. This is very much, these are all very simple methods. Adding glycerol, giving heat shock, adding colloidal chitin is a not big thing for any trichoderma producing manufacturer. He can do and get the shelf life extended. So, but if you see comparatively the solid state fermentation, up to 14-15 months, no need to do any intervention. It will have its own shelf life. And there the water activity, only one thing we should remember, there the moisture content and water activity should be very, very less. It should not be more. So critical factors, if you compare the liquid fermentation and solid fermentation, here the water activity should be more than 0.6. It should be 0.2 to 0.3. And here the packing material exchanges, it should allow the exchange of air. Polypropylene is do not retain the water activity. But so you can use the polythene conditions. Here you can use the both. Any type you can use, no problem. And water content should not, moisture content should not come down 8. Though CIBRC earlier they were mentioning about 8%, now we have requested not to include that stress on that. That is suitable for the solid state fermentation only. And the liquid fermentation based formulation is not tolerant to the desiccation. So whatever I told, I am summing up as for extending the shelf life, we can use these methods. This is in summing up how the interventions that I have mentioned already. I think I have taken more than 20 minutes. This is summing up the interventions for the enhancing the shelf life of the liquid fermentation derived formulations. Thank you.
Sir, can we take some queries and from anybody?
Yes, sir. Yes, sir. Yes, sir. We will take. Sir.
Now it's open for question or query.
Sir, I have one query, if you kindly.
Yes, sir. Yes, sir. Yes, sir. Please, sir.
Sir, problem is that in India, the Central Insecticide Board, they get confused about that biological fertilizers or biopesticides and chemical pesticides. Okay, so it really becomes difficult because, for example, in rice, you know that trichoderma promotion, usually it was reported earlier that it does not work. But we have got very good endophytic trichoderma, which is working excellent, even for drought tolerance also. And we are not able to distribute among the farmers because they think that unless you have toxicity data, you cannot distribute as biopesticides. But if we have tested in the farmers field, which is enhancing the yield from starting from 15% to 24%. Okay, so sir, what should we do? Can we distribute them as biostimulant?
Sir, first thing, to test their bioefficacy distributing among the farmers, as long as you are not selling it as a commercial unit, you are demonstrating, you are showing the efficacy, you can do it without doing any profit. You are not doing any business. You are not doing asking the license to sell it. So that you can do in the beginning. You can do it as the demonstration free supply as a material, you can do it. Then for registration purpose, they are asking the toxicological data. You can say that in Indian Institute of Horticulture Research earlier, Dr. Chaudappa made a seed pro. It is also having one trichoderma and Bacillus subtilis. So it is promoter registered as the promoter only, not as the plant growth promoter only. But when the label claim comes that it is controlling such and such a disease, then it becomes disease control agent. That time again CIBRC needs the toxicological data. If you need label claim, then toxicological data is required, sir. If you don't claim the disease control, then you can promote it as the plant growth promoter.
That is our point actually. We want to use that as biostimulant.
Yes, sir. Biostimulant you can use, sir. For biostimulants, no registration is required, sir. Biostimulants, no registration is required.
And we commercially sell also on no profit. No
yes, sir. Sir, when it comes to biostimulant, if you are telling the plant growth promotion and all, it can come under either the fertilizer control or the state level only. You can take the license, they can do it, sir. It is not needing the registration at the CIBRC level, sir.
Thank you, sir. Thank you for the information.
Sir, one of participants has asked one question in chat box, sir. What are the quality control measures of trichoderma formulations, sir? Please, can you...
Actually, for the quality control, there are first important, they say, is the contamination, sir. First, CFU we have to see, we have to use the trichoderma specific medium. In that 2 into 10 to the power 6 population should be there. Minimum it should be there. When the serial dilution is done, and we have to check the 10 to the power 6 dilution at minimum more than 2 colonies should be there. First criteria. Second criteria, we have to check the bacterial population 10 to the power 4. It should be, when we make the 10 to the power 4 dilution of the formulation, there should not be any bacterial contamination, especially the human pathogenic bacteria should not be present. That means 10 to the power 4 dilution, if I put the plating, there should not be bacterial contamination in that. Third, again, it comes the moisture content. It should not be extreme moisture content. It should not be dry powder. It should not be completely. Some formulation, by touching, you can see that moisture content is more than 25 to 30%. That is not recommended. So moisture content recommended is there 8%, but 8 to 10% is fine, sir. So these 4 CFU, bacterial contamination, moisture content, these 3 are the quality parameters required, sir.
Mukhar sir.
Yes.
May I put one question?
Yes, sir.
Yes. May I ask one question?
Yes, sir. Yes, sir.
Yes, sir. Go on.
Okay. Sriram sir, in different forums, I was also continuing to ask this question also regarding the shelf life of biocontrol agent formulations or trichoderma based formulations. Is few 6 months to 1 year or that, but in chemical formulations, their shelf life is more expiry date and manufacturing. But this is the problem for that. The pesticide dealers are hesitating to keep the biocontrol agent based formulations, since due to expiry problem they are not purchasing and delivering to the community. So how the problem will be solved? Thank you.
Actually, for promoting this use of biocontrol agents, mainly the government is using the subsidy mechanism. But the problem is, in the same subsidy mechanism, the poor quality materials is supplied, the farmers are not getting the confidence. So while creating the awareness, the good quality formulation should be given to the end users, then only they will develop the confidence. Once they develop confidence, like earlier the biofertilizers people were not using, then later people are asking for the rhizobium in the pulse. So there also the shelf life is less, but building the confidence in the farmers is very important. Then Dr. Mukhopadhyay Sahab, he suggested one recommendation that all the seed companies are selling the seeds. They are using the seed treatment with the chemicals. So he wanted to include in the seed policy also this bioagents treatment may be recommended in the while selling, so that the viability, so that there is a pickup in the uptake of this bioformulation, as well as the more usage, people will get confidence. Somehow introducing another organism into the seed material, it has not been taken up so far, but he is making continuous effort to include A.N. Mukhopadhyay Sahab. He has written to the government of India to include in the seed bill, but it is yet to be finalized, sir. It is only confidence build up in the farmers is required, so that they, once they ask, the demand comes, automatically the product will be available in the shelves.
Sir, is there any research to increase the shelf life? What the existing is here, 6 to 1 year, 6 months to 1 year. Is there any research that can be possible to increase the shelf life of the biocontrol based formulations?
Just now I presented same thing, sir. I have presented the same thing. In the liquid fermentation, the increase the shelf life, there are 3-4 interventions. Just now I presented, sir. I will share my information again later, sir.
Okay, sir. Next, I think we are running shortage of time now.
Yeah.
So, thank you, sir. Thank you very much.
Thank you very much, sir. Thank you, sir.
Thank you very much.
Sir, thank you very much, sir. You have given so much very exhaustive and informative lecture. And because topic is very good and everyone wants to put question, but time is limitation. So we have to sum up here. And thank you very much, sir.
Thank you, sir. Thank you, sir. Thank you. Sorry, sir. I have taken more time than given.
Sir, actually, I was about to stop, but you were giving so much informative lecture, so I could not stop you. So I sorry to all members.
Okay.
Shall I request now Dr. Someshwar Bhagat to present the invited lecture and formal introduction from the organizers, sir,
Yeah.
about Someshwar Bhagat.
Thank you, sir, for your impressive talk. And I am sure, sir, everyone has taken some points from your presentation, sir. So next we move on to the next invited speaker, Dr. Someshwar Bhagat, who is a principal scientist. Actually, he started his scientific career at ICR Central Highlands Agriculture Research Institute, Port Blair, as a scientist. Then he served at NRCIPM as a senior scientist for 6 years. Since then, till date, he is excelling in his scientific caliber at ICR NRRI. Dr. Bhagat, to his credit, has 71 research papers, 5 review papers, and many other type of publications. His work citations are 16 with H index of 13 and I-10 index of 20. He bagged many awards and recognitions, in which to quote Jawaharlal Nehru Outstanding Award, M.K. Patel Young Scientist Award by IPS, and Sir E.J. Butler Memorial Award by Indian Mycological Society, Kolkata. His explorations in the field of biocontrol of plant diseases are very commendable. So now I request Someshwar Bhagat sir to have his deliberation.
Good afternoon to all. Respected Chairman sir and dignified member of the society, I am going to present a lecture. That topic is plant disease management by Trichoderma species. You know, Trichoderma is very important organism nowadays, and everyone talks about Trichoderma. So, what is the mechanism of Trichoderma? So, just in brief, we can say that one antibiosis, it can produce by volatile and non-volatile compound, and then second one is mycoparasitic. It comes through physical contact, followed by coiling of hypha and penetration and production of hausteria and lysis, and then different types of coiling, again by producing enzymes, hydrolytic enzymes, and then competition for space and nutrient, and then tolerance to biotic and abiotic stress, and then solubilization of plant nutrients, and then induced resistance. So here the just diagrammatic showing of the action of Trichoderma, that is shown here, that different type of Trichoderma mechanism of action. Like here we can see that better abiotic stress tolerance and better induction of resistance, like that. And here again, the Trichoderma can produce some iron form that is insoluble in form to soluble form, which can plant take up. Then again here some information that Trichoderma, how it can act in the rhizosphere region. It produces enzymes, peptide walls, oligosaccharides, auxins, cytokinins, and then later on it will cascade, MAPK cascade, and then priming of and defense and increase photosynthesis and carbohydrate metabolism and abiotic stress response. Here again we can see that plant Trichoderma interaction involves the recombination molecules of MPS, and effector molecules bind to PRS and intracellular receptors, and thereby initiate the MTI and ETI immunity to plant respectively. This interaction also leads to the production of ROS, which serve as signaling molecules and initiate a defensive response in plants by synthesis of antifungal molecules like phytoalexins and volatile organic compounds. Here we can see that trichoderma, how can it coil the pathogen hyphae, and then it lies through the secretion of hydrolytic enzymes, and later on finally it can kill the pathogen hyphae. Here again we can see the microparasiting of trichoderma to the plant pathogens. We can see here the healthy hypha how it is, and then the trichoderma just coiling here and giving the depressed hypha, and it can also produce papilla-like structure which can press the pathogenic hyphae, and then it can lead to a depressed hyphae. And they can also produce cell wall degrading enzymes. So it has very multifunction action to the plant root system, by which they can induce resistance to the plant system. Here some commonly produced compounds which is used in plant protection. So here we can say here IA, like growth development of plant and their root system, and then GA3, growth promotion by degradation of growth, repressing chelasia and proteins and reduction in ethylene level, and then alteration in transpiration, regulation of stomatal aperture via induction of ABA receptor, and improved tolerance to biotic as well as abiotic stress by regulation level of SA and J as well as their signaling pathway. And then jasminic acid is produced by Trichoderma spirillum, and then salicylic acid produced by Trichoderma atroviride. Similarly, some other compounds like hydrolytic enzymes, like cerolytic enzymes, produced by Trichoderma varians, and then exo-1,4-glucanase produced by T. viridis, T. hergianum, T. ricci, T. coningi, and then endo-1,4-glucanase that is produced by T. viridis, longibrachiatum, pseudoconingi, and T. ricci, and then endochitinase that is produced by T. hergianum, T. coningi, like that, many other species, and then exochitinase same produced by the above mentioned this species, Trichoderma species. And then other group of compounds are PL, then CH, production of phytoalexin and produced by Trichoderma species, then glucan and chitin synthase produced by Trichoderma species, and MAPK produced by Trichoderma species, and ETR1 and CTR1 produced by Trichoderma species. Like that, LOX1 and PL1 produced by Trichoderma species, and these all have some functions which either promote growth as well as use in plant protection properties. Some compounds they also produce like pyrones, coningins, trichodermides, viridins, hergianopyridone, hergionic acid, here glycotoxin, glyovarin, like many other volatile compounds, and hydrophobins. Here some cultural characteristics of Trichoderma you can see in the photograph. Many species have several types of their cultural characteristics. Here some microscopic view of some species of Trichoderma. This is same. This is viridi, and this is Trichoderma hamatum. And here some spores of T. hergianum, Erinaceum, T. spirillum, brevicompactum. These species so far we have reported first time from Andaman Nicobar Island, and earlier it was not reported from that island. And so far my career, we have worked several states of India, and this much isolate we have isolated from different parts of India and characterized and used in management of different diseases like vegetables, pulses, rice, like other crop. This is just a picture of antagonistic potential, how we can do this is dual culture technique. Here you can see here pathogen, here trichoderma, how it can overgrow. Like here Rhizoctona solani, here Macrophomina fagellina with different isolates. Here same different pathogens with different isolates we can see. This is a hyphal infection, how it happens. This is just how we can do in a laboratory to get the clear-cut picture of hyphal interaction of trichoderma with plant pathogens. So this is just coiling of trichoderma hyphae coil that hyphae of pathogenic fungus and then secretes certain enzyme, hydrolytic enzyme, and later on it can lyse the hyphae of pathogen. This is another form of antagonistic potential of trichoderma by producing volatile and non-volatile compounds. And this is biopriming of seed we do. Earlier speaker also told that biopriming is seed treatment is very, very important, and this is just used in all crop, and we have used in case of chili, tomato, brinjal, chickpea, mung bean, French bean, like that several other crops. This is the field trial result of where we can see that the infected roots are very less, and then the healthy plant, the more root volume, and very healthy plant. And these are some example where trichoderma can be used for plant disease management. That is bean, we can see that seedling rot, and then cabbage, in the alternaria blight, cauliflower, like that brinjal, French bean, tomato. This is just number, but you know that in India and abroad, trichoderma has been most extensively used biocontrol agent, which is used in management of plant disease of all categories like cereal, pulses, vegetables, and even some forest plants also. Here this is in case of pulses crop, chickpea, lentil, mung bean, pigeon pea with different disease. It has been used for management of these mentioned diseases. Likewise, some cereal and sugarcane crop also, it has been used for management of many diseases like foot and root rot of barley, then charcoal rot, banded leaf seed blight, maize, rice seed blight, burnt buccanay disease. Here again not mentioned, but blast also it can be managed. Brown spot is there, and then wheat, low smut, root rot, and then sugarcane, root rot, red rot, wilt. These also have been managed by trichoderma species. Here some our work with some DVT project in northeastern area. We have work, and we have successfully managed the wilt of pea, and then root rot of cabbage, and then Symposium rot of mustard. And this is just biopriming we have done, and just increase the germination, seedling vigor, and biomass of seedling, and reduce disease incidence of polar rot of cabbage and cauliflower, wilt of pea, and stem rot of mustard. And this is we have isolated some isolates from the Rajasthan region, and it has been successfully used against the white rot of chickpea. Uh, in conclusion, yeah, I can say that like other organic trichoderma are also sensitive to extreme environment. A better understanding of the influence of environment factors on pathogen trichoderma interaction can help improving their efficacy, and exploration of naturally occurring trichoderma species from various agroecological zones and geographical regions of India tolerant to adverse climatic conditions like saline, high temperature, desiccation, etc., or development of a strain with desirable biocontrol trait with help of biotechnological method will be appropriate solution to tackle the adverse environmental condition due to climate change. So I conclude this my lecture, sir, here.
Thank you, Someshwar Bhagat sir. Thank you for sharing your experience in biological control of different diseases in different crops. I request participants to ask few questions. I request participants to ask few questions.
Hello, sir. Good afternoon. Am I audible?
Yes. Yes.
Gopal
I have a
Chandra.
question that since now we have been seeing that trichoderma can be used as a biocontrol agent and it is a fungus. Is there any notice or is there any possibility that this fungus could be turned into a pathogen, plant pathogen?
Actually, plant pathogen, it has been not reported, but it can act as pathogen in case of mushroom cultivation. So far, the study, we have not observed or I have not seen any report that it can act as pathogen of plant.
Shall I respond to that? Yes, I will supplement to that information by Bhagat Saab. The trichoderma is basically a saprophyte. It uses the organic matter and grows. Even in the rhizosphere, it only colonizes. It does not penetrate the root cortex and goes inside. The signal mechanism is such a way that the plant and trichoderma, when they interact, the host pathogen interaction signals don't act in that. It is only a rhizosphere colonizer and it is a saprophyte. It has evolved as a best saprophyte. It can colonize any food material and survive long. So it is not so far it has not acquired the ability to, or there was no need for it to infect plant to derive nutrition.
Yes.
That is the advantage. That is why it has not turned as parasite or the pathogen, because it has a very good capacity to colonize whatever food is available in the soil. Organic matter, it can utilize it.
Sir, I have a query.
And one more, this mushroom, I will mention that mushroom also, whatever the bioagents we are using, they are not infecting the mushroom. The mushroom as a pathogen, the trichoderma as a pathogen of mushroom, they belong to different species. They are called aggressivum, and there are two subgroups. Probably what contamination in the substrate because of this normal trichoderma may be there, but infecting mushroom by trichoderma as a pathogen, it is totally a different species that we are not using for biological control purpose. That is totally different species, trichoderma aggressivum and its subgroups we are not using in biological control.
Sir, one thing, now scientists are also applying trichoderma as a spray solution for foliar diseases.
Sir, for the, shall I reply, Someshwar sir?
Yes, sir. Yes, sir.
If you have isolated the trichoderma from phylloplane, if you are able, if that is having the survival on the phyllosphere, that it can be used. But unless it is having the phyllosphere survival ability, it is not worth using it for the foliar for targeting the foliar pathogens. That is my stand. Unless it is having the endophytic nature and it is having very good colonizing capacity on the phylloplane. That is, after spraying, it can stay for 7-8 days, and it is having the ability to use the excretions from the leaf surface. If it is able to survive like other bacteria, then it is fine. Otherwise, it won't give better result. That is my stand.
Sir, it is better to apply trichoderma as soil application as a seed treatment.
Yes.
But
Yes.
some of the scientists, they are presenting papers, spray the trichoderma on the foliage at the rate of 4 to 5 gram per liter. That's why we are confusing, sir.
No, my stand I told, unless it is having the endophytic ability, phyllosphere colonizing ability, we should not recommend.
That means it is totally confined to the persons who have isolated that one.
Sir, many a time we talk of rhizosphere competence of the trichoderma strains. So, sir, can you tell us something about using indigenous strains in identification in all those aspects?
Shall I reply, Someshwarji?
Yes, sir, yes, sir.
No, actually, the trichoderma is the very good root colonizer. Maybe for other bioagents, the rhizosphere, that native population. See, for example, if you take 1 gram of soil or any soil sample you take, what is culturable microbe is only 4% of the total microbes are culturable. Remaining are all not culturable. So what we are introducing is one among the thousands or 10,000s of species present in that. So the trichoderma, what we are using should be a very good colonizer. It should compete with all the microbes. 4% culturable I am speaking, there are other 96% of microbes, multiple microbes, it has to compete. So if it is performing in one place, similar soil condition, if it is there in the same crop in different places, it should be having the colonizing capacity. That is why TH22 isolate of the US has covered a huge area. Yes, the local strains are better because they are used to that, but same soil type, same crop, it should be able to perform in some other place also. Same soil type, same crop, same exudate. If it is there, it will be performing in other places also.
Sir, from soil dilution, we are using 10 to the 6 or 10 to the 8 for isolation of this trichoderma.
For isolation, it is at the lower 10 to the power 6 is sufficient, even 10 to the power 4. That trichoderma special medium, when you are using, you will get that individual colonies. For quality check, minimum 10 to the power 6.
So normally you can use 10 to the 6 to 20 to the 8.
Yes, yes. 8 you may not get isolation. For isolation, you depend on 5 to 6 for isolation purpose.
Okay, sir. One thing, regarding the fermenter, what is the sizable fermenter and what is the cost now available, sir, in the market?
No, for 10,000 liter, you can get in with a fabricator is available. If you give the design, some fabricators are ready to make by 4 to 5 lakh rupees also, 10 liter for laboratory purpose. There are some fabricators, even the 1,000 liter fermenter, they can make with 40, 50,000. 1,000 liter also is possible. If you are going with the brand name, then the problem is there. Then there are, if you want to adjust the pH, you want to use the automatic adjustment of the pH, you want to have lot of sensors, then the cost increases. If you are not interested in many of, say, for example, trichoderma, what are the microbes you are going to grow is very important. If you are restricting only to trichoderma, then your requirements may be less. But if you are going to have a wide range of microbes, Pseudomonas, then Bacillus and others also, then you may need a different sensors. Then the cost will go with the types of sensors you are using. Maybe the pH adjustment, aeration adjustment, temperature, many sensors are now available. So it depends on the sensors that you are asking for that. Otherwise, you in local fabricators, if you give very good design with a small boiler, with fermentation only with agitator, there are many innovative fabricators are there. They can make it a very cheap rate.
Thank you, sir.
If you want, I can share some of the addresses.
Sir, kindly give me your mobile number, sir, please.
I will give 944
Sir,
955
sir.
1070.
Okay, sir. Sir, I am Dr. Onsing speaking from OUAT. I have mailed to you, sir. Last
Yes,
time
I will respond. I will respond to it, sir. I was busy with another work. I will respond to that.
Thank you, sir.
Thank you.
Sir, it is the time to move on to our oral presentation, sir.
Okay, sir. May I request the first presenter?
A small note, sir. Actually, among these oral presenters, we have to select 2 presenters as a for giving, sir.
Okay, sir. Okay, sir.
Sir, 2 best presenter you just select and send the
Yes,
next
sir.
email.
We will do, sir. We will do.
Hello, sir. Is it
visible?
visible?
It is visible. Just you go for slide sharing.
Okay, sir.
Slide.
Good afternoon. Good afternoon, my respected teachers. Today I'm going to say in vitro.
Excuse me, you can put it in the slide. This one, slide show.
Just put it in slideshow and start from beginning. F5. Use F5.
Is it visible, sir?
Yeah, it is visible, but you are not selected slideshow. Please select slideshow.
Okay, sir.
Yeah, now you can go on.
Okay, sir. Today I'm going to say about my topic, in vitro antagonistic effect of different biocontrol agents against Sclerotium ralphi causing color rot of disease of lentil. And first of all, introduction, is it soil-borne pathogen that commonly occurs in the tropics, subtropics, and other warm temperate regions of the world. According to Agrios, it is wide variety of plants, and the most important fungal diseases of lentil, Alternaria blight, Botrytis grey mold, Anthracnose, Ascocata blight, Fusarium wilt, and collar rot. And the most important collar rot, the most destructive disease, which is caused by Sclerotium ralphi, is most very lentil growing regions in warm areas with high soil moisture 30 to 40% and high temperature 25 degrees centigrade at the seedling stages. And that the main objectives is selection of the most virulent strains of Sclerotium ralphi, and second objective to identify the most potential native biocontrol agent against Sclerotium ralphi forming color rot of lentil, and collection and isolation of Sclerotium ralphi. Color rot infected lentil plants which are collected from different regions of West Bengal, SRC 1, 2, 6 and 7 and during Rabi season 2018-19. Here I select before most virulent strains of Sclerotium ralphi which already been published in my paper, and then the collected samples are isolated in my Department of Plant Pathology, DCKV. And then the places collection from a region and isolated from my department and from farmers field, KBK, Murshidabad and Shakda. And 4 biocontrol agents, Trichoderma harzianum, Trichoderma viridi, Bacillus subtilis, and Pseudomonas fluorescens were evaluated against Sclerotium ralphi from dual culture technique in vitro condition. But main aim of this study is disrupt mycelial growth and kill the organism with the help of native biocontrol agent in vitro condition. And dual culture plates assay, a 5 mm disc inoculant disc Sclerotium ralphi was placed near the edges of petris and containing potodextro agar medium. And plates were incubated at 25 degrees centigrade, colony diameter of the pathogen and the test organism were recorded. 3 replication of each treatments were maintained. And then growth of Sclerotium ralphi in vitro. Colonies of test organism and pathogen meet each other. Pathogen overgrew the colony of test organism, and colonies of test organism and pathogen or test fungus was observed, and colonies of pathogen and test fungi intermixed. And the result, It was observed that maximum inhibition recorded in DS1, Bacillus subtilis 1, 47.88%, followed by Trichoderma harzianum and 45.12%, isolate SRC1. And then isolate SRC2 maximum percent inhibition was recorded in BS2, 46.83%, and followed by TV2, 37.38%. Whereas in case of isolates, SRC6 maximum percent inhibition was recorded in BS5, 47.21%, followed by TV3, 34.45%. While isolate SRC7 maximum inhibited percent was observed in BS6, 46.12%, followed by Trichoderma harzianum 4, 33.94%. That figures will be so different days after incubation, 2 days, 3 days, and 4 days. And showing this Petri plate method that will be regarding Bacillus subtilis more compared to another biocontrol agents, Trichoderma harzianum and Trichoderma viridi and Pseudomonas fluorescens. Most effective one is Bacillus subtilis. All the native biocontrol agent, Bacillus subtilis so the most effective, and so the biocontrol agent can be used a alternative way management of for color of lentil. It could be replaceable the most effective biocontrol agent, that is Bacillus subtilis, and it could be replaceable by the most common fungicide Mancozeb 75% WP, which already been recommended for the farmers field. And this indicate above all native biocontrol agent have dynamic effect and important role in biologically based technique for the management of soil-borne disease in lentil and improve the quality of seal, plant growth, and yield. That's all. Thank you, sir.
Any questions from participants? Any questions from any participants? Mandal sir, Amitva Mandal, I have one question. Hello, are you able to hear me? Dr. Mandal, can you hear me? Mandal, can you hear me? Hello, am I audible?
Yeah, sir, you are audible, sir. You are audible.
Mandal, you just now presented. This is part of your thesis work? I think he is not available. I think Mandal.
Amitav Mandal. Amitav Mandal, just please respond.
Yes, sir.
Yeah.
Part of your thesis work?
Yes, sir. It's
So
my
have you studied
MSc
further?
thesis work.
Have you studied further about its performance in the pot culture or field studies?
Yes, sir.
After the in vitro, so there also Bacillus subtilis was
Sir,
giving better.
Actually, I have selected 4 native isolate soil samples correct, and then isolated from dilution technique method, and then I have put dual culture technique. Then I have
No, in field also, did you do field experiment?
No, sir, I already were pot culture.
Pot culture. In pot culture also you got the better results. Same.
Bacillus subtilis is the most effective one.
Okay, what may be the probable mode of action for Bacillus subtilis?
Sir, it is a so it is the actually sir mycoparasite and numbers of plant pathogens actually disrupt mycelium growth, and I shown that pot in every pot that I have collected 3 lentil varieties, popular lentil varieties, Moitri, BM6 and Hub 57, and then I saw Moitri is very effective for the control the color rot of lentil.
Any other participants having any queries? Any other queries?
Sir, which media actually they have used for isolation of this bacillus?
Please respond.
Nutrient agar media.
Normal nutrient agar you have used or any specific medium you have used?
Yes, and normal nutrient agar media.
Actually, nowadays Bacillus subtilis is also coming as a contaminants in many of the medium. How can you differentiate whether it is a contaminants or normal Bacillus you have isolated?
Sir, actually, Bacillus subtilis does contaminated in
maximum PD medium if you are using in the laboratory it is also coming.
So nutrient agar media I've used in this experiment.
You have to confirm the identity of the biocontrol agent used.
Yes, sir.
You have to confirm the identity of the biocontrol agent used. Confirm the identity.
Sir, biocontrol Trichoderma harzianum I have isolated.
No, for the Bacillus subtilis, what you are mentioning as the efficient one, you need to confirm the identity.
Is
not now, later.
Sir, it is a marketed sample.
No, marketed samples not. You might have isolated from the soil or from anywhere. Then you go for different biochemical procedures are there. Procedure for production. There are different type of biochemical characters are there. At least may confirm 2-3 characters for most potential bacillus.
Sir,
Sir.
We call the next presenter.
Yes, sir.
Presenter is Sabyasachi Parida.
Yes, good afternoon, everyone. I am Dr. Deepak Kulbe from OUT. I will be presenting the paper Sabyasachi and co-workers.
Okay.
And I am going to share my screen with you. Is it in presentation mode, sir?
Yeah, you can.
Okay. Good afternoon, everyone. I am going to present the paper entitled Bioprospecting Plant Growth Promoting Rhizobacteria from Rice Ecosystem for Antagonistic and Plant Growth Promotion Traits. As per the topic of the symposium, which is on sustainable plant protection strategies, and in view of the United Nations Sustainable Goal Development, 3 of the pertinent goals fall into the category of agriculture, through which we can add to those goals. And among those plant protection strategies, use of plant growth promoting rhizobacteria is one of the most important strategies. Plant growth promoting bacteria are the soil inhabiting bacteria, as we all know that they live in close vicinity of the crop root and have the synergistic and friendly effect with the plant by various mechanisms which are well understood as direct mechanisms and indirect mechanisms. Direct mechanisms are increase of plant uptake of the nutrients which they secrete in the environment or soil environment, nitrogen fixation, plant growth promotion, root colonization, etc. And there are certain other indirect mechanisms also, like other things which are happening in an intricate way and in a biochemical way. This is the gist of those mechanisms. With this idea in mind, the study was planned with objectives, isolation and characterization of antagonistic PGPR species from rice ecosystem, and then their assessment for antagonistic potential And in terms of plant disease management in case of rice, which we have taken 3 major diseases. Much of the statistical account of the importance of this crop has been explained by previous speakers in earlier sessions also. So rice is a very important crop, and it's a staple crop worldwide. So we need to sustain the production, and a lot of pesticides, I mean a major share of pesticides is being used in this very crop, which is rice. And we all are aware of the hazardous effects of pesticides and chemical plant protection technology. For that matter, we are undergoing this symposium. And this is again a statistical information that on an average, farmers lose about 37% of their rice yield because of the pest. And among those, diseases like blast, sheath blight, brown spot, sheath rot, etc. account for a good share of their loss. So diseases need to be managed for hunger alleviation and quality and production and all those parameters to be taken care of. So in this study, the methodology followed is like this. The work started with collection of rhizosphere soil samples from rice fields, and then isolation of antagonistic microorganisms on selective media. Different media were used. Then cultured those isolated microorganisms were maintained and cultured, and the pathogens, major pathogens, 3 pathogens, Bipolaris oryzae, Magna porthe oryzae, and Rhizoctonia solanae, which are the major pathogens of rice in all over the world, India, and particularly in our area, which is coastal areas, regions of Odisha. So these 3 pathogens and cultures were maintained following routine laboratory techniques. Then in vitro evaluation was done, and in vivo evaluation was done under field conditions. These are the experimental details. Rice variety Swarna, which is susceptible to all these 3 pathogens, and it's a major variety used by a large chunk of farmers in this region, and it's a good high yielding variety with the 9 treatments of chemicals, positive control, and selected PGPR. These are the cultures, and these are the selected rhizobacterial strains which were taken for the study. These strains were isolated from approximately 25 strains were isolates were isolated following serial dilution method from soil, and their activity against these 3 pathogens was tested, and in this way the growth was rated on preliminary screening. And based on their cumulative pathogen inhibition, 3 strains were taken, and they were named as strain A, B, C. In later on, I will be using strain A, B, C. Then in dual culture, those 3 strains were plated with 3 of the pathogens, and these are the growth inhibition status of all those 3 strains. The radial growth of Bipolaris oryzae, Magna porthe oryzae, the brown spot pathogen, blast pathogen, and sheath blight pathogen reduced significantly, indicating approximately 75% inhibition over control in case of Bipolaris, brown spot, 57% in case of leaf blast or blast pathogen, and 38% in case of sheath blight pathogen. Then coming to the in vitro screening again, which was done for growth promotion by roll towel method. The seeds were treated with 3 isolates. Germination increased up to 93%, whereas it was 81% in case of control. Shoot length, root length also increased as compared to control material. Then vigor index, likewise vigor index also increased with this PGPR treatment in case of rice varieties Swarna. Those 3 strains were also tested for phosphate solubilization using appropriate media and inhibition zone with the help of mathematical formula, and solubilization efficiency was 90 up to 94%, 66%, 37% in all the 3 isolates. Similarly, the solubilization index was also fairly good. Then in vivo screening of those 3 isolates was done in field condition, and for comparison, Pseudomonas trichoderma bacillus were taken as biochemical standards, biopesticide standards, and 2 chemicals were also taken as chemical control for positive check. One untreated control was taken. Germination was calculated by counting the planted seeds in nursery, and germination increased to the extent of 53% as compared to the control, and where it was 61% in control, whereas it was up to 94% in with seed biopriming with isolate A. The seedlings were planted, seeds were sown after treatment, transplanting was done, and the crop was also sprayed with, foliar application was also done twice at regular intervals starting from 35 to 40 days with first appearance of this spot in the crop, and then repeating it after 15 days. In between, at the final stage after second spray, the disease incidence, final disease incidence and disease index was also calculated following standard evaluation methods for rice disease evaluation assessment. The incidence of brown spot was up to 21% with isolate A, 24%, 28 to the extent of, below 30%, whereas it was 33.9% in case of controlled plots under field condition. The disease index, which is a true measure of efficacy of any control measure, the disease index in case of these foliar diseases, disease index was 24%, 18%, which was, strain B was quite effective in this matter, whereas it was 43.17% in case of untreated control. Likewise, in case of leaf blast, the incidence reduced to 15% as compared to 15% in untreated control, and the index was even lower in case of leaf blast. Then sheath blight incidence was also assessed, and it was assessed by measuring the relative lesion height by calculating the relative lesion height, which was up to 44.5% with isolate A, 48.9% with B, and to the same range with C as compared to 65% in case of untreated control. In all these disease status, these isolates are able to manage the diseases to the extent of chemical fungicides, which are widely used for management of these diseases in the region. So these can replace, these can be better alternatives to these chemical pesticides. These are some of the symptoms of this rhizoctonia solanae, and this lesion height was calculated. Then seed, ultimately, after harvesting, the yield was taken. All the 3 strains were also effective in increasing the grain yield, which was increased up to under field condition, experimental condition, 25.33 quintal per hectare as compared to 12.03 quintal per hectare in case of controlled conditions. And it showed fairly good amount of increase over control up to 110%, 88 to 110% with these 3 strains. So grain yield and straw yield also increased with the application and biopriming of these strains. So it can be concluded from the study that application of plant growth promoting rhizobacteria, which inhibited the growth of the pathogens under laboratory conditions, resulted in growth promotion in vitro screening of in vitro conditions, and also increased the grain yield of the crop. Therefore, the application of plant growth promoting rhizobacteria needs to be validated and promoted as a competent strategy for sustainable plant protection and sustainable plant disease management. Thank you.
Request participants to ask few questions. The presentation is open for discussion.
Madam, one small doubt. Generally, tricyclozole is used for paddy blast, no? You used it against sheath blight. Generally, hexaconazole is used for sheath blight management and tricyclozole for paddy blast. So, can you please clarify?
So that was actually used for, blast disease and brown spot. And their effect on sheath blight was also assessed, because it might have, for this thing, any fungicide was not used.
No, but if you are using, you told lesion height of the, in case of reduction of the, when used at that time, you should use the specific fungicides for the sheath blight, no?
Sir, in case of sheath-based data was taken because the PGPR are also supposed to increase the plant growth. So if the plant is healthy, it will be not affected by the diseases. So the incidence of sheath blight was also there. So that data was also taken.
Okay, madam.
Oh. Madam, did you use the fungicide treatment as a seed treatment alone, or foliar application also did you use these fungicides?
Foliar spray was also done in the nursery. Just a minute, sir.
Hello.
While sowing the nursery, the seeds were treated with these rhizobacterial isolates, Pseudomonas, Trichoderma, Bacillus, and tricyclazole and Vitavex power also. And after transplanting, with the first appearance of the symptom, first spray was done. That was between 35 to 40 days after transplanting, and repeated after 15 days. So 2 sprays of all the fungicides and foliar spray was done.
Actually, bioagents, they are prophylactic. So before the disease comes, we should spray it, and fungicides can be used as a curative as well as when the initiation of the disease. Just for the benefit of the others participant, generally whenever we use the biocontrol agents, it is better to apply them as a prophylactic even before the disease starts. Otherwise, once the disease comes, the host pathogen interaction has happened, bioagents will not be of much use because they cannot go inside the plant system or do that. They are only preventing prophylactic in general.
So as a prophylactic measure, seed treatment was done.
Yes, no, while spraying also I am telling, generally the treatments you can plan in such, because we are going to recommend it as prophylactic. So better to plan in such a way that it is given before the disease comes. Our recommendation should go like that. Any other question?
Yes, sir. Hello.
Please.
Yes, sir. Sir, excuse me, madam. Can you let me know how you calculated the vigor index?
Vigor index?
Yes.
Oh, vigor index is a percentage of seedling length and germination percentage.
No, madam. As far as I know, what you have presented, that is vigor index 1, and there are 2 types of vigor index. One is vigor index 1,
which is calculated with the help of germination percentage and seedling length.
Yes. And another is vigor index 2, that is germination percentage into seedling dry weight. So you can mention there vigor index 1.
Yes.
Okay, that's fine. Thank you.
Thank you so much.
Any other questions? If there are no questions, shall we go to the next presentation?
Yes, sir, we can.
Helen Evan et al. Helen Evan or Epsiba Helen Evan.
Yes.
I request the next presenter. Slideshow mode. Can you change it to slideshow mode?
Go left corner.
Just top left corner. In top left corner.
Yes, your slide is visible now.
You can start. You can start. Helen Evan, you can start your presentation. You can start your presentation.
Helen, even do you hear us? Please start your presentation. Your slide is visible for us.
Helen Evan, are you able to hear what we speak? Helen Evan, you are not audible.
Helen, even if some problem is there, just you unshare and try again.
You can close your sharing. You can redo it later. Helen Evan. The next person is Kaku et al. Hello. Tage Kaku.
Yes, sir. Yes, sir.
You can share your presentation.
Yes, sir. Sir, is it visible?
Yeah, go to slideshow. It is visible. Go to slideshow.
Sure, sir. Okay, sir. Is it off fine, sir?
Yeah, fine. You can go on.
Continue.
Okay. Good afternoon, everyone. I welcome you all to my presentation for the East Zonal Symposium. My research entitled antagonistic potential of bioagent and botanical against banded leaf and sheath blight of maize caused by Rhizoctonia solani. So generally for the management of disease, we generally go for the chemical control because of its immediate results. But we forget to notice its impact on environment that cause undesirable changes in environment and also toxic effect on non-target organisms. So keeping in view the drawback of chemical control, the use of botanical agents and biocontrol agents and botanicals appears to be the best solution for the long-term and sustainability and efficient management of plant diseases. So considering the mentioned facts, a study entitled Antagonistic Potential of Bioagent and Botanical Against Banded Leaf and Sheath Blight of Maize caused by Rhizoctonia solani was carried out with the following objectives. First, to screen the efficacy of selected plant extract and bioagent against Rhizoctonia solani in vitro. Second, to evaluate the effect of selected treatments on banded leaf and sheath blight disease and yield of maize under field conditions. So now, for the introduction of the causal pathogen Rhizoctonia solani, it is a non-sporulating pathogen because of which it is kept under the order Agonomycetales, and it is soil-borne in nature. Because of its competitive saprophytic ability and high pathogenic potential of potentiality, this pathogen is persistent and a destructive plant pathogen. Now, in a disease cycle, the sclerotium serves as the primary inoculum and secondary spread of this disease occurs by contact of diseased leaves and sheath with healthy plants. Now, the genus Rhizoctonia is characterized by its hyphae. This is multinucleus septate, barrel-shaped and branching at right angles. Here it is the microscopic picture showing the mycelium of Rhizoctonia with right angle branches and slight constriction at the base of the lateral branches. Now, it is the systematic classification of pathogen according to the Ainsworth 1973. It is under the kingdom fungi, division Eumycota, subdivision Deuteromycotina, class Hypomycetes, family Agonomycetales, or we can also say Mycelia sterilum, family Agonomycetales, genus Rhizoctonia, and species Solani. Now, this is the typical symptoms of banded leaf and sheath blight of maize. The typical symptoms observed are small brown lesions or brown large continuous patches on leaf sheath and brown lesions on stalk as well as rotting of ears. This is the infection on sheath on the lower part of the plants near the soil. Here the symptom shows on both the leaf and the sheath. This is the infection on leaf, and as the infection during the disease advances, the infection also occurs with cough, and later stages we can see the sclerotium also on the infected parts. So now is the morphology of the pathogen. So the cultural characteristics is identified by the formation of sclerotial bodies abundantly on culture. So the pathogen was cultured on a artificial media, which was used with potato dextrose agar. After 2 days of incubation, the fluffy white mycelium growth was covered in the wall over the petri plate, and after 7 days, in 7 days culture, the sclerotia started to develop, and the last picture shows the picture after 14 days culture, where the whole the culture is filled with the sclerotial bodies, which is the characteristic features of this pathogen. Now, according to the first objective, to screen the efficacy of selected plant extracts and biocontrol agents Rhizoctonia solani in vitro, the biocontrol selected was Trichoderma hertzianum, and it was dual culture with Rhizoctonia solani, whereas after 72 hours of incubation, Trichoderma started to overgrow the pathogen colony, and in 144 hours, that is after 6 days, Trichoderma fully covered the pathogen colony and also totally inhibited sclerotial formation. Now this is the next the effect of botanicals on mycelial growth of Rhizoctonia solani. It was done by this evolution was done by using the technique, poison food technique. So here the botanicals were tested on 3 different concentrations, 5, 10, 15%, and the treated check was used was carbendazim. It's also at 3 different concentrations, 0.1, 0.2, and 0.3%. So this graph shows the performance of different botanicals. So the botanical selected was neem, garlic, tulsi, balm, onion, and the treated check carbendazim. Now it was as the concentration, this graph shows as the concentration increases, the efficiency of the botanicals also increases. Hence, the most efficiency of the botanicals was found at 15%, followed by 10, and then 5%. All the also for the significancy at 5 and 10%, all the treatments were found significant over control, and at 15%, neem and garlic and neem and tulsi showed non-significant to each other, but they significantly reduced from control. Now to the objective 2. To evaluate the effect of selected treatments on banded leaf and chip blight disease and yield of maize under field conditions. This experiment was accomplished during Kharif 2019 with simple randomized block design. 6 treatments, that is includes 1 biocontrol, that is Trichoderma herbacearum, and 5 botanicals with a treated check, that is carbendazim, and 1 untreated check control were used in this experiment, which were replicated 3 times. So for the botanicals, method of application was spraying, and for biocontrol agent, it was soil application incorporated with FIM, and the treated check, that is carbendazim, was seed treatment at the rate 0.2%. Now the different disease and yield parameters, that is plant height at 45, 55, 65 days after sowing, number of crops, test weight of grain yield were recorded, and for disease assessment, disease incidence and percent disease index at a 10 days interval after the spraying of the treatments were recorded. So all these parameters were recorded after the artificial inoculation of the pathogen at 35 days after sowing by inoculating this sorghum seed covered with mycelial growth of Rhizoctonia. Now this as the graph shows the performance of biocontrol agent as well as the botanicals on the disease incidence. Here the Trichoderma herbacearum showed the best result, and followed by the botanicals. And for the significancy, after first spraying, among the treatments, non-significancy was found between the neem and garlic, and after second spraying, it was non-significancy was found between the treatments Trichoderma neem and garlic, and after third spraying, the non-significancy was found between the treatment neem and garlic. Although all the treatments have been significantly reduced disease over control. Now this next graph shows the performance of botanical and biocontrol agents on percent disease index, that is on the disease severity. So for the significancy, after first spraying, among the treatments, all the treatment was found significant to each other. After second spraying, neem and garlic were found non-significant, and after third spraying, garlic and neem, garlic and tulsi and onion were found non-significant among each other, but although they were significantly reduced disease severity over control. Now this shows the plant height. In the plant height as well, for the significancy, after first spraying, among the treatment, non-significancy was found between neem and garlic and tulsi and onion. After second spraying, non-significancy was found among the treatments neem and garlic, and after the third spraying, non-significancy was found among the treatment is garlic and neem. And also this whole as a general, all the treatment has significantly increased the plant height over control. Now next was on the number of copes. In number of copes also, all the treatments have significantly increased the number of copes compared to the control. And for the significancy among the treatments was found between the treatments trichoderma and neem and garlic and tulsi, garlic and onion, and also bhang and onion. But all the treatment has significantly increased the number of copes over the control. Now, also for the test weight, the 100 seeds of the maize grains were weighted to calculate the test weight, and here also for the significancy among the treatments were found between trichoderma and neem and garlic and tulsi and garlic and onion, and they were found non-significant, and although significantly increased the taste weight over control. And also finally for the yield of grains after harvesting, the non-significancy among the treatments were found between the tulsi and the onion, but all the treatments has significantly increased the yield over control. Now for the summary, in dual culture technique for an in vitro evolution of biocontrol agent, which was used, the technique used was dual culture. Here it has proved that trichoderma has significantly been antagonistic against the rhizoctonia, and for the botanicals, maximum efficiency was found by garlic, followed by neem, tulsi, onion, and minimum by bhang. Now for the evolution of the effect of botanicals and biocontrol agent on banded leaf and sheet blight of disease and yield of maize under field condition. Here as well, the percent disease incidence, the best performance was shown by Trichoderma hertzianum, where it recorded the minimum disease incidence, which was superior among the treatments in all the data recorded at 10 days interval. And the next also for the percent disease index, the best result was shown by treatment Trichoderma hertzianum, where it recorded minimum disease incidence, and minimum was by the bhang in 3 different data recorded at 3 data recorded after 3 spraying at 10 days interval. So the maximum plant height was also recorded in Trichoderma as compared to control, followed by the botanicals. So there's maximum test weight of maize shown recorded by Trichoderma hertzianum, then followed by botanicals, and also the grain yield of maize recorded in treatment Trichoderma hertzianum as compared to control. Now, as for the conclusion, this study reveals that biocontrol agent and botanical not only reduce the disease severity index, but also recorded lower percent disease incidence in maize, with an increase in plant height, number of copes, test weight of grains, and yield by suppressing the fungus Rhizoctonia solani, causing banded leaf and sheet blight of maize. Thus, these eco-friendly treatments found as a better alternative than fungicides due to their fewer negative impacts on the environment and easy availability as well as economically feasible.
Presentation is open for discussion. Few questions, 2 questions. We are running behind schedule. 2 questions we can have. I have one question. You mentioned about the plant products.
Sir?
You mentioned about the plant products, na? They are coming next
Yes.
to buy. What will be the cost of cost for having this garlic or other plant products? What will be the cost benefit ratio? Because it will become more costly if I use it for larger
Yes, sir.
area.
Yes, sir.
It
Sir,
will.
for cost benefit, yes, sir. So for the cost benefit ratio also, the most efficient was found in trichoderma only. Then in botanicals, it was found for the garlic. And then
Because whenever we recommend garlic and other plant extracts, if you calculate the cost, it will be huge.
Yes, sir.
Any other question? I request participants to respond. If any other question is there, we can continue. Otherwise, we'll go to next speaker. Manas sir, shall we go to next speaker? Someshwar sir?
Yes, sir, we can go next.
Next speaker, please. The earlier Helen Evan, you have Helen Evan. Can you project you now? Can you share your presentation?
Yes, sir.
I think you are having two devices in the same room.
Sir, am I audible?
You are audible. You are having two connections for the same program in same room. That is why the sound is coming.
Yes,
Yeah.
yes.
You mute one contact, one connection in that.
Helen, please get sign off from one system, and which system you are using, in that only you get signed in.
Be the next presenter we will ask.
Yes, sir.
Please. Next presenter is
Rahula Prashant.
Sheikh Wajullah.
Started sharing, sir. Helen.
Yes, sir.
Yes. So we are getting echo of your voice. We could not hear your voice clearly.
Maybe you are using two devices. One you unmute, mute it. One you mute it. One you use for talking.
So I think she was trying from two systems at a time.
Yes.
Yeah, Mrs. Helen, listen. Are you listening? No, like you tell her slowly.
Then can we go for next presenter, sir?
Yes. Next presenter, please. Next
Rahula
presenter.
Prashant. Yeah, Rahula Prashant Kumar.
Ask her to answer.
Helen, just you please just unshare the screen.
Rahula Prashant Kumar.
He is in list. Rahula Prashant Kumar, you are on the list. Just please you start up your presentation by sharing. So otherwise, can we move on to the next, sir?
Next, Sheikh Wajidullah. Mr. Sheikh Wajidullah. Effect of thyme essential oil against leaf spot.
From till morning onwards, they have not responded, sir. I think next is that Srivalla Navina, probably they are present, sir.
Srivalla Navina,
Yes, sir.
are you ready?
Yes,
Are you
sir.
ready? Can
Yes,
you
sir.
share your presentation,
Yes, sir.
please?
Hello, I'm calling from Institute on IPS.
Sir, is my screen visible, sir?
Yes, make it as a slideshow.
Okay.
Sir?
Slideshow. Yes, yes.
Sir. Good afternoon, sir. Myself, Sirvella Navina. I will be going to present my seminar on topic screening of rice rhizobacteria against Xanthomonas oryzae, pathover oryzae, causing bacterial leaf blight of rice under in vitro condition. I'll give small introduction about rice, sir. Its scientific name is Oryza sativa and it belongs to family Poaceae. It is the second most important cereal food crop and it is a staple food for large part of the world population, sir. This is the data of the rice area production and productivity in the world, India and Bihar. Coming to the economic importance of the bacterial leaf blight, it is the most devastating disease during monsoon season and it is reported about 20 to 30% yield losses. It have also reported to reduce Asia's annual rice production by as much as 60%. The disease broke out in an epidemic form in Bihar in the year 1963. Coming to the symptomology of the bacterial leaf blight, it is caused by the Xanthomonas oryzae, pathover oryzae. The disease symptoms can be observed mostly during tillering stage of the crop. The symptoms also appear during seedling stage as a crescent phase. It is also known as wilting syndrome. In the symptoms also appear during leaf blight phase. Symptoms appear as leaves with the wavy yellow marginal necrosis covering the entire leaf. Isolation of the pathogen was done on the nutrient agar medium. On the nutrient agar medium, this pathogen appear as yellow colonies, and this is a pure culture of the Xanthomonas oryzae on medium. And the microscopic view of the Xanthomonas appear as a rod-shaped red color colonies, and it is a gram-negative bacterium. My objective is isolation and screening of the rhizobacteria against Xanthomonas oryzae, pathover oryzae. Isolation of the rhizobacteria was done from the soil and screening of the effective rhizobacteria against Xanthomonas oryzae, pathover oryzae. 1 gram of soil closely adhering to the rice plant was collected from the field and it was serially diluted on the nutrient agar and King's B medium with and the bacterial colonies were obtained. After incubation for the rhizobacterial growth on a nutrient agar and King's B medium, these colonies were found. A total of 14 different isolates were selected, and a single isolated colony from the petri plates of the King's B and nutrient medium was picked, and it was streaked on pre-sterilized nutrient agar slant, and it was incubated for about 24 to 48 hours to obtain the pure culture of the isolates. And screening of the rhizobacteria against the pathogen was done by the
Voice is not audible. Sirivella, your voice is not audible. Srivella, your voice is not audible.
I think she has unshared, sir. Srivella, just you have been unshared, I think. Please share your screen and start.
Sir?
Yeah.
Yes, continue.
Yes, sir. Among all the different isolates, Pseudomonas showed the highest in inhibition zone, sir. After 24 hours and 48 hours, it has shown 17.4 mm after 24 hours of incubation and 18.1 mm after 48 hours of incubation. Followed by the Pseudomonas, Bacillus showed the next highest inhibition zone, sir. It showed 15.7 mm after 24 hours and 17.1 mm after 48 hours. And these are the pictures depicting the inhibition zone produced by the rhizobacterial isolates against the Xanthomonas oryzae, Pathor oryzae. And this is a graphical representation of the inhibition zone produced by rhizobacteria against the Xanthomonas oryzae, Pathor oryzae after 24 and 48 hours of the incubation. And coming to the conclusion, among these 14 rhizobacterial isolates, 4 rhizobacterial isolates showed the antagonistic property towards the Xanthomonas oryzae, Pathor oryzae. Among them, Pseudomonas showed the highest inhibition zone of 18.1 mm, followed by the RRB 14 Bacillus of 17.1 mm, followed by the two different isolates of RRB 10 and 6. So it can be concluded that biocontrol being an eco-friendly and cost-effective strategy, it can be substituted for or integrated with chemicals to achieve a great level of protection and sustainable yields. There were also reports which have shown the Pseudomonas as an effective biocontrol agent. Thank you, sir.
Query, whenever you are making tabulation and statistical analysis,
yes, sir.
For the in vitro studies or the lab experiments,
yes, sir.
CD value you have given at 5%, na?
Sir?
Confidence. CD at 5% probability you have made. Generally, for all
Yes, sir.
in vitro
Yes, sir.
studies, it should be 1%.
Yes, sir.
But you are concluding with in vitro inhibition that it can be very effective.
Yes, sir.
How confident you are?
Sir, I had only done in lab in vitro conditions, sir. Didn't do any field experiment or any pot culture experiment, sir. In field condition, these isolates showed inhibition zone, sir. It is effective.
Pot culture did you do? Pot culture?
No, sir. No, sir.
Okay, I will ask one general question. Other than
Yes, sir.
this Pseudomonas and this fluorescent Pseudomonas, earlier in literature, is there any report of other biocontrol agents?
Yes, sir. There were also reports showing trichoderma is also effective against the Xanthomonas, sir. But I have collected only rhizobacteria, sir. So
Classical example for Xanthomonas. It is used in the forewarning of bacterial blight also.
yes, sir.
Can you remember any other old literature? Bacteriophages, running water in the irrigation water, bacteriophages, they used for forewarning the bacterial blight also. Very few literature is there. Biocontrol of bacterial blight in rice using the bacteriophages. Another way of having biocontrol for bacterial blight. Any other question? Any other questions, Someshwarji?
Yes, sir.
Please.
Actually, this presentation and second presentation, people are using only Pseudomonas and Bacillus. If they can use full, that is Pseudomonas, fluorescens or any other, not like that only genera. Hello, Srivella. Hello.
Even
Sir?
other isolates, they should do at least basic biochemical tests and tell what it is.
Yeah, yeah.
Whether it is a bacterial, bacillus, pseudomonas, or actinomyces, whatever it is, they should tell what it is. Because sometimes they may be opportunistic human pathogens. Burkholderia comes, we can.
Human pathogen as well as other plant pathogen also.
Yes, yes, yes. Any other question?
Sir, third presenter is ready now.
Third presenter is Rahula. Rahula,
Yes, sir. Hello.
if you are ready, you can share.
Yeah,
Rahula
yes.
Prashant Kumar.
Yes, sir. Sir, am I audible, sir?
Yes, you are audible.
Okay, sir. Sir, good evening, sir, to all the dignitaries who are present here. So my topic was the efficacy of bioagents and botanical extracts against false maternal rice caused by the Pistillaginodia virens. So, rice. Basically, we know the rice was the most important crop which is widely cultivated all over the world, and especially in India, it is a major staple food. As rice is caused by the lot of the diseases. False mud is the one of the before it was a minor. Nowadays, it has become the major disease, and it is caused by the Pistillaginodia virus. Nowadays, it is also causing a lot of. Hello, sir. Hello.
Rahula, you can continue. We are hearing you.
Yeah, sorry, sir. Actually, the disease incidence, basically it is in different states, it has shown the different types of the disease incidence. Like I have mentioned that the disease incidence in Punjab, it was a 10 to 20%, and whereas in case of Tamil Nadu, it was the 5 to 8%. So basically, however, the best treatments nowadays we are using is the fungicide, but as students, we are just saying that the alternative methods for natural methods like botanicals and the bioagents how we are using. So by keeping all the view mentioned facts, my title is the efficacy of bioagents and botanical extracts against false maternal rice caused by the Pistillaginodia virus. So my two objectives are the use of the only selected plant extracts against Pistillaginodia in lab conditions, whereas in case of field, I have used the bioagents as well as the certain botanicals to calculate the disease incidence in field conditions. So my methodology starts with the during isolation of this fungal organism. Basically, I have collected these muted balls in the field, and then as usual the basic method just clean through the sterilization and just I have done the cultivation in the petri plates, but I have used the PSA media instead of the PDA media because I have seen in a lot of research papers that potato sucrose agar media was proven the best growth media when compared with the potato distress agar media. So after the isolation and identification, then I have used this for the food poison technique, and for food poison technique I used in this plant extracts, the plant extracts with the lantana camera leaf extract, bulb extract of garlic, mentha leaf extract, neem leaf extract, and for the check I have used the propiconazole 25 ml concentrates, whereas I using the three types of concentrations 5, 10 and 50 so this is the method for the preparation of the botanicals. Actually equal ratio 1:1 weight by volume ratio I have taken and I made the stock solution. Then whereas this is the treatments which I have used in the field in field conditions. In field conditions I have used in the trichoderma at 5% and 10%, and other botanical same as used in the lab conditions. This is the disease incidence formula, and this is the particular showing the radial mycelial growth by using by the certain uh, uh, in food poisoning technique, and these are the results where in botanicals name leaf extract has shown the best when compared with the other botanicals, whereas the mycelium, where I have seen the less mycelial growth in case of neem extract at 15%, then later in 10%, and later in the 5%. And so in percentage inhibition also, neem leaf extract has shown the 71.87% percent inhibition at 15%. And whereas in case of 10%, it has shown the 67.50%, whereas in case of 5%, it has shown the 61.25%. So overall the percent inhibition means highest percent inhibition has been shown in the neem leaf extract. And this is the graph showing in among the different treatments, whereas we can see that treatment 4, which is the neem leaf extract, as showing the maximum percent inhibition, whereas in treatment 5 it is used as a cyclophosphamide, and this is in concentration wise, whereas in C3, which is a 15% concentration, it has shown the maximum inhibition. Whereas in case of fail, the maximum disease incidence less disease incidence was reported in the neem at the rate of the 10%, where in case of trichoderma at the 10%, it has been shown the less disease incidence, that is 32%. So this is the graph showing the disease incidence. So overall my conclusion is that actually for this false metal of the disease, we have to need much more research work in the field conditions, because as it is for only one season, we can't get enough data, because we have to apply more botanicals, and we have to see the different growth parameters in case of the rice to get the data perfectly. As I have taken only the disease intensity, it has to be required disease intensity also to calculate the exact data, the act of the botanicals against this disease. And however, this rice pulse is also in lab, it is very somewhat tough to when compared with the other pathogens, which it is, as it has required the selective media. Yes, sir. I have done, sir.
Any questions from participants? Someshwarji?
So, hello, Rahul.
Yes, sir.
You have used trichoderma.
Yes, sir. Yes, sir.
10%.
Yes, sir. 5% and 10%, sir.
10%, what does it mean?
So 10%, sir.
Here you can see, yes. A table.
Project this table.
Total table.
Yeah.
Yeah. Yes. Well, 10%, trichoderma viridis 10%. What does it mean?
So means I have using this as 10 means 100 grams in 1 liter water, sir.
Too high, na? 100 grams per liter is too high. So for how much spray volume is there for 1 acre? For 1 acre, how much volume you will use, then how much quantity of trichoderma is required?
So it may be huge enough, sir.
It is very, very huge number.
Yes, sir. It is maybe in a lot of kgs, sir.
Well, it is basic information we should know before going to apply, that
yes,
what concentration, what pH you have adding that.
Yes, sir.
What was the population of trichoderma before spraying? Did you check?
No, sir.
Actually, what is recommended dose is 2 into 10 to the power 6 per gram of the formulation.
Yes, sir.
And the recommended dose is very less only. You are using 100 grams per liter is very huge number.
Yeah, sure, sir.
And cost also it increases. And the other propiconazole, you are using 0.2%.
Yes, sir.
Isn't it?
Yes, sir.
0.1% generally recommendation. So if you calculate the volume of the material used is very high.
And in this experiment, which variety you have used?
Shiraz-Dan one, sir. Shiraz-Dan one.
Shiraz-Dan, whether it is hybrid or high-yielding variety?
Sir?
Whether it is hybrid variety or normal?
Means it is our college variety, sir, which is more susceptible for this disease.
That's what, if you are doing any experiment for pathological studies, you should use susceptible variety.
Yeah, it is more susceptible, sir. That's the reason I have used
And
this variety.
not to extract what time you have sprayed, when you have sprayed in the crop.
Means in July I have transplanted, sir. In last year in 2019.
I am asking what stage of plant growth you have sprayed.
Sir. At booting stage, I have sprayed this artificial inoculation. I have done, sir. So then, after the inoculation, then after 4-5 days, then I started spraying this treatment, sir.
Actually, biocontrol agents should be prophylactic.
Yes, sir. Yeah, just in before lecture, I have learned, sir. I have seen sooner, sir.
Because they have to stop the host pathogen interaction. They should colonize, survive there. Then only it works.
Yes, sir. Yes, sir. Got it, sir.
Any other question?
Prasad,
Yes, sir.
you have done dual culture in lab condition.
Sir, I have done pure culture, but I have planned to send my culture to ITCC, but I was not able to send due to pandemic, sir.
You have seen the effect of trichotoma in clay.
Trichodoma, I have made, yes, sir.
In vivo result is asking. Sir is asking about in vivo result.
In vivo, sir?
In vitro.
In vitro, I have not done.
Vitro.
In vitro, I have not done, sir. Dual culture, I have not done, sir. Only botanicals I have used, sir.
Then how you assured that the effectivity of this against this pathogen, Osteochondria?
Yes. Actually, I was able to do lab work, but at that I have not able to done, sir. Then, but I have, sir, go through the lot of discussions in previous papers, sir.
This commercial formulation, did you use?
No, sir. Commercial formulation, I have to check, sir, once again, sir.
Any other question from participants?
Sir, actually, what happened? I have tested this Trichoderma viridi. Actually, it covered the whole inoculum of Osteochondria within very few days.
Okay. But it is mycoparasitism.
But the thing is, when I re-isolated that Osteochondria inoculum and allow it to grow in the fresh culture, it actually freshly it grows. That means trichoderma has not done any effect on the inoculum.
It is only covering, growing on that, not preventing it.
Yes.
Maybe it has to be applied as a preventive spray well in advance.
Yeah.
Maybe here some bacterial biocontrol agents may be of much use, I feel.
Yes. Sir.
Thank you, Rahula.
Okay, thank you so much.
Next presentation.
So it is the last presentation. Helen, are you ready?
Helen.
Good evening, sir.
Yeah, you are.
Helen, you just start slideshow. Enable editing. Just use slideshow.
Am I visible and audible, sir?
Yes,
you are audible and the slides are visible.
This is also we are listening. Now you enable editing and go to slide mode and start.
Okay, sir.
You just choose that enable editing.
Okay.
And then go to slideshow.
Now is it okay, sir?
No. You go the top yellow line. There is one tab is there. Enable editing.
Second line is there, na protected view. In that enable editing, you just click it.
Okay.
In that yellow highlight is there, na?
Sir, protected I couldn't. View. Yes, sir. I couldn't see any.
Just you select that enable editing.
Yes.
You select that enable editing. Just
Yes, you click
yellow
it.
highlight is there.
Yes, you are right pointed. Rightly pointed. You go there again.
Here, sir.
You have right pointed. You go to the enable editing. The word enable editing is there, no? Protected view lines.
Just you are having tabs now, home, insert, design. To that below you are having one yellow highlight, protected view. You are having home, insert, design. These tabs you are having no.
Sir.
Just another IPS logo.
Yes, sir.
In that, just enable editing. You just click that enable editing. Just
Above.
that place. Click.
Down. Next line. Down. In that above.
No, now you
protected view. You read at the end of that line. Protected view. This file originated from an internet location and might be. That thing you click.
Then you go to slideshow.
Down.
Okay.
Yeah, that's all. Okay.
Okay, sir.
Okay, you give okay.
Okay. Sorry for the inconvenience.
No.
Good evening, everyone, respected dignitaries and all the members sitting over there, the organizing committee and all. I'm extending my hearty welcome. And my name is R. Hepzibah. Today I'm going to present about antifungal effect of Trichoderma species and botanicals against southern corn leaf blight disease of maize. This disease is also called maize leaf blight, and here on the screen you can see actually the causal organism on the left hand side and the Trichoderma viridae is on the right hand side. And this introduction, maize is actually the third most important crop after rice and wheat in rice in area and production, but in productivity it is the first. Actually it is called the potential crop because per square meter or per square area it would give more yield than other crops, so it is called a potential crop. And it is well grown in Karnataka, Andhra Pradesh, Maharashtra, Uttar Pradesh, Bihar, Rajasthan, Madhya Pradesh and Punjab. Actually currently this research has been done in Uttar Pradesh, and Uttar Pradesh highly promotes the environment in Uttar Pradesh is highly favorable for the particular disease which I am talking now. The warm, humid nature, the subtropical condition. Actually, Uttar Pradesh has all those conditions, so the disease is very severe here, and this region is obviously an endemic region. So the productivity from this Uttar Pradesh from this particular area is actually low when compared to the world, India, and other states. So actually Uttar Pradesh is large in its area, but actually productivity is quite less because it promotes many other diseases also because of its temperature and other factors, biotic and abiotic factors. And maize production in the world from India is the 6th position. And by the disease, this disease was first reported in 1970 in Southern America, and it is mainly due to that time it has become an epidemic because of the monoculture of cytoplasmic male sterile corn hybrids they have been using, because detasseling and all takes time, so they have used hybrid corns, but that has affected in another way, and it has become an susceptible thing for this particular pathogen, and it has resulted into an epidemic. And in India it was reported first by Munjal and Kapoor from Malda, West Bengal. And in this particular pathogen there are 3 races by now has been identified, and race T is the one which have actually caused that epidemic in 1970 in Southern America, and race T has been very virulent in its nature, and it's when it causes an epidemic there will be a huge loss. And race O presently is being in India, and race T is seed-borne, and race O is actually exists in crop debris and all, but it has no placement for like seed borne and all. And yield losses from this particular disease to India are from 9.7 to 11%, because favorable conditions it provides for this particular disease, the warm and humid temperature, and 15 to 30 degrees temperature is much important for this disease. And this disease actually in symptoms, it starts from the lower leaves and then it goes to the upper leaves, and it starts as small brown dots, but they later merge with each other and develops as large patches, but they are constricted to the veins. And about this particular bipolaris medis, and this is a this is not this is race specific actually. The T race is seed borne and other races have no basement, like no haven't been said like seed borne, and they have been airborne, and the conidia spread by wind and air splashes. And my objectives have been usage of biocontrol agents like Trichoderma viridae and Trichoderma hertzianum and some of the botanicals against this pathogen, both in vitro, in vivo, and I have also worked out the cost benefit ratio of these treatments. And in vivo, this is the pure culture, and this is the culture, and these are the conidia and conidiophores. And in the in vitro, I have done poison food technique for the botanicals and dual culture technique for biocontrol agents. And these are the treatments which I have used in vitro, and I have references also. And all the treatments in lab conditions I have done in 5, 10, and 15%. And propiconazole is the fungicide. Actually, I have taken it as a treated check, but not to check its efficiency. We already know, because fungicides are much more efficient than all these things. But right now my objective is to promote and also support this biocontrol and botanicals. So I was using this, and these are my in vivo treatments. And in vivo, I have used Trichoderma viridae as I have mass multiplied it on FYM and applied to the plots at the rate of 100 grams per meter square 1 week before sowing as a prophylactic measure. And Trichoderma hertzianum also I have used as seed treatment or seed dressing at the rate of 20 grams per kg seed. And rest all I have done as foliar spray at the rate of 10%. I have used 10% because it is cost efficient. If it's becoming 15%, it's not being economically standard for the farmers, and if it's 5%, it is not efficient. So I have used 10%. And propaganol I have used at the rate of 0.1% as foliar spray. And the first image I have mass multiplied the pathogen on the sorghum grains, and the second one is I have inoculated the field with the sorghum grains. Third one is mass multiplication of Trichoderma on FYM, and I have applied that FYM in the field too. And the downside images are the plant extracts and the spraying of treatments in fields 36 and 46 days after sowing, and seed treatment with Trichoderma hertzianum. This is 5 and scale disease. And results in vitro, I have found that both of the trichoderma are actually promoting inhibition. So but trichoderma viridae being more efficient than trichoderma just 5%. And botanicals that have in vitro and the inhibition percent, and neem and the botanicals have been actually not significant to each other in their efficiency, and rest all are the efficient ones. And in vitro, onion has been more efficient than rest all, and then the garlic.
There is some problem.
Sir,
Please continue.
Sir, from this slide.
Continue, please.
These are the in vitro images of all the botanicals and biocontrol agents. And
Come to the main part.
and this is the in vivo observations, and I have worked out disease incidence percent disease index. Before first spray I have taken one reading, and rest two sprays are done after the second spray. I have taken before first spray one reading, because some treatments I have done while sowing and some are foliar sprays, so I have worked out before spray also. And these are the graphs representing, and this is the 1,000 grain weight and weight of cobs, and in all aspects Trichoderma viride and Herzianum has been proved the best among all the botanicals and biocontrol agents. And they have already promoted growth parameters also of the plant, not only managing the disease but also the health of the plant has been promoted, and the grain yield have been observed here. And these are the cost benefit ratios I have worked out of all the treatments. And since Trichoderma viride has been used much more, the cost benefit ratio actually it is giving less benefit, because it is being used in more amount than Herzianum, but in efficiency Trichoderma viride has been proved the best one. And these are the conclusions. Trichoderma viride and Herzianum not only managed the disease but also promoted number of the cobs, weight of the test weight of grains, and yield, suppressing the disease actually. And eco-friendly treatments can also be a better alternative to the chemicals, since chemicals are more efficient, we know, but it is good to actually produce a sustainable agricultural thing. So we are promoting biocontrol agents and botanicals. Thank you so much, sir.
Presentation is open for discussion. I request participants to ask questions.
Hey, Helen, am I audible?
Yes, sir.
One interesting question.
Yes, sir.
Can you tell us that what is that T in rest T?
Sir, I didn't understand.
What is the T? T, race T you have mentioned?
Where?
Race T. T for tiger.
Yeah,
Race T.
no, sir, no. Race T is T is sterile. No, sir. Texas male sterile cytoplasm.
Yeah. And there is a toxin, do you know, which this
Yes,
fungus
sir.
produces?
I forgot.
It is T toxin.
Yes, sir. T toxin. Sorry. Yes, sir.
Race T toxin,
Yes, sir.
Texas.
Cochlea bolus produces T toxin.
Yeah. Thank you. All
Thank
the
you.
best.
Okay.
Any questions? Any other questions? Someshwarji, you have some questions?
Okay, sir, no problem.
No, this is the last presentation, na?
Yeah.
Another one, sir. Actually, whether he is present or not. Sheikh Wajidullah. Okay, sir.
I want to make a few comments. In general, most of them have used the word Trichoderma viride, but we have already established that the viride is present in the northern hemisphere of the world, where it can grow at a low temperature only. The culture which is having the accumulated conidia, that is finely warted conidia, is that can produce chlamydospores and that can grow at high temperature is Trichoderma asperellum. So most of the cultures what we have in our country, with which appear to be Trichoderma viride with morphological characters, but they are asperellum. But most of the presentations they are using the word viride. Viride cannot grow at higher temperature. In our country, soil temperature is very
Sir.
Oh, yes, some problem in network. Okay. Yes, yes.
Connection.
Hello.
Sir, Commissioner, sir, can you carry on?
Yeah, yeah, yeah. Actually, that this session, all presenters, they have presented well their paper, but thing is that so far the biocontrol aspect and the plant extract, we have to. Okay, okay, no problem. Yes, sir. So, keeping in view of biocontrol agent and plant extract, we have to take very careful, and before setting any experiment in the either field or either lab, we have to take care, and accordingly the result what we are presenting, it should be as per their efficacy. So I want to make point that whatever the biocontrol agent or isolate they are keeping, they should use full name, either genus and species, because sometimes what happens, the same genus have some biocontrol agent as well as plant pathogens also. And regarding plant extract, it is very unstable. So whenever we are using for plant disease management, it should be used very freshly, and the efficacy also, it will go very early. So we have to take care about that. Okay, okay, sir. Sriram, sir. Hello.
Sir, actually, sir, till now not connected, sir.
Oh.
You can give your comments.
Sir is not there.
Yeah,
Hello.
Dr. Bhagat.
Sir, actually, sir could not be connected, sir. I don't know what is the.
Okay, okay. Then if sir is not there, then I would like to thank all presenters who have presented in this session, and I would also thank to the organizers, the Zonal President, Dr. Mukherjee, and Zonal Councillor, Dr. Bag, and the Secretary of our Indian Phytobiological Society, Dr. Ravin Gogoi. And with this, we can conclude this session, I think. Dr. Bag?
Yes. Dr. Samas sir, thank you for accepting our invitations and for a nice presentations. And at the very last moment, due to some unfortunate.
No, he is connected.
Okay. Sir.
If sir is there, let him.
Just he is connecting, sir. He is getting connected.
Okay, we can wait.
Wait for a while.
Yeah.
Sridham, sir, you have been connected, sir. Just you can unmute yourself.
There was some problem in the net, sir. It got disconnected, Wi-Fi.
Okay, sir.
Okay, sir.
I wanted to mention about that aspergillum and viridae. So most of the cultures what they have used must be aspergillum. so they can recheck it. That is what I wanted to mention. And some of the presentations, they have used the bacterial antagonists. They have not identified it. Most probably, they have to do at least basic biochemical characters so that we can rule out some of the agents which are opportunistic human pathogens or not good plant pathogens we can avoid. I request Someshwarji to offer his comments. Someshwar.
Sir, before I have already made.
Okay. I thank organizers. I thank you very much, sir, for giving the opportunity.
Sir, on behalf of IPS and also on behalf of ICR NRRI, we extend our heartfelt thanks for both of you, sir, for sparing your valuable times in doing all the thing in a very nice manner, sir.
Thank you. Thank you very much. Thank you, sir. Thank you very much, sir.
Thank you, everybody. Thank you. So today we are ending with this our session, technical session 2. So we are requested all to be present tomorrow at 9:30. We will start our technical session 3. Okay, at 9:30. Dr. Gagoi sir.
Yes.
This session is over.
Yeah, you have completed today's session very successfully. For tomorrow also, speakers are ready, I hope.
Yes, they're ready.
Who will be the first speaker tomorrow morning?
Tomorrow, actually, session number 3, actually integrated pest management. Dr. K.K. Mondal and Dr. P.B. Bhattacharya, they will co-chair the sessions. They will be also invited for giving their talks also.
Okay, sir. Okay, I think both the speakers are ready to start, huh?
Yeah.
Okay. Then today we can close.
Yes.
We can have host, no? They can close. We can end up. So you can end the meeting.
Sir, you can end the meeting.